A very good morning and a warm welcome to our guests. We will be beginning with our oration sessions. For the first session, may I please invite Dr. J.V. Tilavari, Dr. M.P. Sharma, and Dr. Philip Abraham to please chair this session. I will also take the honor to invite Dr. Rakesh Agarwal, the first speaker. Thank you.
Good morning, friends. Thank you for inviting me to chair this session. It gives me great pleasure to say a few words about this legendary physician, P.N. Chutani, who lived up to the age of 81. ISG meeting was held in 1964. The Punjab Chief Minister at that time told Dr. Chutani and other doctors, get the best doctor, Indian doctor, no matter where he comes from, no matter where he lives. And that's where my story comes in when I met Dr. Chutani in London in 1976. And I was working in University College Hospital, London, as a lecturer. And he met me, he said, what are you doing here? Come home. In Punjabi, he said, "Aithe ki karda main, chal kar wapas." And I joined the institute in 1977. PGI was inaugurated by Pandit Nehru. Sorry, what are you doing? Not going along with my speech. Anyway, inaugurated by Pandit Jawaharlal Nehru. Next one. Dr. Chutani was from Gujranwala in Pakistan. That's where Rishi Sunak, his parents, and his grandparents come from. There seems to be something in Gujranwala which produced great people. He went to Lahore for his medical training and then finally settled in Chandigarh. Next one, please. In this college in Lahore, there was Nobel laureates who studied, prime ministers, film actors, and Olympic hockey players. Next. He also loved sports, was champion in swimming, tennis, and golf. Next. He also loved farming. Next one, please. Chutani has great communication skill. He could talk to anybody no matter what the rank was. And here you see he's talking with Pandit Nehru at one-to-one level. Next one. And he could also entertain officially like he's entertaining Indira Gandhi at that time. Next. He also has a soft corner for the ladies who enjoyed feminine company, although he was bachelor and apparently a brahmachari as well. Next one. Dr. Chutani received Padma Bhushan Award from President of India. And not only that, next, he was on behalf of Physicians for Prevention of Nuclear War. He also received Nobel Prize, signatory to Nobel Prize 1985. Next. Finally, when he died, he gave all his wealth which he accumulated to this great institute, PGI. Thank you. Now I invite Rakesh Agarwal.
I will invite Dr. Rakesh Agarwal to give this oration. Dr. Agarwal, I am fortunate that he was my undergraduate student. Then he did MD in the All India Institute of Medical Sciences. Then he did his DM from PGI Chandigarh. He was professor of gastroenterology at Sanjay Gandhi Postgraduate Institute, and after that he has shifted over to PGI JIPMER as the director. He is an avid teacher, scholar, and a keen research worker. His work already he has been working on viral hepatitis for a long time, and you will be hearing again on viral hepatitis today. Dr. Rakesh Agarwal is a very good, clinician, great clinician and researcher and a teacher. Dr. Rakesh Agarwal, please.
Thank you, Professor Tilavari and Professor Sharma, for the kind words. Ladies and gentlemen, good morning. I am grateful to the Indian Society of Gastroenterology of all its members for the love and affection that they have given me over the 35 years that I have been a member of this august family. And particularly today, when the society has asked me to deliver this prestigious oration of the society. Professor P.N. Chutani, as has been said, was a very able physician, teacher, administrator, and was responsible for the postgraduate institute at Chandigarh being what it is. I had the occasion of seeing him several times, including as a teenager when I had enteric fever. However, unfortunately, I did not have the occasion to work with him. However, I today have the honor of being able to deliver an oration named after him, and that to me is a big honor. Over the last 45 years, when I joined medicine as a medical student, I've had the pleasure of working at 4 institutions, all of them counted among the very best institutions in the country. Firstly, I started off my career as a medical student and then as a resident at All India Institute of Medical Sciences in New Delhi. Moved to PGI Chandigarh for my training in gastroenterology. Thereafter, a long period of 28 years at SGPGI in Lucknow, except for a brief 2-year period when I moved back as a faculty member at All India Institute of Medical Sciences, and now for last 4 years at Pondicherry. Each of these institutions has given me a lot. I have learned a lot from each of them, and it has been a fulfilling journey over these years. So I think it's time for me today to reflect at what has changed, how viral hepatitis has moved along in the nearly 35 years that I have been in the field of viral gastroenterology, and share with you a little bits, a small part at places that I have been able to play in what has changed in viral hepatitis over the years. So let me take you back to 1989. This is a paper. At that time, we knew viral hepatitis as hepatitis A, B, and for those of you who are relatively fresh, something called as non-A, non-B hepatitis, NA-NBH, which was further thought to have 2 subvarieties, parenterally transmitted and enterically transmitted. 2 months before I finished my fellowship in gastroenterology, appeared this paper in Science, where the agent causing parenterally transmitted non-A, non-B hepatitis had been sequenced. Its genome had been sequenced, and based on that, a small protein had been made from which we could get a definitive diagnosis by detecting antibodies using immunoassays. And this was possible because newer techniques in molecular biology had just become available, and hepatitis C was the first pathogen to be identified at the genomic level using these new techniques. So we had a test, and that means we could think of doing further things with this virus. So that's when I finished my gastroenterology training. And voila, the next year, the same technology had been used for another virus causing hepatitis, the so-called enterically transmitted non-A, non-B hepatitis. So the field was ripe. I was finishing my gastroenterology training right at that time, and that's when this paper appeared in 1990 March, and July 1990 was when I moved to SGPGI, a new institution that had just been set up. My love, association, whatever you call it, tryst with viral hepatitis was very fortuitous. A few months after I had joined at Lucknow, early 1991, this is a headline in the newspaper, and my mentor, Dr. Nayak, said, "Hey Rakesh, let's go, let's investigate what's happening." And that changed the course of my journey. The basic shoe leather epidemiology was what we did. We did a sample survey in the city, covering about 800 houses, in which 3,600 people lived, and nearly 4% of them had icteric symptomatic hepatitis. And when we extrapolated this to the entire population of the city at that time, 2.1 million, it was apparent that it was a huge outbreak affecting about 80,000 people. A great learning in epidemiology. My training in MBBS had inculcated a love for public health in me, and I think that's where I went back to my primary love. Number of deaths was small, but a disproportionately large number of deaths were in pregnant women. Very reminiscent of what had been already described from India, from the Kashmir Valley by Dr. Kuru a few years ago. And Dr. Kuru has been a role model, was a role model even before I joined there for his work, and continues to be so even today. When we looked at this further, we found that of the parts of the city shown here with dotted areas, which received water from the river, which was on the right side of the city, we looked at certain areas within these area which received water from the river, and the frequency of hepatitis in these 4 blocks that we studied was much higher than that in some of the blocks that we studied but were not receiving water from the river, indicating that it was the water supply that was responsible. And we could find out that there was contamination of water by a sewage drain that opened just upstream of the water pumping station for the city, establishing that this was the cause of the outbreak. The outbreak was bimodal, and it seemed that there had been water contamination from this drain always, but on one particular day in, sorry, I can't get the cursor to move. Okay, on one particular day in February 1991, there had been failure of chlorination, and that was responsible for the large peak. More importantly, what we had, as I described to you in 1990, the genome of hepatitis E had been described. I showed you that paper. And here we were within a year using those sequences to do polymerase chain reaction PCR and showing from the stool of patients in an outbreak that they were positive for hepatitis E virus RNA, and that this outbreak was caused by hepatitis E. We continued surveillance in the city over the next 1 year and were able to show the previous outbreaks had been unimodal. There had been a short period of contamination of water as shown on the left. Can I get a mouse maybe? And that was followed by a unimodal peak. Here we had a bimodal peak, and that would suggest that there could be intrahepatic, there could be person-to-person transmission, intrafamilial transmission. However, it was clear that the second peak was not related to person-to-person transmission, but was related to continued contamination of water. And as contamination was controlled, water chlorination was introduced, the outbreak came back, and this was an important finding that person-to-person transmission is infrequent. Why? We today know that the inoculum needed to cause icteric hepatitis is fairly large, and person-to-person contact cannot lead to transmission of such a large amount of virus, and that's why possibly this happens. Subclinical infections can occur, but clinical infections don't occur, and I'll talk about that more later. Since then, over the last 20 to 25 years, if you read literature on hepatitis C, a lot has changed. At that time, it was thought that this was a disease of South Asia and Africa, but today we know that there is indigenous transmission of hepatitis E in Europe and North America, where water contamination is infrequent. All enteric diseases are infrequent. However, the source there is very different, and it has been found that this virus circulates in several animal species, in particular pigs, deer, and wild boars. And the infection that occurs in those countries is because of zoonotic transmission from these animals to humans, particularly when undercooked meat derived from these animals who have the infection is eaten. Also, in those countries, chronic HIV infection, especially among organ transplant recipients, the immunosuppressed people, has been noticed. Blood transfusion also appears to be an important cause of transmission. However, most of the infections there are subclinical and only a small proportion are clinical. In those countries, the genotype most prevalent is genotype 3. We initially did not realize that there were other genotypes, and that's what has come out in the last 20 years. So, we have worked on how our disease is different from that in Europe, and I'll talk about some of that. Also, the important point is we have outbreaks that occur at some times. In between, we see very little activity. And where is the reservoir? Is this that some people continue to excrete virus, a bit like we have chronic typhoid carriers? Are there subclinical infections and the infection continues in the community, and sometimes a lot of them are subclinical, and then sometime the disease becomes much more symptomatic, what is known for polio, for instance, or is it a zoonotic source? So we addressed first the question whether there could be prolonged excretion. In an outbreak in Lucknow, we took 20 people and we tested their stools and sera serially collecting at multiple time points, and we could show red dots indicating presence of the virus and blue indicating absence, that viremia and viral excretion don't go on beyond 4 weeks. So prolonged excretion is not the reservoir for this infection. Something that we couldn't do here, but I had the opportunity to do through a year and a half that I spent at US CDC in an animal model in cynomolgus macaques, which are a very small primate. We inoculated primates with different doses of the virus, and we found that if you gave them a large dose, 10 to the power 4 cynoinfectious doses, then one can see that liver enzymes go up after about 3 weeks. There is viral excretion, there is viremia, which starts in the incubation period and persists for some time and then disappears with development of antibodies. However, if the infection occurs with a very small amount of the virus, minimal dose needed to cause infection, I'm not showing you the results for doses in between, one can see that there is no ALT elevation, there is no liver injury, but there is viremia and viral excretion, and actually that viremia and viral excretion appear to last somewhat longer in subclinical infection than in clinical infection. So it's entirely possible, and today we know that it does happen, that humans, when they get a small inoculum, they get subclinical infection, they pass it infection on to the others, and that is how the virus persists in the community. This is also illustrated by this study that we did in Lucknow, where on alternate days we collected sewage specimens from a big sewer in Lucknow and looked for the presence of HEV RNA in sewage. And we could see around the year, not just during the monsoons when we see clinical disease, one could detect HEV RNA in the sewage all through the year, indicating that there are people, even though there is no outbreak in the city, that the virus is continuously present in the population and is being excreted. The next issue was whether we have zoonotic infection in India. So we collected specimens from patients with acute hepatitis E, sporadic hepatitis E, from several parts of the country, nearly 100 specimens, and we sequenced them, and we found all of them belong to genotype 1. They did not cluster with the other subtypes of genotype 1, genotype 2, 3 or 4, indicating that our infections are all due to genotype 1 virus and the other genotypes don't exist in India. On the other hand, when we looked at animals in India, different species, and we tested fairly large numbers, we found antibodies to hepatitis E virus were nearly universal in them. We were able to get genomic sequences of the virus from pigs, not from the other species. However, the interesting part was all the human specimens from India clustered with genotype 1 and the swine sequences clustered with genotype 4, indicating that the two infections, even though genotype 4 is prevalent among animal species in India, it does not cause human infection. Whereas if you look at other countries where we see zoonotic transmission, such as European countries, Japan, USA, the swine as well as human specimens are all belonging to genotype 3. So our epidemiology is very different from those countries. Also, we looked at in 2 studies large number of people who have undergone renal transplant, and we found no evidence of chronic hepatitis E in them. So what we know as hepatitis E today in the world has two totally different faces. What we see is genotype 1 disease. Reservoir is in humans, so there is human to human transmission, mainly through water, sometimes through food. Outbreaks of our variable size. Hepatitis E forms a large proportion of our sporadic hepatitis. Most of the cases occur in young who do not have coexistent diseases, and this virus causes a very high mortality in pregnant women. Chronic infection does not occur. Whereas that in Europe and US is a different disease caused by genotype 3 virus. Reservoir is in animals. Transmission occurs by eating animal meat, zoonotic transmission. There are no outbreaks. Hepatitis E is responsible for a very small proportion of sporadic hepatitis. Most of the cases are among elderly, those with coexistent diseases, in particular those who are immunosuppressed. There are no data on pregnant women, and chronic infections occur. On the other hand, what we see in India is also something very different, and that is if you take patients with liver cirrhosis who have had recent worsening, the so-called acute on chronic liver disease, we found that nearly half of them had evidence of recent hepatitis E virus infection. Whereas if you take stable cirrhotics, in them recent hepatitis E virus infection is very infrequent, indicating that this virus not only causes acute hepatitis but is a major cause of acute and chronic liver disease. I'm not going to go into details of this, but we have looked at immune responses to hepatitis E virus, particularly the innate and cellular immune responses. These studies showed that in acute hepatitis E, in the peripheral blood, NK cells and NK T cells are reduced in number, but those that are present have a very high degree of activation, suggesting that the viral infection activates these cells, and also that these cells get sequestered in the liver, and that's why their number in the peripheral circulation is reduced at that time. The peripheral blood mononuclear cells also show proliferative responses in response to HEV capsid protein and also smaller peptides of this protein, indicating that there is an activation of cellular immune responses, though their role still remains unclear. In fulminant hepatitis C, one actually sees that the production of interferon gamma is reduced and the regulatory T cells are increased. Also, there are some differences in liver biopsies when we study gene expression using microarray compared to other forms of fulminant hepatitis. And these findings suggest that though they are not sure, that a lot of viral damage may not be mediated by immune response but might be mediated by the virus itself. Things have moved on. We today have a vaccine which has been tried in a large trial in China. A large trial of more than 100,000 people randomized to receive hepatitis E vaccine or a placebo, that is hepatitis B vaccine, a good follow-up, and we can see a high degree of protective efficacy of around 95%. But has that changed things? Not really. The vaccine is approved in China and Pakistan, no other country in the world. The issues remain that we know that it protects for about 4.5 years, that's the maximum data. Beyond that, we don't know. The groups where it should be of particular use, pregnant women who have a high mortality because of this infection, or those with pre-existing chronic liver disease where the infection could cause acute and chronic liver disease. We don't know whether the virus really would induce antibodies and whether it would protect. The vaccine is costly, and we don't have data to study the cost effectiveness of the vaccine. How much is the disease burden? What proportion of people in a city of 100,000 would actually get hepatitis E over a year? Does it make sense to vaccinate? What is the cost benefit ratio? And of course, I have talked about high cost, so funding remains a problem. The role in interpreting outbreaks, whether it would be useful and given during outbreak, remains unclear. And that's when you realize as you grow older, it's not important just to have tools to treat or prevent. What matters is a much bigger picture. You need to look at a much bigger picture. And as also you age, you move into a role where you play a role in policy making and deciding health policies. So as a part of a WHO team, I've been involved more recently in trying to find out, though it's not a formal study, whether this could interrupt hepatitis C outbreaks. So in South Sudan, there is in a protection of civilians camp, which has existed since 2014 with about 100,000 population, there is an ongoing outbreak of hepatitis E. So you can see that in wilderness there is this camp very well laid out in which about 100,000 people live and there is an outbreak occurring. That's the kind of place it is. And in the last few months, hepatitis E vaccine has been introduced, not in a randomized manner, but covering everybody. And one is trying to see whether the outbreak will be taken care of. One plays only a minor part in doing such things, but I think it's very satisfying. And I'll come back to hepatitis E vaccine in my last two slides. So I talked about enterically transmitted non-A non-B hepatitis. What has happened in what was then parenterally transmitted non-A non-B hepatitis? 1990, the virus was discovered. We already knew, even though we did not know the genomic sequences, that there were interferon work to some extent with a response rate of about 7%. Over the years, we have had improving therapies and now we have DAAs with more than 90% response. So we are thinking of elimination of hepatitis C. Once DAA came in, and even though we had generic DAAs, there was a bit of a reluctance to use them in a public health program because it wasn't clear whether doing so is beneficial from a public health point of view. And that's where we played a role. We did some disease modeling, and based on that we were able to show that if we treat patients with hepatitis C using generic DAAs at the prices that were operative in India, not today, at that time, today it's even lower, we could, if we treat people over their lifetime, people would gain anywhere from 3.5 to 6 years, depending on which genotype in virus they had and whether they had F0 to F3 disease or they had pre-existing cirrhosis, with benefit being more in those who already had cirrhosis. Treatment would be useful. Not only would treatment add to the life years, but actually, because that would save money by not having to treat cirrhosis and HCC later, it would actually save money over a lifetime, anywhere from about 1,000 to 2,000 US dollar per person treated. So in the first year, in the beginning you spend money, but as you come along, you find that over a period of years, you recoup the money that you have spent, and people showing that it was actually beneficial to spend public funds to treat people using generic DAAs in India and in other countries where drugs were available at similar prices. Once Velpatasvir became available, we did a similar analysis comparing the pangenotypic drugs with pangenotypic drugs, and we worked out that if the relative cost of pangenotype treatment was a particular value and that of genotype specific drugs was a particular value, what range of prices would make actually the pangenotypic treatment to be cost saving. And we showed that actually it was better to use a pangenotypic treatment because then it does away with the need for genotype analysis. And this work we did actually as a part of a WHO collaborating center for viral hepatitis, which we were fortunate to have at our department in SGPJ, and the WHO collaborating center continues. Dr. Amit Goyal is the current head after I left. And these findings had an impact on WHO recommending that everybody, irrespective of whether they had cirrhosis or not, till that the time WHO had been saying that only those with cirrhosis should be treated. Our findings led to their saying that everybody should be treated irrespective of whether they had cirrhosis or not, and the same thing has been taken up in national guidelines. A little bit of hepatitis B. So what matters is the public health adoption of measures that we have. So we had hepatitis B vaccination till 2000. We used to think we need to screen mothers and then vaccinate their babies. We did an analysis showing that universal hepatitis B immunization for hepatitis B in India was extremely useful and very cost effective and actually cost saving, and that was changed into advocacy document. So we helped prepare this for WHO, and that led to introduction of HPV vaccination in the country in 2003, initially as a pilot project, and in 2007 extended to the entire country. For hepatitis A, I didn't work on it much, but recently had a role as chair of the WHO's hepatitis A vaccine working group, and this is what was published about 2 months ago. So what the working group was able to achieve was to change the WHO recommendation that for a public health program for hepatitis A vaccine, instead of 2 doses, only 1 dose should be used. Many countries that could not afford a 2-dose schedule can afford a 1-dose schedule, and we are certain that it works as well as a 2-dose schedule, and that should lead to increased take up of hepatitis A vaccination around the globe, and I'm particularly proud to have been able to play this role. Coming back to hepatitis E vaccine and my recent work, we have set up an Indian hepatitis E virus vaccine initiative. It's an industry academia partnership funded by Department of Biotechnology, BIRAC. So we are Zydus Healthcare has developed a vaccine, and we are helping them in their trial, and in particular to study cellular immune responses. So the company is doing its trial in 6 sites. They will look at development of antibodies to hepatitis E virus and its clinical safety. At the same time, we are piggybacking on that to study cellular immune responses and to show in an in vitro experiment that antibodies are protective. So I think life's come a full circle, starting from epidemiological studies, lab studies, and going on to public health impact. In all this, I've been helped by a lot of people. My 2 main mentors, Dr. Subhash R. Nayak, who's been a member of the society and whom many of you know, and Dr. Krzysztof Krobzinski at CDC, who was my mentor in lab sciences. There have been several collaborators in different places. I've talked about different institutions, but many of these collaborators have come from small places, municipal corporations, water supply workers, etc. A lot of students and lab colleagues. In particular, I must mention Dr. Amit Goyal at SGPGA Lucknow, with whom I have done a lot of work, and also several funding agencies that have funded some of these studies. With that, I come to an end of my talk, and again, let me thank the society for all the love and affection that I have received from the society over the years. Thank you.
Thank you, Rakesh.
Sir, can I request you to felicitate Dr. Rakesh? Shall we have a...
Okay.
Congratulations. Thank you, sir. Thank you. Please support, sir. For the next oration session, can I please invite Dr. K.R. Palaniswamy, Dr. Y.K. Joshi, Dr. Ejaz Habib, Dr. Subhash Nepalia to please chair this session.
Good morning. I'm honored to speak few words about Professor C.M. Habibullah. Professor C.M. Habibullah did his DM gastroenterology from PGI Chandigarh. He was one of the first to receive DM degree in India. And Professor Habibullah served as a head of Department of Gastroenterology, Osmania Medical College, and subsequently served as a principal in the same college. And then later he became the director of ORC Hospital and Research Center. And he had a lot of interest in the research, especially he had a keen interest in hepatology, and he had special interest in hepatocyte transplantation. He did a lot of work in hepatocyte transplantation. And he had, the Professor Habibullah published more than 150 papers, both in national and international. And Professor Habibullah published, he had chapters in many textbooks, and he served as a president ISG between 1988 and 89. And Professor Habibullah received several awards, among which important ones are received FP Auntie a Lifetime Achievement Award and prestigious BC Roy Award and Padma Shri in 1992. Professor Habibullah's son Muhammad Aizaz Habib is with us, and with this I'll hand over the mic to Dr. Joshi.
I just will add few words about Dr. Habibullah. I had a long association with him for several years when I was at AIMS. Dr. Habibullah means always smiling face with very soft Hyderabadi Hindi and always asking everybody, "Arey, tu kaisa hai, tu kaisa hai, tu kaisa hai." He was a very frequent visitor of our department in gastroenterology in AIMS. And I am really lucky that I had a long association with him and I learned so many things about him. The oration will be delivered by Dr. Ajit Sood, who is head of the Department of Gastroenterology at Ludhiana Medical College. He is the person who has done lot of work on inflammatory bowel disease in this country and also has established Crohn's and Colitis Foundation of India. Dr. Sood, please.
Thank you, sir. Professor Jitmur Muhammad Habibullah was a great teacher, astute clinician, and a researcher par excellence. These are the three main responsibilities of a medical professional, and he excelled in all three. I'd like to thank Indian Society of Gastroenterology for awarding me this important oration. For Professor Habibullah's work, he was deservingly awarded Padma Shri, and as mentioned, he was the first DM student to pass out from a prestigious institute, PGI Chandigarh, and established the Department of Gastroenterology at Osmania Institute, University Hyderabad, and it took it to great heights. It's a great honor for me to speak for this oration. I'm going to talk on journey of inflammatory bowel disease in India. All journeys begin with small baby steps, and there are some secret destinations. Same has been the story of IBD. When we started working on IBD in 1991, we were quite unsure of the disease. Now, in last 30 years, we, the gastroenterologists in India, have started to understand the disease much better. Historically, when we think of India, the infections and communicable diseases have been the main disease burden in a country, but things have changed over the past 30 years, and the disease epidemiology has changed. When we look at these two maps, the color has changed from red to blue. So what does this mean? For assessing the disease epidemiology, there is a tool called as epidemiological transition level, which is calculated by the ratio of disability adjusted life years due to communicable diseases plus maternal, neonatal and nutritional diseases as a group, and the ratio is, and it's divided by non-communicable diseases and injuries together. What is depicted in the map on the left side, which is a red color, means the ratio is more than 1.7, and very small part of India in 1990 had a blue color. The rest of the country was red or brown or orange. Means the ratio was more than 1.7 of infectious oblique non-infectious diseases. Thankfully, the scene has changed altogether in 2016 when we see the whole of the country has changed from red to blue, indicating that the ETL ratio is now less than 3 in the areas where it's all blue. So there is a changing epidemiology of diseases, and we know that communicable diseases or non-communicable diseases are becoming more common. And for a gastroenterologist, the major non-communicable disease is IBD as a group.
So that is what I am going to focus on today. The first case of IBD in the world was reported in 1859 from the West, and it took nearly 100 years for us in India to report a first case of IBD. But in the new industrialized countries, once IBD has started, it's rapidly increasing. We in 2003 reported the prevalence and the incidence of ulcerative colitis, and we found that the incidence is more than 6 and the prevalence of 44.3 per 100,000 population. Though these values were not same as what has been reported in the West and the US, but it became very evident that we are not far behind. When we look at the total disease burden, you will be surprised to know that if you look at the population of India, which may be more than 130 millions now, the disease burden of IBD in India may be just less to only US in the world. Though when we look at the incidence and the prevalence rates, they are still may be lower, but the total disease burden in India is not that far behind. We established this first national IBD registry, and very interesting figures came out of it, that among the IBD patients, more than half of the ulcerative colitis patients were reported from North, while more than 50% of the patients of Crohn's were reported from South. And when we look at the disease severity, in North what we have noticed that the disease, majority of the patients had moderate to severe disease, while in in East the disease had milder course. Coming to Crohn's, the Crohn's in South was noticed to be starting in a younger age group, had mainly ileal disease, fistulizing disease, including perianal fistulizing disease was more often reported from South, and the disease severity was moderate to severe, while in North the disease was mainly in the ileum, and the disease course was reported to be mild to moderate. So there is a change. North has a UC predominance and South has a Crohn's predominance. The trends of IBD at a center, over the years what we have noticed that the E1 disease, means proctitis, has been becoming more common. Maybe we have started to pick up more cases of proctitis now once we have better awareness. And for Crohn's, we have started seeing more of ileal disease. And when we look at the trends, the trend for UC is somewhat plateauing, and the R square is 0.5, while trend for Crohn's show that the Crohn's is still on the rise. So IBD is increasing in our country, and there is a regional and intra-regional variability. But then the important point is that why is there heterogeneity? When we go back into the history, we look at the genetic makeup of Indians. The ethno-linguistic groups in India descend from a mixture of two divergent ancestral populations, ancestral North Indians and ancestral South Indians. The ancestral North Indians were related to West Eurasians, means people of Central Asia, Middle East, Caucasians and Europe, while South Indians ancestral were related to indigenous Andaman Islanders. But the populations did not remain confined to themselves, and there was a mix-up, as it was expected and natural. The important point is that what is the era when the mixing happened? When we look at this started somewhere around 5,000 years, and we look at the original ancestral North Indian and South Indian, they appear to be somewhat like, if we depict it like red and blue, and what we have seen that over the years with the mixture, now this is the current Indians, and if you see, none of us now is totally red or totally blue. So what does this mean? That over the past 2000 to 4000 years ago, there has been a mixing up of the populations, and we are no more akin on, we are no much totally alike or similar to the ancestral population. And the mixing has not happened recently, this happened 2000 to 4000 years ago. And another study, the same, if we look at the ethnically and linguistically diverse populations in India, if we see how much is the genetic diversity among them, and this principal component analysis shows that all populations in India, they come under this cluster, suggesting that the genetic divergence is low, except in a race which is Tibeto-Burman or some of the non-Indians. So North Indians and South Indians now genetically are not so much different as they originally were. Now coming to genetics of IBD, we know that Asians do not have a similar genetic predisposition as in West. NOD2 gene, which we didn't find very common in our IBD population, was reported from South as well. And NOD2 is not commonly reported in Asian countries. And one of our studies identified few HLA independent novel genes, and these findings do help us in understanding the disease at a molecular level better. In spite of the fact that genetic predisposition to Indians is there, but that doesn't explain the exponential rise of IBD in a country. So what does this mean? When we look at the pathogenesis of IBD, it's not just the genes. The environmental factors also play a very important role. IBD will occur only if there is a environmental factors interaction with the host in a person who is genetically susceptible. So here starts the story that with urbanization of societies, the various environmental factors are coming into play. The major being the diet, the use of drugs, hygiene, microbial exposures. When we look at the diet in the West, which is traditionally thought to be inflammatory diet or not so healthy diet, it comprises of red meat and sugars and high omega 6, while traditionally Indian diet has been based on comprises of vegetables and fruits and a lot of fiber. But it's the things are changing. When we look at the fast food industry in our country, there's a very rapid growth. The trends of in the food processing trades is again disturbing, that there's a rapid rise, and India's per capita soft drink consumption is very high, and you'll be surprised to know that India is the second largest manufacturer of sugary beverages in the world. So these are alarming figures, because this kind of food is not considered healthy food. The sales of antibiotic in India is rising consistently. When you look at the sanitation, when we go back to 2000, only 10% of the population in a country had a safely managed sanitation facility. It has improved quite a bit. It is more than 40%, but still it is below the world standard. This report of inflammatory bowel disease and Indian perspective, they reported the known and the unknowns and the challenges. And one of the statement was that IBD in India may be milder, and I'm sure that we all will agree to disagree on this, that IBD is no more very mild in a country, and because the disease has evolved from mild to severe disease, with evolution comes a revolution, and there have been a change or availability of more and more drugs for treatment of IBD. It started with sulfasalazine and corticosteroids. In early 90s, if we knew the two names, sulfasalazine and corticosteroids, we were competent enough to treat IBD. And now time is going to come when there will be so many drugs, and it will be difficult to make a right choice out of different drugs. I'll just go through that. What is the information, what are the studies which have been reported from our country. We have data on use of thiopurines from our country. We have NUDT variants data in our country. I'm just going through the important studies which have been carried out in our country. We have information on use of biologics in different conditions, acute severe ulcerative colitis and fistulizing disease. And this is a miracle study, because for us, acute severe ulcerative colitis is the main indication for use of biologics, but very few of them actually go on maintenance therapy. But that's not so disturbing, as what we have noticed that a combination of azathioprine and sulfasalazine maintains remission in fairly good number of patients, though relapses do happen. facilities of TDMs is available now. Coming to FMT, which is of great interest to us, we started doing FMTs in 2015, and we first reported its efficacy in steroid-dependent ulcerative colitis, and we found very satisfactory results. And we reported this RCT on its role in maintenance of remission in ulcerative colitis. It's more acceptable to patients than medical healthcare providers, but side effect profile is not very disturbing. We have predictors of response to FMT that young patients, E2 disease or Mayo Endo 2 are a group of patients who are more likely to respond. And I must say, a very important study has been recently reported from AIIMS, Dr. Vineet's group, that FMT with anti-inflammatory diet does much better job than pharmacotherapy alone, and they emphasize the role of anti-inflammatory diet. Not only it's useful in induction of remission, but maintenance of remission as well. And this study was a runner-up as the best paper in Gut 2022. We have reported that the malnutrition in patients with ulcerative colitis is not far less as compared to malnutrition what is traditionally thought to occur in patients with Crohn's. And so we should be looking for evidence of malnutrition in UC also. Exclusive enteral nutrition has been studied, and its role has been found to be there for severe Crohn's. Even some patients who had no response to biologics, who didn't respond to biologics, they did well on EEN. And again, Dr. Vineet's group has reported and they documented the efficacy of EEN in management of acute severe ulcerative colitis. So what is required? That there was a need that all IBDologists in country, they come to a platform to share their experiences, and that's how Writers Crohn's Foundation was established, and under the presidency of Professor Kocher. And I'm so thankful to Professor Kocher and Dr. Ajay, who started with, all of us together started as a team to establish Writers Crohn's Foundation India. And recently the 7th National IBD Meet was held, and CCF India has been involved in laying the guidelines for diet and inflammatory bowel disease. Asian Working Group was hosted in India, and CCF India has laid down the guidelines for the commonly used drugs, 5-ASA and thiopurines. We are now planned a meeting for guidelines for FMT and the use of corticosteroids in IBD. We are focusing on the commonly used drugs in our country. So IBD is established in India and is actually increasing. Can we do something to prevent it? Normally when we say prevention, we talk of prevention for communicable diseases. But is it time to talk of prevention for non-communicable diseases like IBD? For prevention of a disease, the roadmap is to first to understand the preclinical events. I'm saying preclinical events before the disease actually starts. If we know the disease pathways, we can target those pathways for invention of new therapeutic targets. And once we have the targets and we have the pathogenesis and the various steps, we can identify the biomarkers which can predict and which can help in prevention of a disease. Coming to IBD, we don't know what causes IBD. We know genetic predisposition is required. We know genetic predisposition alone is not adequate. We don't know which environmental factors is the cause, but we know that there are various environmental factors, and I think 2 minutes are there. For that, we have to go back into a hypothesis that IBD occurs in 4 phases. The first phase is preclinical phase, in which patients or people are at risk. Means there is an interaction between the environmental factors and the host, and the person is genetically susceptible, but there is no inflammation. The T cells have not been initiated. The innate and the adaptive immunity system is still not into play. There's no dysregulation of the immune system. While in stage 2, when the disease begins, the changes what happens is that there is a disease initiation, there is a dysregulated immune system, there is dysbiosis and there is a change in the gut barrier. And this gets expanded, what is seen in disease expansion phase, and then comes the phase of diagnosis, in which there is a microscopic followed by macroscopic changes, what we see endoscopically as hyperemia or ulcers in the colon. I like to say that the diagnosis is not based at the beginning. Even for a diagnosis, a critical level of inflammation has to be there before we can successfully make a diagnosis. Now, at stage 1 or people at risk, can we do prevention? What is happening currently that our focus has been on treatment, and what we can do is a tertiary prevention by modifying the disease course and preventing the disability and improving the quality of life. But what is required is a desired paradigm for prevention at the primary level, which means we should modify the environmental factors. And what is the action plan of CCF India is that we should focus, as I mentioned, on the modifiable risk factors, intervention to improve the diet, and this is finished, right? Just 30 seconds. And promote judicious use of antibiotics and NSAIDs, and emphasize on the role of breastfeeding and regular physical activity. Identify at-risk populations, means the siblings and the family members of patients with diabetes, and they need very close monitoring. And we should pass on the information at a young level of schools, colleges, and general public. And we should focus on therapies suitable to our country. And there is a need for more of health insurance coverage and early detection. If we wish to have early detection, we need to improve our awareness among not only gastroenterologists but among pediatricians, physicians, including family physicians, surgeons, and young gastroenterologists. And we need to define the goals of therapy very well, because just symptomatic relief is not the goal of treatment. We want a mucosal healing. So how has been the journey of IBD in our country? Then in 1960, when the first case was reported, 80s, we were quite happy in knowing that I know the treatment of IBD because there were only 2 drugs and infections have been the main brunt of the disease. In 90s, we started using more of thiopurines as the main immunosuppressive drugs. In 2000 onwards, biologics became available, but the use has been quite limited. Till now, we have been talking more of ulcerative colitis, at least in North, but Crohn's have started appearing. In 10 years earlier or 20 years earlier, the Crohn's have started becoming more common, and now we know it is quite established. And this decade, we are going to focus on how to prevent, if we can prevent IBD in a country. Thank you very much.
Can I request the chairpersons to please felicitate Dr. Ajit Sood? There will be another.
Those who do not know, he is son of Dr.
Thank you. Thank you, chairpersons, and congratulations, sir. Thank you. For the next oration session, can I please invite Dr. S.K. Acharya, Dr. B.S. Ramakrishna, Dr. Vivek Saraswat, and Dr. Manish Tandon to please be the chairpersons for this session.
Who had been teachers of many of us. Professor M.P. Sarma is sitting here, Professor D.K. Bhargav is sitting, and with Professor M.P. Sarma, Professor D.K. Bhargav, Professor Rakesh Tandon, and Professor B.N. Tandon, who established Department of Gastroenterology at AIIMS. And these were the initial mentors who trained multitude of gastroenterologists, whom we were seeing all over, who are leading in various fields of gastroenterology. Now, I think I am a small one to introduce this great man, And luckily for us, his son, Manish Tandon, who is also a gastroenterologist, and now is an established gastroenterologist in the United States of America, has been kind enough to be amongst us, sitting on the right side of me. And I thought it is apt, and it is probably in a right perspective that Manish, being a gastroenterologist and is being son of a great gastroenterologist, is the right person to introduce his father, who had been the doyen in this country as well as globally. Manish, will you please introduce your father?
Thank you to the ISG Organizer Committee for arranging this oration lecture in honor of my father, Dr. Rakesh Tandon. It's an honor for me to be present over here today, especially to see all his old friends, his colleagues, and thanks for regaling me with all the stories from his old days. My father, as you know, is a renowned gastroenterologist, pancreatologist, and he was a dedicated teacher and mentor to a number of young gastroenterologists who, as Dr. Charya mentioned, have since excelled in their fields, and it's a pleasure to see them in leadership roles all over the world, no matter where you travel now. Dad graduated from KGMC in '66, and he went on to do his studies in the US, but then he came back to India, the country that he loved and was so devoted to, and joined AIIMS in '71. He was there as the head of the GI department over there from 91 until he retired from AIIMS in 2003. Did his PhD over there, and he was a mentor to a lot of physicians that I see over here now. He then, after his retirement, was at PSRI over there in Delhi, where he's head of the GI division over there and the medical director over there until his unfortunate demise in 2020. He did extensive research, as you guys know, in biliary and pancreatic diseases. He was passionate about it, and he was one of the first to introduce therapeutic ERCPs, papillotomies, which is one of my passions, in the early 70s in India now. He published extensively and has over 300 research articles, 87 chapters in various books, and 6 books, including one that he wrote in Hindi that he was actually very proud of. He's been recognized with numerous awards and over his distinguished career. Just to name a few of them, he did receive the Lifetime Achievement Award from AIIMS, ISG, and the Crohn's Colitis Foundation. He received the very prestigious Dr. B.C. Roy Eminent Teacher Award from the President of India in 2000. He was recognized internationally by being awarded as a Fellow of the Royal College of Physicians at Edinburgh, which is quite an honor. And he was also named a Master of the World Gastroenterology from the World Congress of Gastroenterology. He co-founded over here the Indian Pancreas Club, served as the President and Secretary General of the ISG itself, and helped organize a number of very important and very well-attended international meetings in India, some of which I was fortunate to attend, in conjunction with the Indian Association of Pancreas, the AG, and the WGO. He was also on the editorial board of a number of journals, including being the editor-in-chief of the Journal of Gastroenterology and Hepatology. So my father passed on the morning of August 3rd, 2020, doing what he always loved to do, which was really taking care of his patients. He passed peacefully, knowing that his legacy and his contributions to GI will live on with his colleagues, students, and future GI leaders in the field. My father always considered the ISG and its members, which is yourselves, as extended family. And I sincerely thank the ISG for the love and honor that you have shown to my dad, Dr. Rakesh Tandon. Thank you very much. Thank you, Dr. Charya.
Thank you, Dr. Manish. And I think we now move to the first Dr. Rakesh Tandon Memorial Oration, which will be delivered by Dr. Sandeep Laktakia. It's a proud moment for me to introduce Sandeep. Sandeep was one of our very bright and distinguished alumni from the Sanjay Gandhi Postgraduate Institute, where he worked in the mid-90s. He is presently the Director of Endoscopy and EUS at the prestigious Asian Institute of Gastroenterology in Hyderabad, and the current President of the Society of Gastrointestinal Endoscopy of India. He has also been the Secretary General of SGEI and Vice Chairman of the International Committee of the American Society of GI Endoscopy and Steering Committee member of the Asian EUS Group. With 170 original publications in national and international journals, authorship of several guidelines on pancreatobiliary EUS and endoscopy in Asia and Europe, and there's a long list of other achievements which I will, for want of time, not go through. He is an appropriate person to develop the first oration in the memory of Professor Rakesh Tandon, who was one of the fathers of pancreatology in India. Sandeep.
Thank you, sir. Good morning, everybody. It's a proud privilege and an honor to be awarded the oration in name of Professor R.K. Tandon, as rightly mentioned, who has been a father figure in field of endoscopy, gastroenterology, especially pancreatobiliary endoscopy. I'll be speaking on the topic of EUS guided intervention in pancreatic diseases. How far have we come? Before I go forward, I was taught by two of the students of Professor R.K. Tandon, Dr. Gauradas Chaudhary and Dr. Professor Vivek Saraswat. Professor Saraswat is on the dais, and it's a proud privilege again for me to be chaired by my teacher. Moving forward, I'll subdivide this talk in fewer sections. And endoscopic management of pancreatic fluid collections comes first, because this is the most common problem that we face in the pancreatic disorders. There are several publications which have come from India, especially from AIIMS and other centers. Pramod Garg's there. So management of pancreatic fluid collection has evolved over the last few decades. In the early 90s to early 2000s, we transited from management of these collections by surgery to endoscopic drainage. Initially, we used duodenoscopes or forwarding endoscopes to drain the pancreatic fluid collections, which transited to EUS guided drainage for two simple reasons. One, we could drain non-bulging collections, as well as we could avoid the complications of bleed or perforation associated with pure endoscopic drainage. So EUS helped overcome this. Also, we transited from two-scope technique to a single-scope technique, which meant that first we would do EUS, mark a point of entry, take out the scope, put a duodenoscope or a forwarding scope, and then enter the same point of entry. We changed over to purely EUS-guided method of drainage. So one-scope procedure, and Peter Willman was for the first authors who published this on endoscopic drainage using EUS in 1998. In late 2000, the last 15 years or so, things have evolved further. We moved from multiple strep drainage to a single strep drainage, which I'll mention later. We also have started using more of metal stents. Plastic stents have been the norm for many years. They're still there, they haven't gone away. But metal stents have made the job easier and probably safer. They started with tubular, fully covered metal stents, which are meant for biliary interventions, and then the same were placed through the EUS scope into the pancreatic fluid collections. But they had their own challenges, and then came the dumbbell-shaped, fully covered metal stents, of which the LAMS or the lumen opposing metal stents are the most popular. Nevertheless, the biflange metal stents are equally safe and effective. I'll touch upon these in my subsequent slides. Ken Binmoller, whom I consider as a guru of EUS, not only by myself, by most of us young gastroenterologists who are practicing EUS, he designed this stent LAMS. In 2010, this was proposed as a method of drainage of not only pancreatic fluid collection, but several collections around the stomach or the upper GI tract. And Takao Itoi and they together published the first clinical report in 2012 in GI endoscopy. Not only they drained pseudocyst, as they were called, all collections at that time were called pseudocyst. There was no difference of Vaughan and pseudocyst at that time, including gallbladder also was drained by the stent. Very soon after, almost simultaneously, Dr. Nageshwar Reddy, as Nagi is called, is sitting right in front, designed a similar kind of a stent, but this was made by a Taiwan company. The Nagi stent has been very popular in US, not in US, sorry, in India and in Europe, whereas LAMS is very popular in US predominantly and partly in Europe as well. Soon after, there was also a publication, clinical report, which came in GI endoscopy from the Japanese group using Nagi stent. There were also some other transitions happening same time when these stents were evolving. The definition changed of all pancreatic collections which are called as pseudocysts now are not defined as two distinct entities as pure pseudocysts which have fluid in them and walled off necrosis which have fluid as well as various amount of solid component in them. These were the first initial publications, and that made the difference of how we should approach these collections. Very importantly, silently carbon dioxide entered into the endoscopy suits, and we transitioned from routine air as an insufflating agent in GI endoscopy to carbon dioxide. This made our job easier. This prevented perforation, and even if it happened, it sealed off in its own. We learned from surgeon and we carried on endoscopically. Now, third space people are doing it. Fantastic job using carbon dioxide, and we do all now. So, differentiation of pseudocyst from walled off necrosis is essential before we treat these patients who have pancreatic fluid collections. Then came the concept of direct endoscopic necrosectomy, where we take the endoscope directly inside the walled off necrosis and take out the solid debris using various instruments. This den is actually tiger inside a den. Managing this, you can have several adverse events, and one must be wary about it when you deal with the problem. Keeping this in mind, we published in 2017 in GI endoscopy, endoscopic step-up approach using dedicated biflange metal stent to show that it reduces the need for direct necrosectomy in walled off necrosis. We used Nagi stent in this publication. We selected patients, 205 patients of walled off necrosis, and when we placed Nagi stent, 3/4 of them improved at the index procedure. They did not require any further interventions. Pseudocysts, almost 100% would improve with these procedures, but walled off necrosis are a different problem altogether. If those patients who continue to have problems, we went up to do declogging, which I'll allude in the next slide. That is cleaning up the stent. If that didn't work and the debris was adherent to the back wall of the cyst, we went ahead and placed a nasocystic tube and irrigated vigorously using normal saline and sometimes hydrogen peroxide to clean up the cyst. If that didn't work, we went inside the tiger's den and did necrosectomy. Using this approach, or the step-up approach, our incremental success increased from 75% to almost 96.5%, and this was quite gratifying. Let me show you the steps. Some of you may have seen this. This is declogging. The stent is blocked by a debris, and use a simple teasing device. Here a snare is being used to drag it out. This itself opens up the lumen and allows the stent to function better. But if the debris is adherent and not coming out with whatever methods we try, we place a nasocystic tube and irrigate with saline and hydrogen peroxide. If that doesn't work, then we do necrosectomy as mentioned earlier, where we take several devices one after the other inside the cavity, including the endoscope, irrigate, catch the devices using snares, baskets, nets, and there are new devices are now getting available in this field. Moving forward, Nagi also became hot, just like Axios became hot Axios. Quite similar in technology, where high wattage is used to directly puncture these walled off necrosis, and place the lamp. So in one step, you can do the entire procedure. So you don't have to do multiple steps like with Axios or hot Axios, if I can say that. Changing gears, disconnected pancreatic duct is another major problem that we face in patients who have pancreatic necrosis and thereafter. In disconnected, the upstream pancreas is broken away from the downstream pancreas and the juice does not flow to the papilla. And if variable segment of pancreas is functioning upstream, pancreatic juice continues to get produced and can cause problems. This can occur in the head or the proximal body or the distal body. The length of disconnected segment can be small or large. This has been approached by various authors differently. The initial publication about 15 years back from Jacques Davier's unit, Mariana reported that if you drain a pancreatic fluid collection, that time they were called pseudocysts, with plastic stent and leave them permanently, even if they have disconnected duct, they will never have a reoccurrence at least 1 year or more. Sham, in recent publication 2 years back in CGH, showed that same concept can also be used for metal stents, but since metal stents have to be removed, we can replace a metal stent with a plastic stent, and if they are successfully replaced, then these patients will not develop a recurrent pancreatic fluid collections. The problem is not so easy. So how do we manage disconnected duct in the era of metal stents? Jahangir, my younger colleague, worked on this and published in a high impact journal, Gut, Impact of Disconnected Pancreatic Duct on Recurrence of Pancreatic Fluid Collections and New Onset Diabetes. Do we finally have an answer? So we followed all our patients who had walled-off necrosis, trained by Nagy stent, did an anatomy of the pancreas using MRCP and even ERCP at the time of stent removal, and we differentiated them into a disconnected duct or a leak or a normal PD, and then we followed them up. We found that 3/4 of our patients, 75% of patients who had necrosis drained had disconnected pancreatic duct. Does it mean that all these 3/4 of them should have some intervention done? We also compared their MRCP and ERCP, as most had the paired imaging, and we found that MRCP actually overestimates disconnected duct and underestimates leak. And that's why probably secretin is better than pure simple MRCP, but we don't have it there. These were some of the images of the patients who were included. There you can see the disconnection here, and the upstream duct is dilated. This is at the position where the lamps is located. And this is an ERCP of the same patient where you can see the disconnection happening. So when we followed all these patients of disconnected duct and looked at the reoccurrence of pancreatic fluid collection, we found that only 10% had a symptomatic fluid collections. 13% had oral collections, but 10% of the oral patients had the symptomatic ones. Even these, if left alone and small, spontaneously resolved, or very few required re-intervention. 6.6 of the overall group required intervention, and that could also be managed endoscopically. At this stage, we could place a plastic and leave it permanently. Not only that, we also found that these patients also had a new onset diabetes. We know acute pancreatitis can lead to diabetes, but these patients were the ones who are the higher risk. Disconnections in the proximal segment had a higher chance of diabetes developing in them compared to those in the distal part. So conclusions that we draw from this study is that disconnected duct is very frequent after wound drainage. However, overall symptomatic recurrent collection is only in 10% or so. Out of that, only 6% required drainage. Some implications: risk of reoccurrence is almost negligible in those who have normal pancreatic duct. So if you have a normal duct, leave them alone, do nothing. But those who have a recurrence, 50% of them are asymptomatic and often subside spontaneously and then can be managed conservatively. But we wanted to change the accepted practice, that is, of keeping the plastic stent indefinitely or exchanging metal with a plastic at the time of removal. This may not be true. Hence, a simple approach of just removing the metal stent and leaving them without any plastic stent is what we suggested. Radhika from our group then published further, went deeper into this, an impact of transmural plastic stent on reoccurrence of pancreatic fluid collection after metal stent removal in DPD. This was an RCT which got published last year, 2022 in endoscopy. In this, we have compared a group of patients who had, at the time of removal of stent, either a plastic stent reimplanted or no plastic stent placed in the collection. Same type of group that we had. The follow-up was similar. So one group had stent inside, a plastic stent placed at the time of removal, and then they were followed carefully at 3 months, 6, 9 months, and 12 months. And we looked at the recurrence of symptoms. So this was the randomization. Drained by Nagy stent, they come with disconnected duct. At that time, they were included. And one group received a plastic stent after removal of metal. Other group just had no stent replaced. And we looked at the reoccurrence of collections at 3 months, but the secondary outcomes were also at 6 and 12 months. The technical success of replacing these stents, adverse event, and stent migration. We found there was no difference at 3 months. There was slight increase at 6 months in those who did not have a plastic stent, and little higher at 12 months, but still not significant by the statistical terms. And this was the flow chart that at 1 year, 13.5% and 25% were respectively having in the stent and non-stent group a reoccurrence. The trend is higher in the non-stent group, but at 1 year it was not mathematically significant. So conclusions were that replacement of a metal stent with a plastic stent did not significantly reduce development of recurrent PFC in patients with disconnected duct at 1 year. Majority of the recurrence remain asymptomatic and do not require any reintervention. Very recently, in last journal of IJG, Roop and Wahid is our fellow, we followed these patients also pancreatitis and looked at diabetes now, and we found that at 12 months, those patients who had acute necrotizing pancreatitis and some who were treated with metal stents and drainage developed diabetes. So either they had pre-diabetes or diabetes. So that means about 1/3 of the patients are at risk or they develop diabetes by the end of a year of follow-up. This endocrine dysfunction is frequent after an episode of pancreatitis, as we all know, and intervention of these wounds was another risk factor. Probably they are more sicker patients. Changing gears, we collaborated with our Indian colleagues all over the country, and Vinay led this group of endoscopic management of pancreatic fluid collection guidelines from SGI, Indian Pancreas Club, Indian US Club, which is published in our journal JDE in 2021. Anthony Teo over here, along with our colleagues in Europe, in Asia and Europe, participated in this consensus guidelines on the management of EUS procedures and several EUS guided interventions. We also published a digestive endoscopy use of other lamps, that is Spaxus stent in pancreatic fluid collection and WAN. We also collaborated with our European colleagues and published in their guidelines of endoscopy in ESG guideline therapeutic endoscopic ultrasound on several interventions including pancreatic fluid collections. Changing gears a little bit, malignancy in pancreas also needs to be managed endoscopically, and EUS comes in forefront for getting the tissue. But not only tissue, some of these patients of malignancy, pancreatic head cancers come with gastric outlet obstruction. How do we manage that? Conventionally, we have been doing intra-stent placement, but these stents get also blocked because of tumor ingrowth to the uncovered part of the duodenal stems. But can we do GJ? There are several publications which come in last 4 or 5 years. Moin Kasab is one of the leading authors in this area. Jahangir once again took lead, and we looked at our patients carefully of those who had malignant gastric outlet obstruction because of pancreatic cancers, and if they had ascites, how were the outcome if we placed a lamps with GJ, gastric enterostomy. We used a balloon provided by Takao Itoe, a friend from Japan, a E-pass balloon which anchors the small segment of duodenum. We can distend it well and see it under EUS and place a metal stent. And placing this stent allows us to remain away from the tumor area, which allows us, the patient, to eat more freely without the risk of stent getting blocked. We had a small number of patients, but definitely we were able to place the stent even in presence of ascites. They had comfort of life, but the overall outcome was not so good because of ascites, which was a malignant ascites. Last few slides, pancreatic neuroendocrine tumors can be ablated with EUS guided RFA, which is a little area of interest for me. Not only that, in pancreatic cancers, in collaboration with Korean colleagues, we first published the role of EUS RFA in insulinoma. It's a very small study, but a definite difference has been made. Several publications from world over have now come in this field of using RFA for pancreatic neuroendocrine tumor, both functional and non-functional, with gratifying results. A small video, if I can show here, this is a small tumor, neuroendocrine tumor, which is being burnt during RFA. It's a very safe procedure, so much so that even our surgeons are getting now impressed with the results and passing on their cases because of the risk of pancreatic fluid collections happening later on. In the end, I would like to sum up by saying that EUS guided pancreatic interventions are surely and definitely evolving. Pancreatic fluid collection drainage, both Vaughan and pseudocysts, can be done with plastic and metal stents. Disconnected duct is common after Vaughan drainage. Plastic stent need not be used to replace a metal stent after resolution of wall of necrosis. Diabetes is a frequent problem. One should be aware of it, especially if they've undergone drainage. And in pancreatic tumor ablations, RFA is making its place felt. It's safe and effective. Thank you very much for your kind attention. This lecture would not be complete if I don't mention about the team who has been behind and with me throughout our journey. This is how we started more than 15 years ago at AIG in Somajigoda, Hyderabad, and this is the current EUS team. I tweeted in Twitter recently that each one is better than the other. Thank you very much.
Can I request the chairpersons to please felicitate sir for the commendable contribution? Thank you. Thank you, sir. For the next presidential oration, may I please invite Dr. Ramesh Roop Rai, Dr. Rakesh Kochar, and Dr. Sandeep Nijhavan to please chair this session.
A proud moment for all of us that we are going to listen to our president, who has contributed so much in last 1 year for the progress of ISG. Dr. Mahesh Kumar Goenka, he is presently the director of Institute of Gastrosciences and Liver Transplant at Apollo Multi-Speciality Hospital, Calcutta. He is fellow of various societies including ACG, AGA, ASGE and RCP, Glasgow and London. He is the governor of India, American College of Gastroenterology. He also is the editor-in-chief in position today of the Journal of Digestive Endoscopy, Journal of Society of Gastroenterology and Endoscopy of India. His enormous contributions in gastroenterology bagged him Cisco Pentax Oration by Society of SEI, J. Mitra Award by Indian Society of Gastroenterology, Lifetime Achievement Award by Association of Physicians of India, I. M. Modi Award for Excellence in Medicine by Global Association of Physicians of Indian Origin, in America, and also the prestigious International Leadership Award by ACG in 2020. Recently, he has been selected by American Society of GI Endoscopy for International Services Award to be given at the Chicago during DDW 23. We are all feeling proud about his achievements. Dr. Goenka has published more than 250 scientific articles in book chapters, and he is involved in the collaborative work with the institutes like Johns Hopkins Institute and the University of Pittsburgh. And Dr. Goenka has been a medical teacher for at last 35 years and has been mentor for not only to the national board students but also other students all over the country. He's been training for short-term trainees from all over world, including Europe, Africa, Bangladesh, Bhutan, and USA. He is convener of Specialty Board of Medical Gastroenterology in India, thereby being instrumental in policy decisions of GI training in India. And fortunately, I am also associated with him. He has served as National Secretary and President of SGEI of India. He is an adjunct professor of gastroenterology at Medical School of Wisconsin, USA. He has been on International Committee of ASG and ACG. So many achievements, so much to describe, but a few more to his credit. He was selected as President of Asia Pacific Digestive Disease Week 2019 and organized the largest Asia Pacific meeting of gastroenterology in December 2019. He also serves on Governing Council of India Asia Pacific Association of Gastroenterology and the chief editor of periodic EWGN from World Gastroenterology Organization. Overall, Dr. Goenka has made a difference in medical field in general and gastroenterology in India and all over the world. May I request Dr. Kocher to invite him for the presidential oration.
Well, friends, it is indeed my pleasant duty to invite Dr. Goenka for this oration. Dr. Goenka, I am proud to say that has been a fellow at PGI Chandigarh with us many years ago, and he has excelled himself in the field. And it is equally proud for me to say that the other orators in the morning today were all trained at PGI, and what better can be that the feeling for a teacher. I invite Dr. Goenka for the oration.
Thank you, Professor Roy and Professor Kocher, Dr. Nijhawan, and good morning, everybody. I joined gastroenterology in 1987, as Dr. Kocher just mentioned, as a DM gastroenterology student at PGI Chandigarh. It has been quite a journey for the next 35 years. But as I said yesterday, I never imagined in those years that I would ever be a president of this prestigious Indian Society of Gastroenterology. Here I am delivering the presidential address. So before I go into the subject, we have been working with the help of the Governing Council and our dynamic Secretary General for the last 1 year. And some of the achievement for this year has been that we have engaged ourselves with other associations like ACG and Global Association of Physicians of Indian Origin. We have done a number of public outreach programs, and I must compliment the chapter for that. We started the fellowship and membership for Indian Society of Gastroenterology members. We, for the first time, started Women in Gastroenterology in India. We have started new chapter and strengthened our relationship with state chapters. We have started e-voting for the conference bidding. Also, for the young gastroenterologists, we have added 500 new members in the last 18 months. We have partnered with NBE board for the DRNB program and are in the policy making. Now the president of the ISG is a de facto member of the medical specialty board. We continued the exclusive PG training programs and added one program which is evidence-based gastroenterology. As you saw, we published the annual book, and we have almost finalizing the guidelines for PPI, antiplatelets, and corrosive injuries, which are so relevant to our country. I'm also happy to announce that we would be rolling out tomorrow the first multicentral Indian polyp study, which will involve at least 25 centers, and we will have the data of 20,000 patients who are undergoing colonoscopy. I think you already heard three excellent orations today morning in the field of luminal gastroenterology, liver, and endoscopy. But I think what potential we have for the artificial intelligence, we have not really explored that much. Artificial intelligence has progressed dramatically, as you can see by the number of publications, which has improved to almost 1200 in the last 2 years worldwide. And there is in fact a journal now called Artificial Intelligence in Gastroenterology, which addresses this. I think we need to do more work. So what I did and what I plan to do in this presidential oration is to share with you what is artificial intelligence in gastroenterology in Indian context. I contacted the leaders from various institutes, both the government and the corporate organizations, and I could find out what is work is being done, and I would summarize that in the next few minutes. So what I found that there are 13 projects which are almost completion or are being published from mainly these 7 institutes: Apollo, PGI Chandigarh, Asian Institute of Gastroenterology, BITS, ILBS, SGPGI, and All India Institute of Medical Sciences. So for those who are not non-initiator, let me just in 1 or 2 slides define what is artificial intelligence. Artificial intelligence refers to simulation of human intelligence in machines which are programmed to think and act like human. So there are various platforms of artificial intelligence. The advanced ones are machine learning and deep learning. The deep learning involves convoluted neural networks in which there are multiple hidden lines so that the information is filtered to reach the final outcome. It was only about 5 years back, when it was found, 6 years back, that it was found that artificial intelligence system can work better than the human mind. So there are 2 advanced technology. One is machine learning and other is deep learning. I'm not going into the details of that, but they recognize the patterns. And as you can see that the machine learning is recognize a pattern of a cat and would pick up a cat in this position, but may not be able to pick up a cat in this position because of abnormal posture. Deep learning, which involves convoluted neural network, can pick up much more patterns and is likely to recognize this cat in any posture. So artificial intelligence in gastroenterology has been used in all 4 fields: endoscopy, pancreaticobiliary, luminal, and liver. Now in GI endoscopy, it can be either computer-aided diagnosis or CADE, computer-aided diagnosis detection or CADE, computer-aided diagnosis with CADEX, predictive modeling, endoscopic quality assessment, and artificial intelligence assisted reporting. Adenoma detection rate, we all know from the Western colleagues that it is so important. In the West, it may be hovering around 30%, but in India also, polyps do happen. And this is a retrospective study from Asian Institute of Gastroenterology, which showed that the polyps happened in about 10.2% of the cases. I'm sure from the prospective data which we are going to collect over the next few months, this will be much more. We also must understand that 1% increase in adenoma detection rate can reduce the CRC, colorectal cancer risk by 3%. So this is a very important parameter. Now we must also understand that during endoscopy, we collect lot of images. In a 15 minutes procedure, average about 27,000 images will be collected. And it's not possible for a human brain, which can only work without fatigue for a limited period of time, to interpret all these data. And that's where artificial intelligence comes into the picture. This was the first report of artificial intelligence in 2016 when this was used to detect polyp. A number of systems have been developed. Some of them are already in the market. Others are going to come to help us in the artificial intelligence. We conducted a small pilot study recently where we evaluated 515 consecutive patients in the age group of 18 to 80 years with and without artificial intelligence. As you can see, using this simple GI Genius system, we could increase the polyp detection rate by about 8% from 10.3 to 18.5. And this difference was more marked for smaller diminutive polyp. Of course, this is a pilot study and we need to do much more work on this. If you look at worldwide data, the adenoma detection rate has improved significantly from 25.2% to 36.6%. And so, this is definitely going to impact the decrease in the colorectal cancers. A step forward would be using this system to differentiate adenomas from hyperplastic or non-adenomatous polyp. This is the EndoBrain system, has been used in the West, and recently I came across this study from Asian Institute of Gastroenterology, which will be published in the next issue of Journal of Digestive Endoscopy, where the system was found to be accurate in about 90% to differentiate neoplastic from non-neoplastic lesions with a positive predictive value of 84% and negative predictive value of 100%. So if this is validated, as it seems to be becoming true, we may move from change in our approach from resect and histology to diagnose and leave at least small polyps which are shown to be non-neoplastic. Now, the other areas where artificial intelligence has been used for diagnosis is Barrett's esophagus, early gastric cancer, and even for evaluating the activity of inflammatory bowel disease as to whether this particular segment has an active IBD or non-active IBD. Now, the other area can be very useful is capsule endoscopy. There are various technologies which is being used for artificial intelligence capsule endoscopy. This is from the West, but this is the work which has been done from India. So both have been done with an aim to reduce the reading time from 71 minutes usual to 13 minutes in the study. The study from All India Institute, which has been conducted along with another organization, Indira Gandhi, the Delhi Technical Women Organization, has used a dilated CNN system in which they first modified the images of capsule endoscopy and the segment that suggests them into the CNN system. And this has been shown to decrease the potential of reading to less than 20 minutes. So I think these are the way forward for artificial intelligence in capsule endoscopy. What can be the role of artificial intelligence guided endoscopy in assessing procedural completeness and quality? And we have done some work on this with the help of a group from Hong Kong. We have divided upper GI system into 29 areas, and in the segments of antrum, maybe anterior, posterior, lesser curvature and greater curvature, similarly cardia, incisura, body, duodenum, EG junction, epiglottis, vocal cord, and so on. So examination would be considered complete if you examine all 29 areas. And this short video is just to show you how it works. So as you proceed, you look at the vocal cord and the epiglottis, and the pictures come here and the checkbox is ticked. Then you enter the esophagus, you look at the G junction, and then you go into the stomach. So each time the area is shown here and the box is ticked. And at this moment, 8 out of the 29 areas has been seen, and this gives you an overall bird's eye view. Now we are in the first part of duodenum, and then as you enter the second part of the duodenum, the artificial intelligence system recognizes it. Then you come back and retrovert, and by this time you've seen 23 out of 29 areas, and that's how you proceed and complete the examination. So at the end, you get a report that in this particular case, all 29 areas were seen, and so the examination was 100% successful. We conducted a small study in our department where we have evaluated 315 endoscopies, some done by trainers and some by trainees, and we, as expected, found that the trainers did better than the trainees. However, interesting part was that even the trainees did not look at the epiglottis or the vocal cord. So I think these are the areas which were neglected, and we feel as we develop this technology for other areas like colonoscopy, this can be a very important tool for teaching endoscopy. So this is the result. The first picture shows 68% success, 20 out of 29, and the second picture shows 100% success. Now the same system has been used by others for other areas, like this one has been used for EUS examination of pancreas. They divided this into 6 stations, and as you see one station, the box gets ticked and the images are collected, and so this can again evaluate pancreas at EUS with a complete fashion. Now the other areas where work has been done is to look at the endoscopic quality of at the time of colonoscopy, Boston bowel preparation, and this goes on showing you cumulative result of what grade of preparation is done, and this has been found to be accurate in about 93 to 97% of the studies. So let's move from endoscopy to pancreas and biliary system. So artificial intelligence is used in pancreatico-biliary system for pancreas, gallbladder, evaluation of indeterminate biliary stricture, predicting post-ERCP pancreatitis, and so on. I could not come across any work on pancreatic cancer which has been done in India, but there are work which is published from outside where it has been found to be very useful. But what interested me is this work from PGI Chandigarh. Here they collaborated with IIT Delhi, and they just simply looked at the ultrasound images of suspected carcinoma gallbladder, 218 patients, 1255 frames, and they used CB RADs, just like liver RADs, and found out that with artificial intelligence, the overall accuracy can be 92%. So carcinoma gallbladder is a disease specific for our country, and so I think a study like this is going to be very useful. So indeterminate biliary stricture continues to be a challenge at cholangioscopy. So we collaborated along with bits from Bombay, and we contributed our images from Spyglass DS1 and DS2. And this was a study which was done on 25 centers, including 7 from Asia. The 227,000 images were used for training and 56,000 images for validation. And we found a good area under curve, and we find that this can be a very good tool subsequently for diagnosing the etiology of CBD strictures. The other area which is of interest is post-ERCP pancreatitis. We know there are a lot of risk factors, and there are a lot of meta-analysis and network analysis which has been done on this area to evaluate the importance of these risk factors. So in this study, we collaborated with other centers from USA, Germany, Korea, Malaysia, and Taiwan, and Asian Institute of Gastroenterology is a part of the study, where 12 multi-center randomized controlled trial evaluating PEP were included, and a gradient boosted machine learning was used. We evaluated 7,300 ERCP with 8.6% PEP rate, evaluated 22 risk factors and 4 possible interventions to reduce these risks: rectal NSAID, hydration, stent, and combination. And we found that using artificial intelligence, these were the decreasing importance of risk factors, a good area under curve, and we formed, like any other medical calculator, a post-ERCP pancreatic risk calculator. So use all these parameters are entered and gives you a figure of what is the possible PEP rate and how the effect of interventions would be. So this is an example of a data being entered in one particular patient, and the computer tells you with artificial intelligence that if you don't use a PEP prophylaxis, this patient is likely to get 18% chance of getting a PEP, and if you use endomethacin, this will be reduced to 10%. I think these are early days, but these are interesting data to be shared. I think Asian Institute of Gastroenterology is involved in another study published in JCC, where they evaluated 134 videos of ulcerative colitis, and they compared the scoring done with CNN, convoluted neural network, with Mayo scoring and UCIS, and found that the CNN improves the way you do the scoring. Now, this is another work which was done from Lucknow, SGPGI, Dr. Ghoshal. So they looked at response predictor for ulcerative colitis, and when they did a univariate analysis, there were multiple parameters which were found to be useful. But if they used an artificial intelligence based analysis, it was found that only hemoglobin and albumin were responsible for altering the outcome of ulcerative colitis in these patients. So let's now finally move to the area of hepatology. I think these are some of the areas where work has been done in India: liver fibrosis prediction, predicting ACLF outcome, predicting variceal bleeding risk, and so on. We all know that there are existing fibrosis risk factors, and we've been talking about it in this meeting as well. FIB4 and ARP3 is the one which is most commonly used, but most of these parameters, most of the scoring system are based on the presumption that the data for different parameters will be widely separated and you can draw a straight line between them. But actually it is not so, and the data are usually haphazard. So if you use artificial intelligence, you can create a liver fibrosis score which is better than many others. Apollo has a strong artificial intelligence network system, and we have developed scoring system for many other diseases, including cardiovascular disease during COVID era. These have been published in high impact journal. And we feel that artificial intelligence based scoring, because of the validation, feedback loop, accuracy, and machine learning, may be better than the traditional scoring system. Here we used our own data over a period of 8 years, 26,000 elastography, of which 11,000 were done for NASH or NAFLD, and we collected the retrospective data of 5,150 patients who were NASH, who had complete data. We evaluated these risk factors using propensity matching details and used the gradient boosting method to find out if we can develop an artificial intelligence liver fibrosis score. So this is the development cohort of 5,150 patients, 11 parameters, and this is the validation cohort. These are some of the graphs. And we found that there was a very good correlation. In fact, those patients who underwent liver biopsy, we found that artificial intelligence liver fibrosis score has a very high AUC curve. We presented this data last year in the American College of Gastroenterology meeting, where we compared AILF with FIB4 scoring and found that sensitivity, specificity, most of the parameters, AILF was better than FIB4, and NRI, which means the reclassification improvement with AILF over FIB4, was almost 0.5%, 0.5, that means 50%. We developed a clinical algorithm, and we developed the first clinical API in liver disease in India, which has been certified. This is on a social media where a patient or a general physician can enter all the parameters of related to the liver disease and then can come out with a prediction of possibility of what is the likelihood of having the fibrosis, and this can also guide the subsequent steps to be taken. A somewhat similar work has been done from hepatology department of PGI Chandigarh with a larger number of biopsy-proven patients from Co-Asia cohort. 220 patients in India was validated, and they also found that a machine learning scoring system was better than FIB4. The other areas where work has been done in India is in ACLF, again from hepatology department of PGI Chandigarh on ACLF patients. They had 3 variables, creatinine, INR, and circulatory failure, and they found compared to the MELD score and ARC score, ARC AI model was better predictor, and they divided the patient to high, intermediate, and low risk of mortality based on this. Now, this was a collaborative work between ILBS and AIMS Delhi, where we all know that endoscopy has high risk and low risk endoscopic varices. Now, but we also know that these are not the only one which will predict the risk of bleeding. There are other factors, and they combined these 2 factors in artificial intelligence model, and they found out that compared to endoscopy alone, which had 58% predictability, this had a predictability of 93% in internal validation and even 85% with external validation. So these are important landmark studies which are going to change our approach to some of these situations. I think the areas where we can work is, we do not have work from India, is LIRADS classification, and to help us, help our hepatopathologists in deciding their results. What's the future of AI? I think the future seems to be bright. Some of the work from Japan has gone a step beyond, where by endoscopic artificial intelligence, you can not only differentiate non-neoplastic and neoplastic lesions, but also decide whether it's an invasive cancer or a non-invasive cancer. So what will be the final potential impact in medicine? I think I would refer to this work from Johns Hopkins from Martin McCurry, published about 3 years back, where he reported that after heart disease and cancer, medical errors is the most important cause of mortality in USA. And I think using artificial intelligence in a judicious way may reduce these medical errors in future. So are everything good? No. There are some limitations of artificial intelligence, limited external validation in the available literature. Sometimes the gold standards are missing. There's a cost factor, and of course there are the issues related to privacy and medical legal aspects. But I think, why did I choose this for my oration? I think we have large volume of data. In fact, West looks towards us to give them data for setting up their artificial intelligence. And we sometimes forget that we have excellent AI facilities available at our IIT institutes, ISI institutes, and many similar institutes. And we also have multiple institutes where we can validate data generated from an institute. What is required is that we amalgamate all 3, and then we can have a mine of publication from our country. Thank you very much.
Thank you, Dr. Goenka. It's my proud privilege to invite our President of Indian Society of Gastroenterology to receive the prestigious Presidential Oration Award, please.
Thank you, sir. For the next plenary session, may I please invite Dr. Mahesh K. Goenka and Dr. Govind K. Makharia for chairing the session. Also, may I request Dr. S.K. Acharya, Dr. B.S. Ramakrishna, and Dr. C.E. Eapen to please be the judges for this session. Thank you.
Thank you so very much. And we had 4 wonderful orations this morning. Wonderful orators, delivered very, very well. So coming to one of the important sessions of our society, that's the best papers and best research in our country. We have 2 sessions for that. One is the plenary session, another one is a Young Investigator Award session. There have been changes in the research in our country, and we get wonderful papers from various institutions. We received about 30 papers for plenary consideration, and of those 30 papers, 6 have been selected by panel of judges for presentation this morning in this session. And there are the 6 best papers. Maybe invite Dr. Goenka to invite the first paper.
So as Govind said, that these are the best papers which we have found, and the first paper is by Dr. Saurabh Kedia from All India Institute of Medical Sciences, and it's related to fecal microbiota transplantation. Dr. Saurabh.
Good morning, everyone.
And Saurabh, you have 8 minutes to talk and then 2 for questions.
So, good morning, everyone. First of all, my sincere thanks from the AIIMS IBD team for accepting our paper for this session. So starting off with the presentation titled Fecal Microbiota Transplantation with Anti-Inflammatory Diet, followed by anti-inflammatory diet alone is effective in inducing and maintaining remission over 1 year in mild to moderate ulcerative colitis, a randomized controlled trial. So we all know that the probable pathogenesis of IBD is the aberrant immune response, which causes destruction of the intestinal epithelium in genetically predisposed individuals in response to the gut dysbiosis. And correction of dysbiosis with the microbiome manipulation would target the IBD pathophysiology upstream of the dysregulated immune response. And fecal microbiota transplantation and anti-inflammatory diet are 2 means of microbial manipulation. If you look at the evidence, then FMT has shown efficacy in patients with ulcerative colitis across multiple RCTs and meta-analysis, and it has also been shown to be equivalent to biologics in network meta-analysis. There is limited data on the role of anti-inflammatory diet in ulcerative colitis. The recent Kraft UC study compared FMT plus diet versus FMT versus diet alone in patients with mild to moderate UC. However, it was a small pilot study and it showed that diet was effective than other strategies. Moreover, we also don't have any study on the role of diet in the maintenance of FMT-induced clinical remission. So the research question for our study was whether the combination of FMT and anti-inflammatory diet leads to a better efficacy, and secondly, whether the anti-inflammatory diet alone can lead to maintenance of FMT-induced clinical remission. So the objective of our study was to evaluate the effect of a combination of multi-donor intensive FMT and anti-inflammatory diet in inducing, and the role of anti-inflammatory diet alone in maintaining remission in patients with mild to moderate ulcerative colitis. So this was a single center prospective open label randomized controlled trial which included patients with mild to moderate endoscopically active ulcerative colitis with disease extent beyond the rectum. Patients were allowed if they were on stable doses of oral 5-ASA, azathioprine and topical therapy, and if they had received biologics 6 months back, and if they were on steroids less than 20 mg of prednisolone equivalent. Patients with severe disease activity, those requiring hospitalization, surgery, comorbidities were excluded from this study. So coming on to the study design, so patients were randomized in 1:1 ratio to FMT and the anti-inflammatory diet arm, where they received anti-inflammatory diet and 7 weekly sessions of FMT from week 0 to 6. And in the standard medical therapy arm, the therapy was optimized by either increasing the dose of 5-ASA or adding or increasing the dose of topical therapy. Responders at 8 weeks in both the arms were continued in the maintenance phase. Those in the FMT arm were continued on anti-inflammatory diet plus baseline medications, while those in the SMT arm were continued only on the baseline medications. So the FMT was done by a pool of rural donors who were screened with the questionnaire and test to rule out transmissible pathogens and AMR genes. FMT was prepared from pool of 2 to 5 donors. Freshly prepared FMT was administered within 6 hours of preparation. First infusion was done in the cecum, rest in the left colon, and no antibiotic or ball preparation was used prior to FMT. So diet was an anti-inflammatory diet which was based on promotion of healthy bacteria, which would, through the healthy metabolites such as AHR ligand and short chain fatty acids, enhance the epithelial barrier, create an immunotolerant state, and increase the mucus production, and which will also alleviate the dysbiosis by decreasing the harmful metabolites such as sulfides, reduction of dairy products, refined sugar, animal proteins, and emulsifiers. So the diet promoted cereals other than wheat, curd, fresh fruits and vegetables, washed pulses and legumes, fish and chicken was permitted, and omega-3 rich oils were promoted. The diet excluded gluten-based grains, milk and dairy products, processed and red meat, emulsifiers and food additives, saturated and omega-6 fatty acids, and refined sugars. This was a sample diet chart which was given to all the patients. Coming on to the outcome measures, the primary outcome measures at 8 weeks was deep remission, which was a combination of clinical and endoscopic remission and clinical response. And at 48 weeks, it was deep remission and maintenance of steroid-free clinical remission. The secondary outcomes were endoscopic outcomes at 8 weeks and clinical and endoscopic outcomes at 24 and 48 weeks, as well as adverse events at 8 and 48 weeks. The sample size was calculated to be 150. However, the trial was started in September 2019. It was withheld in March 2020 because of the COVID pandemic, restarted again in August 2021, and again because of the third wave of the COVID pandemic, ended in December 2021. The donors in the second wave were screened for COVID-19. So this was the consort chart of the study. 113 patients were assessed for eligibility, of which 73 were randomized. After randomization, 7 refused. So 65 patients were included in the modified intention to treat analysis, of which 28 and 19 patients completed 8 weeks. And finally, 15 and 11 patients completed 48 weeks of follow-up. If you look at the baseline characteristics, then the demographics, the concomitant treatment and the past steroid use, and the disease severity parameters such as SCCI, UCI, hemoglobin, albumin, FCP, and CRP were comparable in both the arms. The diet was comparable at baseline in both the arms, while at 8 weeks, there was reduction in the prohibited food items, which was maintained till 48 weeks, and increase in the recommended food items, which was again maintained till 48 weeks. About 2/3 of the patients were highly adherent, more than 80% adherent at 8 and 48 weeks, and 1/3 were about moderately adherent. None of the patients were poorly or not adherent to the recommended diet. Coming on to the primary outcomes, so at 8 weeks, both the clinical response as well as the deep remission was significantly higher in the FMT arm as compared to the SMT arm. At 48 weeks, deep remission was again significantly higher in the FMT arm, while steroid-free clinical though was higher, but the difference was not statistically significant. The secondary outcomes, again at 8 weeks, the clinical remission was significantly high, endoscopic remission was high, and endoscopic response was again significantly higher. At 48 weeks, clinical response was higher, but the difference was not significant. And at the endoscopic remission was higher, significantly higher in the FMT arm as compared to the diet arm. The adverse event profile was comparable between the two groups. Among the predictors of response, the disease severity at baseline and the SCCI was lower in patients who responded to FMT. Other non-significant parameters were the disease extent and the baseline fecal calprotectin. So this trial obviously had some limitations. There was limited recruitment because of the COVID-19 pandemic. Although with the sample size, we could achieve our primary outcomes. There was no blinding because it was an open label study, but the endoscopy was assessed by the investigator who was blinded to the allocation. There were limited samples for fecal calprotectin due to the COVID-19 pandemic, and we could not evaluate the super donor effect because of the pooling of the donors. So to summarize, FMT from rural donors with an anti-inflammatory diet was more effective than optimized standard medical therapy in inducing clinical and deep remission in patients with mild to moderate ulcerative colitis. Deep remission at 48 weeks was also significantly better in the FMT arm, suggesting that anti-inflammatory diet could sustain the FMT-induced endoscopic and clinical remission. Disease severity was a significant predictor of FMT response. To conclude, combination of FMT and anti-inflammatory diet would be an effective and safe therapy for patients with mild to moderate UC. Microbiome manipulation and dietary interventions may play a major role in the management of IBD and should be further evaluated. When we submitted this paper for plenary session, then the paper was under review and in GUT, and subsequently it was published in the journal GUT. And finally, I would like to thank our AIIMS IBD team who contributed to this work under the guidance and leadership of Dr. Vineet Ahuja. Thank you.
Are there any questions? Yes, Dr. Mishra. There's Dr. Shiva Prasad by the time Dr. Mishra is. Dr. Shiva Prasad.
I just have a quick question. You had a patient cohort who was already steroid dependent, azathioprine resistant. So what exactly was your standardized medical therapy that you continued in them? Obviously, such category of patients would have required biologics, but what was exactly the therapy arm for standard medical therapy for the period of observation of the trial?
So the patients were continued on the baseline medications, and the topical therapy in the 5-ASA was optimized. So the 5-ASA was increased and topical therapy was added if the patient was not on topical therapy.
Biologics in that standard art? Biologics?
Sir, less than 2% patients had received biologics in the past. No patient was on concurrent biologics.
During the period of study, no biologics?
No, sir.
Dr. Mishra.
Did you have an arm only FMT or only with diet?
No, sir.
So it can be diet, it can be FMT, it can be both.
Yes.
Secondly, in your methodology, you said curd was given, but milk products were excluded.
Yes.
How do you account for that? What is soy curd or milk curd?
It was milk curd only, sir.
But how do you exclude the milk products?
Sir, other than curd, freshly prepared curd has been shown to be anti-inflammatory. Curd was promoted.
So
Saurabh, last question. Did you do histology here, Dr. Acharya?
Sir.
Did you do histology?
Sir, we did histology.
And did you find any difference?
There was no difference in the histology, but the sample size was limited again, sir, because of the COVID pandemic.
How do you define the adherence in this? You said somewhere moderately adherent, somewhere completely.
So we used two methods of adherence, sir, a qualitative and a quantitative. So for quantitative adherence, we looked for the negative food items. We looked at the number of patients who stopped the prohibited food items, and for the positive food items, we looked at the intake with respect to the recommended intake. So this was the quantitative adherence. For qualitative adherence, we looked at the 7 days diet as to how many days on the particular day of the patient was compliant. So there were two methods of adherence.
Thank you, Dr. Kiria. We'll move on to the next presentation by Dr. The next presentation is by Dr. Ambika Prasanna from AIG, and he's going to talk on gut microbial dysbiosis, gut barrier integrity, and severity of chronic pancreatitis.
Good morning. I would like to thank ISG-CON for giving me this opportunity to present our work today. To begin with, I would like to briefly introduce our earlier clinical study that led to the current one. So we have observed that altered intestinal microbiota in patients with chronic pancreatitis and other metabolic abnormalities that were associated with it. But currently, there are very few studies that are focused on the mechanistic link associated with CP. So the current objective aimed to evaluate the role of the gut microbiome associated with the progression and the severity of chronic pancreatitis in an experimental model. Coming to the methodology, the experiments were divided into two groups. For the first group, the mice received standard diet and CP was induced using L-arginine for weekly once for 6 consecutive weeks, and the mice were euthanized at 3rd and 6th week to track the progression of CP. Mice pancreata, intestines, and plasma samples were collected at 3rd and 6th week respectively, whereas stool samples were collected at day 0, 3rd week, and 6th week. For the second group, the mice received high-fat diet for 2 weeks, and CP was induced using the L-arginine model that was explained for the standard diet group. After the induction of CP and sampling of tissues, CP was confirmed using picroceria staining for pancreatic tissues and H&E scoring that was quantified using Imager software. To assess the pancreatic and systemic inflammation, cytokines were quantified in the plasma samples, and to further identify the changes in gut permeability, mRNA expression studies were conducted for the gap junction proteins zona occludens and occludin. These studies were then they were validated using fluorescent isothianate assessment that was done in plasma samples, and these were further confirmed using transmission electron microscopic studies. And to evaluate the altered gut microbiota, V3-V4 sequencing of stool DNA was conducted. Coming to the results, severe CP was induced in both the standard and the high-fat diet group that was confirmed by positive picroceria staining. These were identified by progressive SNR cell damage, and interestingly, the degree of fibrosis in the high-fat diet mice at 3rd week was similar to that of the standard diet mice at the 6th week. Also, we have checked the alteration of plasma cytokines levels in response to the pancreatic injury, and in the standard diet group, we observed that the pro-inflammatory cytokine IL-6 was significantly elevated at 6th week when compared to the 3rd week, and we also compared the cytokine levels from the standard diet group with the high-fat diet group, and we observed that the cytokines IL-17A, IL-4, and IL-6 were significantly altered when compared to the standard diet group. That shows that with the progression of CP, there was reduction in the inflammation. We also evaluated the intercellular, the intestinal paracellular permeability that was evaluated using FITC-dextrin that passes from the intestinal lumen into the blood. So FITC-dextrin is a fluorescently labeled compound, and the fluorescence level were measured in the plasma samples. So in the CP-induced mice, we observed that there were increased fluorescence levels when compared to the sham control mice, and these studies were further confirmed using electron microscopic studies that showed increased permeability in the intestinal tissues in the CP-induced mice when compared to the sham control mice. So further, the gap junction proteins were also downregulated for the markers zona-occludins and occludin for standard and high-fat diet groups, and these were further confirmed using immunohistochemical and immunofluorescence studies. And in the high-fat diet mice, there was a twofold reduction that was observed when at 6th week when compared to the healthy control mice. After analyzing the microbiome data, the richness and the diversity of microbial phylotypes were assessed using Shannon diversity indices. So for this standard diet group, there was a reduction of the species, microbial species from day 0 to 6th week, whereas in the high-fat diet group, the reduction of species from day 0 to 6th week was the most significant. And the relative abundances of phylum level were also analyzed, wherein in the standard diet group, the most abundant phyla, that is Bacteroidetes, showed alterations from day 0 to 6th week, whereas in the high-fat diet, the alterations of Bacteroidetes, Spirochetus were the most altered from day 0 to 6th week, that were also the significant. Whereas coming to the relative abundances of species, the species Bifidobacterium pseudolongum, Trivitella copri, which is the most abundant species, and Collinsella aerophysians showed alterations from day 0 to 3rd week in the standard diet group, and also the beta diversity showed a shift from day 0 to 6th week. Whereas in the high-fat diet group, the shift in the microbiome phylotypes from day 0 to 6th week was the most significant, and the species Bifidobacterium animalis, P. copri, and Ruminococcus nivis, along with D. sulfovibrio, also showed alterations. We also performed correlation analysis of altered gut microbiome with inflammation and fibrosis. For the standard diet group, the species Animalis and P. copri were negatively correlated with fibrosis, whereas in the high-fat diet group, the species P. copri were negatively correlated with fibrosis, and the pro-inflammatory cytokine IL-6 was negatively correlated with the species Navis. To further summarize our findings, these are the time-dependent events that we have observed in the progression of CP in the Ellagiline model. So, by modulating the diet in the standard diet group, we observed that at 3rd week, there was increased intestinal inflammation along with the onset of loss of gut barrier integrity. As the disease progressed to 6th week, the intestinal inflammation reduced and the gut barrier changes remained the same. Whereas in the high-fat diet group, we observed early changes of CP in the 3rd week itself, whereas the onset of loss of gut barrier integrity increased as the disease progressed to 6th week, which shows that there could be a possible role of these microbial changes in the aggravation of CP. So, a few of the key clinical implications would be that the current findings are concurrent to our earlier clinical study that confirmed microbial dysbiosis in CP patients. We also are currently looking into the role of the microbiome using other modulation strategies like probiotic, antibiotic, and FMT treated mice. These studies would eventually can be explored as one of the treatment modalities or therapeutic targets that can be confirmed in randomized control trials. So to further conclude, I would like to acknowledge DBT Wellcome Trust for funding our study, National Institute of Nutrition for providing us with animals, MedGenome Labs for the sequencing technologies, and AIMS Delhi for the electron microscopic assessment. Thank you.
Can we have some questions? Rakesh?
Dr. Ambika,
Yes.
Very interesting data. You change diet,
Yes.
you get liver fibrosis, you get changes in flora,
Yeah.
and you get changes in cytokines or whatever.
Yes.
How does one say that changes in gut flora are playing a role in fibrosis?
So,
I take something, I grow my hair,
yeah.
and my nails become blue. Can I say growth of hair is related to nails becoming blue? I can't.
Yes.
Secondly, if we go to your slide where you show the profile of gut flora in different weeks, if we could see that.
Yeah.
It's very interesting. They have to permit you to show the slide. Can we go to the
Can
slide, please? Your flow in the fat-rich diet group, the flora went from something. Let's go to that slide, please.
Can you show the slides of our presentation?
By 2 weeks, there was a big change in flora, and on week 3, they came back to what they were at the beginning. How do you explain that?
Yes, sir. So also the
If you
object.
get that slide, please, that will be
Yeah,
very
yeah. I'm
useful
getting this.
for people to understand this.
Sure, sure.
Yeah, slide 12, please.
Yeah.
So if we go to the high-fat diet, day 0, we had a lot of yellow.
Yes.
Second week, that disappears. Third week, we go back to that, and 6th week, we stay there.
It's stabilized.
So why is it that in 2 weeks, suddenly for some days, spirochetes
It is deep.
went away, and then they came back?
Short questions and short
Just-
answer, please.
Yeah. So firstly, we would want to check the because we are giving high fat diet, we wanted to check that spirochetes is the one that is reducing, and if this is the reduction that we are observing, then what role does this would play in the progression of CP? So we are not only checking the alterations itself, I mean the dysbiotic bacteria, but also the role this bacteria would play in the progression also.
My question is, how do we say that this played a role in fibrosis?
So currently we have
Rakesh,
studies on it.
brief answer would be these are observations with cause-effect relationship needs to be worked out. Dr. Mishra, last question.
Pardon my ignorance. How do you see increased permeability on transmission electron microscopy? They only show that the intracellular spaces are increased. That's only by Dextrin effusion that you can see the quantitate the.
Yes, we
No.
haven't quantified, sir, but only histologically we have compared with the control and we have seen the differences in the gap junctions.
So that's
That's it, histologically, only histologically.
So it's a morphological change which is
Yeah.
shown. Thank you, Dr. Sam.
We will move on to the third presentation of today, which is again from All India Institute of Medical Sciences. Dr. Shakira Yusuf is going to present that, and it's related to prevalence of celiac disease in patients with liver diseases, a systematic review and meta-analysis.
Thank you, sir. A very warm good morning to everybody. I'm going to be presenting a systematic review and meta-analysis, and the topic being prevalence of celiac disease in patients with liver diseases. I am Shakira Yusuf. I'm currently working as an SR in medicine in Chettinad Medical College in Chennai, and I was previously a celiac disease fellow at Beth Israel Deaconess Medical Center in Boston, and a majority of this work was undertaken during my time there. To start with a little bit of a background, as we know, as we have come to understand now, celiac disease is a multi-system disease that affects a variety of organ systems. And to zoom in on the liver, it is known to be associated with hypertransaminasemia, steatosis, cirrhosis, autoimmune liver diseases, to name a few. Celiac disease very much still remains an iceberg disease in the sense a lot of atypical celiac diseases presenting with atypical manifestations often tends to be missed. For example, there was a study that was taken up at Washington University where they saw 100 patients that presented with celiac disease at their outpatient clinic. Of them, at least 50% were presenting with non-classical manifestations, non-GI symptoms. And they found that among those patients that presented with non-classical manifestations, they were more likely to have a higher duration of symptoms after which they were diagnosed. They were likely to be present with lower hemoglobin values, lower BMI, and other adverse parameters. So that kind of points to how one must always maintain a high index of suspicion for extraintestinal manifestations. So in that context, a number of studies have characterized the way liver is affected in celiac disease, and especially in the past 2 decades. And Sainsbury in UK in 2011 sort of collated a lot of data on celiac disease and the elevation of LFTs in patients with celiac disease, and he found that 27% of patients with celiac disease were found to have high transaminases at diagnosis, and on a gluten-free diet, almost 90% of them normalized, the LFTs normalized within a year. Likewise, in case of cirrhosis, there was a Swedish cohort with a 30-year follow-up, And they found that celiac disease patients were 8 times more likely to have death related to liver failure, and they were also threefold higher. There was a threefold higher incidence of liver failure in them. And a Swedish study by Cochrane in 2002 also sort of threw light on the fact that around 4% of patients in a cohort of patients that were going to be undergoing liver transplantation, around 4% of them were found to have celiac disease as well. So the objectives of our study the objectives of our systematic review and meta-analysis was to basically find the pooled seroprevalence and prevalence of biopsy-confirmed celiac disease in the following 4 conditions: cryptogenic cirrhosis, all-cause cirrhosis, cryptogenic hypertransaminasemia, and all-cause hypertransaminasemia. So to talk about the materials and methods, so we used a search strategy that was comprehensive. We tried to look at data from both PubMed and the Embase that included Medline. And we basically searched for 2 main keywords: celiac disease and its related keywords, including gluten-free diet and others, and then liver-related keywords. And it was a very comprehensive search. And the inclusion criteria included for cirrhosis. We basically included studies recruiting patients with cirrhosis diagnosed based on imaging, histopathology, FibroScan. The cirrhosis group included 2 groups of studies. The all-cause cirrhosis subgroup included studies recruiting consecutive patients with cirrhosis, irrespective of the etiology, whereas the cryptogenic cirrhosis subgroup included studies that recruited patients with a diagnosis of cryptogenic cirrhosis after having excluded known causes. Likewise, we also had the all-cause hypertransaminasemia subgroup and the cryptogenic hypertransaminasemia subgroup. We excluded studies. We excluded studies where serology tests were not done on all consecutive patients. Known cases of celiac disease were excluded prior in order to make sure that the prevalence of celiac disease was appropriate in our sample. We excluded studies where AGA was the only or the first line screening test to make sure that we were using only anti-TTG and DGP, which are actually the recommended first line serological tests. AGA is no longer recommended as the standard test. We used R for statistical analysis, and a random effects model was used, and we also calculated heterogeneity estimates. If the I-square value for heterogeneity was more than 40%, we decided to explore heterogeneity in these estimates. So we identified 5,400 studies through Embase and 1,470 studies through PubMed, and eventually we zeroed in on 24 studies that fit our inclusion and exclusion criteria for the meta-analyses. Of these, 24 were included for qualitative synthesis and only 20 for quantitative synthesis. The 4 studies that were excluded from quantitative synthesis belong to the all-cause hypertransaminasemia subgroup because we did not find enough studies and there was a lot of heterogeneity in the studies. So in terms of study characteristics, most of the patients were male. Almost all the studies were hospital-based. Most of the studies were prospectively designed, and surprisingly, most of the studies ruled out NAFLD prior to giving a diagnosis of cryptogenic cirrhosis. And this was the case for all-cause cirrhosis and all-cause hypertransaminasemia subgroup as well. So what were our prevalence? We found a high seroprevalence of celiac disease in cryptogenic cirrhosis group. The prevalence was 15% compared to a general overall population-based seroprevalence of 1.5. Likewise, the biopsy-confirmed celiac disease prevalence was also 4.55 compared to a general population prevalence of 0.7%. There wasn't much heterogeneity, so we did not explore that in depth in this group. The second group was celiac disease in all-cause cirrhosis. There again, we found a higher seroprevalence. However, the biopsy-confirmed prevalence was not high compared to the general population. There was also a significant amount of heterogeneity, and when we pooled studies that prospectively screened for celiac disease, we found that the heterogeneity could be explained. And when it came to the effect of a GFT on cryptogenic hypertransaminasemia, among 11 studies, we had around 40 patients that actually adhered to a strict GFT, of which 97.5% showed improvement in transaminases, thereby pointing to a potential causal association. And likewise, cryptogenic cirrhosis subgroup, however, we do not have too many studies that described the improvement on a GFT. However, 2 studies did, Joshi and Cockin, and both showed that with a GFT there was improvement in MELD and CTP parameters. There is also some attempt to have to find out whether there's any potential causation of liver damage due to celiac disease. And one of the hypotheses is that toxins from the gut get carried over to the liver through the gut-liver axis through the portal vein, and that sort of induces inflammation, resulting in liver pathology in celiac disease. There was also a significant seroprevalence discrepancy between seroprevalence and biopsy-confirmed seroprevalence.
Conclusion, please.
Just a conclusion. So the conclusion of our study was the prevalence of celiac disease and cryptogenic cirrhosis was high compared to the general population. Roughly 5 out of 100 patients or 1 out of 20 patients have biopsy-confirmed celiac disease in this group. Cryptogenic hypertransaminism as well. 5 out of 100 people or 1 out of 20 people have high levels of seropositivity as well as biopsy-confirmed celiac disease. And the prevalence of celiac disease in all-cause cirrhosis, however, was not different compared to the general population. Thank you.
Again, Rakesh and Dr. Mishra.
Good morning, Shakira. Very
Good morning, sir.
Lot of data. I have my concerns. Firstly, as everybody in this audience knows, patients with cirrhosis develops high frequency of antibody to virtually everything. And it's possible that this seropositivity is just a reflection of that phenomenon. Second, there is nothing that is typical diagnostic of celiac disease on biopsy. To say biopsy confirmed celiac is very difficult. We know that there is a high association with HLA DQ2, DQ8. Did any of these studies look at those? In the absence of that, it will be very difficult to say that this is. Thirdly, we didn't hear about quality assessment of studies or publication bias. When we look for combination of 2 diseases, publication bias is extremely highly likely.
Thank you, sir. I'll try and comment on the first remark that you made about high seroprevalence. It is indeed the case that patients with liver diseases have a high level of immunoglobulin titers and they have false positivity for many diseases. And this is perhaps the case with our studies as well. But what we did find was that not just the seroprevalence, the biopsy-confirmed seroprevalence was also high in the cryptogenic cirrhosis subgroup. I hope that answers your question.
I said about the biopsy also. Biopsy is not diagnostic ever for celiac disease.
Sir, to answer your question about HLA DQ2, most of the studies did not attempt to look at it. They confined themselves to. So as far as there was publication bias in all the 3 groups.
Dr. Mishra, brief question.
Nothing against you, Shakira. My just concern is that the plenary is the top 6 papers of this country in this area. If we have meta-analysis, there's no science involved in it. It's basically statistical analysis. But agreed. But that means there's no bench science and clinical science going on in the country. We should have more of clinical science, and it's a good paper. It can go anywhere, but probably we'll have more of bench science as well as clinical science in this country.
I think, Dr. Mishra, I do not totally agree with you that meta-analysis, network analysis is also very, very important information, and if they be generated from our own Indian scientists, we have to give importance to that. Thank you.
Thank you for your time.
So we had our first 3 papers. Now moving to the 4th one, and again this is very important from a group of Dr. Ramakrishna. This is being presented by Dr. Rohan, and the title is "Long-Term Effect and Efficacy of Azathioprine in the Management of IBD: a Real-World Experience."
Good morning, all. And at the outset, I would like to thank ISG and the organizers for providing me this opportunity to present at such a prestigious forum. So, the topic, the article stands for Long-Term Safety and Efficacy of Azathioprine in the Management of Inflammatory Bowel Disease, a Real-World Experience. This is the outline of our study and my present talk today. So, coming to the background of azathioprine use in inflammatory bowel disease, what is well known is that azathioprine, we all know, forms the cornerstone of therapy for maintenance of sustained clinical remission in inflammatory bowel disease, both ulcerative colitis as well as Crohn's disease. It is often used as adjunct with biologics, especially infliximab, as it prevents immunogenicity and formation of antibodies against these biologics. It has a slow onset of action with which peaks around 17 weeks, and the side effects can be dose dependent as well as dose independent. TPMT and NUDT enzymes and their polymorphisms often affect its metabolism. There are, however, certain lacunae in literature or practical issues regarding azathioprine use. We don't know about the efficacy beyond 4 to 7 years of usage. There is paucity of data regarding this. The long-term safety, there's always a question mark of malignancy, especially non-Hodgkin's lymphomas. And patient compliance as well as physician apprehension is a big issue in practical terms. And as a consequence of this, there is presently no guideline or society recommendations on long-term safety, dose reduction, or discontinuation of azathioprine in IBD, and that is the focus or the objective of our study. So we analyzed azathioprine use and outcomes in a mixed cohort of IBD patients followed up over a long period of time. This was a retrospective analysis of a prospectively followed database of patients at a single tertiary care center, the SRM Institute of Medical Science Hospital in Chennai, which involved 507 IBD patients, a mixed cohort of ulcerative colitis and Crohn's disease over the last decade or so. The outcomes were long-term compliance, tolerance, dose of azathioprine, the clinical outcome at the point of last follow-up, type and duration of to onset of adverse events, and subsequent amendment to treatment in the form of dose reduction, temporary withdrawal, or discontinuation. So we had certain pre-defined outcomes which were as follows. Efficacy was determined by the presence of durable clinical remission at the point of last follow-up. Durability was assessed by absence of the need for corticosteroids and or not more than one clinical relapse after induction of remission. Clinical remission was assessed by a combination of symptomatic, hematological, biochemical, and inflammatory parameter improvement. And a relapse was defined by the recurrence of symptoms with or without derangement in various parameters. A non-responder was identified by presence of ongoing disease activity or corticosteroid requirement or more than one relapse after initiation of azathioprine. Side effects were classified as dose dependent and dose independent based on whether we were able to reinitiate these patients on azathioprine after the onset of a side effect. So this graph briefly shows the azathioprine exposure in our IBD cohort, ulcerative colitis and Crohn's disease patients. And to highlight that the median follow-up was 72 months. However, the median use of azathioprine in these patients was 33 months with a range of 3 to 421 months. So many of our patients received azathioprine for more than 20 years. We analyzed for a total of 1,359 patient years of azathioprine exposure to conclude to our results. So, out of the 507 IBD patients which were seen at our center over the last decade, 472 patients were included after exclusion of 35 patients who were lost to follow-up after the index consultation. So, out of these, 320 patients received azathioprine, 207 Crohn's disease, and 113 ulcerative colitis. 20.6% developed some form of adverse events, and out of these, 60% were idiosyncratic and could not be reinitiated on azathioprine, and 40% were reinitiated on azathioprine after dose adjustment or temporary withdrawal. 10% required some form of surgical intervention despite initiation of azathioprine. Coming to the adverse events in brief, myelotoxicity or cytopenia was the most common adverse event, followed by GI intolerance. Malignancy was seen in 1.8% of the patients. And to stress upon the malignancies, these were 6 cases, 1 of acute myeloid leukemia, 2 small bowel adenocarcinoma, 1 case of colorectal cancer, and 2 cases of squamous cell carcinoma. The adverse events happened more frequently in the first 6 months of azathioprine exposure, and only 1.2% of these occurred after 5 years of azathioprine use. These were the interventions upon encountering an adverse event to azathioprine. The starting dose to stress upon is just 0.97 milligram per kg, and the maximum dose that was reached in our study was 1.8 milligram per kg. On follow-up, 76.5% of the patients adhered to treatment and continued azathioprine till the point of last follow-up, and more importantly, 72.7% were in durable clinical remission. 11.8% were on monotherapy with azathioprine, and 53.1% on combination with azathioprine and 5-ASA. And 7% continued to have active disease. So this is a Kaplan-Meier survival analysis which shows the dropout to azathioprine as the index, as the event marked over here. And as we can see from this diagram, that the side effects contributed to majority of the dropouts in the first 6 months, and later on there were other reasons like durable remission and self-discontinuation, which formed 6.5% of the dropouts. This is an important cause because many of the patients feel a false sense of cure once they reach remission, and this is a practical issue we face in day-to-day practice. Our study had certain limitations. We did not assess for objective, endoscopic or histopathological parameters of remission homogeneously in all the patients. This was a real-world data, not a not in research settings. TPMT, NUDT 15 and RBC thiopurine metabolite testing was not available for majority of our study period. However, we do have some strengths, like less than 10% of our patients were on exclusive on biologics, and hence exclusive outcomes of azathioprine could be assessed. This was a real world setting, and long term follow up is one of the strengths of our study. We would like to recommend multi-center studies involving heterogeneous IBD cohorts involving different ancestries to further validate. And just as a take home message, most side effects occur within 6 months, and that is the period of strict surveillance. Malignancy risk is low, may or may not be related to azathioprine use. Non-compliance lead to high rate of breakthrough, around 38% in our study, and long-term exclusive efficacy of azathioprine is high as 65% without the use of biologics, and a lower dose max as compared to the West. I would like to conclude by saying physician apprehension and patient compliance are inversely proportional and do contribute to our day-to-day practice. Thank you.
Thank you, Rohan, for a wonderful presentation. Any question? Receive Prasad.
Thank you. Thank you. I think that was very practical information. I have a couple of questions. One is, did you differentiate UC and CD in terms of durable clinical remission and persistent effect over the long term? Because definitely, I think in clinical practice, there's going to be a difference between azathioprine in ulcerative colitis and azathioprine in Crohn's disease. But you had pooled your IBD together. I think that would help us. And second, how did you decide that you would only give a maximum of 1.8? Because most of the recommendations are 2 to 2.5. It is a practical issue as to how much we push up the dose to azathioprine. Where do we can't without the therapeutic drug monitoring markers? It is clearly a clinical problem. But then how did you decide that it's going to be only between 1.5 to 2? Why not go to 2 to 2.5?
Right, sir. So I'd like to answer both the questions. Coming to the question on the dose. So 1.8 milligram per kg was the median maximum dose reached in our patients. We have patients who have reached a dose of 2.5 to 2.7 milligram per kg, and these were a subset of patients. I would like to emphasize that there have been studies in the past which have shown that Asian counterparts tolerate a lower dose of azathioprine as compared to Western patients, and probably that was one of the reasons. And also there was some apprehension because of lack of availability of TPMT and NUDT 15 metabolite assessment in our patients. That was one of the things. Regarding comparison between ulcerative colitis and Crohn's disease, we did assess for the differences, and we could not find any significant differences in the outcomes. Although there has been a recent UK bio registry which has found that long-term efficacy is more in ulcerative colitis as compared to Crohn's disease. This was recently published last year in 2022. Thank you.
You talked about in your conclusion strict surveillance. What surveillance are we talking about? What is there any suggestion based on your used data? How frequently the patient should be seen? Is it clinical surveillance or is it a biochemical surveillance?
In the so usually as per most of the guidelines, what I recommend is in the first 6 months, that is in the first initial 1 month, we do CBC on a weekly basis and LFT on a monthly, followed by 3 monthly assessment for because these are the common side effects which are experienced in the first 6 months of treatment. The malignancy and all come after a much later period.
Yes. One small question. You said that the adverse effects were more important in the first 6 months.
Right.
I wonder those patients did you analyze the TPMT and other phenotypic analysis of those patients who are having side effects? Were there any correlation?
We did try to analyze TPMT in a few percentage of our patients, and that was available towards the later end of our study period. That is between 2019 to 2021. So that constituted a very small proportion of our patients, and that we have discussed about that in details in our paper. And this was just a summary of results. So I've not stressed upon that.
Thank you. As we in interest of time, we move forward. Thank you, Ron, for a wonderful presentation.
Thank you.
We move to the next paper. Again, this paper is from CMC Vellore. The transfusion-related lung complications are uncommon in non-ventilated liver disease patients undergoing low volume plasma exchange. Dr. Vijay Legender.
Good morning. Thank you to ISG for this platform. My study is titled Transfusion-Related Lung Complications Are Uncommon in Non-Ventilated Liver Disease Patients Undergoing Low Volume Plasma Exchange. This was done from CMC Vellore. Plasma exchange is now a standard of care to treat acute liver failure. Transfusion of plasma-containing blood products for any indication carries a potential risk of TRALI, that is transfusion-related acute lung injury, and TACO, transfusion-associated circulatory overload. Low-volume plasma exchange, which is 50% of calculated plasma volume, is now used to treat severe liver injury. TRALI, or transfusion-related acute lung injury, is defined as an acute onset event, onset during or within 6 hours of transfusion resulting in hypoxemia, bilateral pulmonary edema on imaging, with no evidence of left atrial hypertension and no temporal relationship to another risk factor for ARDS. TACO is a new onset or exacerbation of at least 3 of the following within 6 hours of a transfusion, that is acute respiratory distress, positive fluid balance, elevated BNP, radiographic evidence of pulmonary edema, evidence of left heart failure, and an elevated CVP. We analyzed the incidence of transfusion-related lung complications, namely TRALI and TACO, with specific interest in patients with liver disease undergoing low-volume plasma exchange. This was a retrospective cohort study design. Patients with severe liver injury who had plasma exchange in our department between January 2016 and January 2022 were included in the study. Patients who required supplemental oxygen or mechanical ventilation prior to plasma exchange were excluded from the study. Patients who also tested positive for COVID-19 illness and were symptomatic were also excluded. Cases who had onset of dyspnea within 6 hours of PLEX, onset during or within 6 hours of a session of PLEX were identified and included in the study. Now going on to the results. We had 298 patients during this time period who underwent plasma exchange to treat liver disease in our department during the study period. We excluded 51 patients because they were on supplemental oxygen or ventilatory support prior to starting the first session of plasma exchange. 5 patients with symptomatic COVID-19 illness were also excluded. So the final denominator we included in the study was 242 patients. Going on to the baseline demographics. The mean age was 37.3. We had 158 males in the study, which constituted 65%. The syndromes for which plasma exchange was initiated in this cohort was ACLF, which was 117 patients, ALF 74, acute liver injury 27, and others, which were 24. So 242 patients underwent a total of 779 sessions of plasma exchange in this particular group of patients. This slide is just to emphasize on the number of blood products that is transfused in and around the period of plasma exchange, and just to try and ascertain what the transfusion-related complications would be. So we had a total of 242 patients who underwent 779 sessions of plasma exchange, and which involved transfusion of 5,197 FFPs during those days on which plasma exchange was done. So this was about 2,500 in ACLF group, 1,500 odd in ALF group, 492 in the ALI group, and about 586 in the fourth group. The number of sessions per patient, the median was 3 in all of the groups, and the total was also 3, but the range was 1 to 9. And the volume exchanged in the entire group was about 4, which remained consistent in the different syndromes as well. We identified 3 patients out of the 242 patients who developed respiratory distress within this particular period of interest. The first was a 24-year-old gentleman who had ACLF, 60-year-old lady with ACLF was the second one, and the third one was a 72-year-old lady with ALF. All of them did have renal dysfunction. The third patient had evidence of LV systolic dysfunction. And I would like to point out that all 3 patients had a bit of positive cumulative balance, which was in the first patient was about by close to 10 liters, 6 liters in the second and the third patients. So this was from the time of admission till the event occurred. The baseline MEL scores were 49, 36 and 42 respectively. For the first and the third patient, the event occurred after the second session, and for the first patient it was on the day 1 of Plex. And all 3 of them had the event within 6 hours of initiation of a session of Plex. Here, all of them were not on supplement. All 3 of them were on room air at the time of the event. Second and third patient had evidence of pulmonary edema on imaging. The final adjudicated outcome for the cause of the respiratory distress in the first patient was pulmonary hemorrhage. The second and the third patients were TACO. The outcome of all 3 patients were death. Out of the 242 patients with liver disease who underwent low volume plasma exchange, 3 patients developed breathing difficulty within 6 hours of a session of Plex. None of the patients fulfilled criteria to make a diagnosis of TRALI. The incidence of TACO in this particular group was 0.3%, which is about 2 out of 779 sessions of Plex, and 0.04%, that is 2 out of 5,197 transfusions of FFP transfused during for the indication of Plex. All 3 patients died despite maximal supportive care. Low volume plasma exchange appears to be a safe treatment in patients with severe liver injury who are not on respiratory support, with no cases of TRALI identified in our study. However, TACO is a life-threatening problem. This could be reduced by avoiding fluid overload, or at least paying keen attention to cumulative fluid balance in and around the days of plasma exchange. The diagnosis of TRALI and TACO were made retrospectively in a study, and this remains a big limitation of the study. Prospective studies will bring more light to this particular problem. In conclusion, respiratory complications are uncommon following low volume plasma exchange to treat non-ventilated patients with liver disease. TACO can be life-threatening. The low incidence of transfusion-related lung complications points to a possible therapeutic window for Plex to treat liver disease patients. The 2 key points here would be, one is that all the patients were treated with low volume of plasma exchange, and we excluded ventilated patients or patients who had supplemental oxygen. Thank you.
Thank you, Dr. Alexander, for a wonderful presentation. Any question from the floor?
Well, it is expected, isn't it?
Correct.
All the patient who had kidney failure had fluid over. And it could have been possible that their low volume plasma, he should have been negative balance, clinical questions. So how can you say that low volume plasma exchange do not cause TRALI? Because you didn't show any data to say that it's indeed low volume plasma exchange cannot cause TRALI. And you cannot comment on TACO. They should have been excluded because they are as such fluid over.
So what I would actually like to highlight is that while giving a diagnosis of TRALI and TACO, that is what we actually started out with. But making a diagnosis of TRALI and both TACO are difficult retrospectively. What I would like to highlight is that problem of respiratory distress in this entire cohort in and around Apex within this time period was only in 3. Now, whether they had Charlie or Taco is difficult to say, but this actually alludes to the safety of a low volume plasma exchange. These patients had risk factors for Taco, and it is expected that they could have developed. And more attention to cumulative balance needs to be given to these patients in and around if we are planning to subject them to plasma exchange.
Dr. Alexander, I have no question. I would just suggest caution in interpreting data on this question collected from retrospective data. You alluded to it, but I thought I must say that I would be very wary of drawing any conclusions based on retrospective data for such a question.
I agree, sir. Thank you.
2 queries.
Last question.
First, tell me, what was the overall mortality in whole cohort? The patients who did not, mortality in the patients who did not dwell trolley, because maybe many patients died before they could dwell trolley or TACO. Number 1. Number 2 is you should have compared incidence of trolley or TACO with high volume plasma exchange, or actually historical data with other studies should have been compared with your study.
Sure, sir. Sir, to answer the first question, we do not have. I do not have the numbers on how many patients died in hospital from other causes. The second one, due to lack of time, I did not go into the high volume plasma exchange and standard volume plasma exchange for and the incidence of Charlie and Taco.
But overall mortality of patients undergoing low volume plasma exchange is very important, because before they got trolley or TACO, they died. So how can you say that trolley was lower in this thing?
Dr. Sharma, your point is well taken. I think we'll move on.
Thank
Just one comment. You give steroid to all your patients undergoing PLEX. What's the basis of this? Is that the common practice everywhere, irrespective of the etiology?
So all the patients who underwent plasma exchange were given a low dose of steroid, equivalent dose of 10 mg of prednisolone on the days of PLEX, and then subsequently probably for a few weeks. The basis of this is that these syndromes have an inflammatory pathogenesis to it, and we are trying to ameliorate both with PLEX and with corticosteroids. This is not standard practice elsewhere. This is a protocol from our institution. Thank you.
Thank you for your presentation. We move on to our next paper. This is from DMC Ludhiana. It is updated trend of IBD from Global Burden of Disease Study. Dr. Arshdeep Singh.
Very good afternoon to all, and at the outset I'd like to thank ISG for giving this opportunity to me. So I'll be presenting the trends of IBD from the Global Burden of Disease database of 2019. So GBD, we know, is a tool that is well established and well accepted, that provides a comprehensive picture on mortality and disability for various diseases across the globe. And this GBD 2019, which is the latest update in this database, provides the estimates for an exhaustive list of more than 350 diseases and injuries across the 200 countries of the world. So the previous to 2019, the GBD database was updated in 2017, and the IBD specific trends that were extracted from that database showed a growing incidence and prevalence and impact of IBD globally. But what is new in 2019 is that new databases have been added, new data sets have been added, and there is refinement in the analysis techniques of this database. So this is what we are going to present. And we investigated this data for the global and regional burden of IBD and the 3 decadal trends of IBD that we could see from this data. So this is available under the Open Data Commons Attribution License. It is free to access on the Global Burden of Disease website. The GBD database divides the world into 21 macrogeographic regions, and further these regions are then divided into 5 quintiles based on the socio-demographic index. This socio-demographic index is a composite index based on per capita income, level of education and literacy, and fertility rate in females younger than 25 years. So this SDI ranges from 0 to 1, and this has been divided into 5 quintiles across the globe in the 21 macrogeographic regions. So we extracted the 30-year IBD specific data on the disease burden and disability, which was measured using dailies, years lived with disability, and years of life lost. The GBD uses a Bayesian meta-regression modeling, which incorporates uncertainty intervals. The crude estimates, which specify the definite disease burden, were estimated, and age standardized estimates for distribution of the disease across the different age categories was also done. We analyzed both the crude and the rate data. Prevalence, incidence, disabilities were measured, and we also calculated the age standardized 3-decadal averages, the trends over last 3 decades, and the compound annual growth rate. So this is what we found, and I like your attention on few of the regions of the world. We can see the color coding is from blue to red. Blue means the lower prevalence areas or lower incidence areas, and red areas have the highest prevalence and incidence. So majority of the IBD burden comes from North America and Europe. But if we look at the changes that have occurred over the last 30 years, is this Central Europe. It has changed from red to orange, both for prevalence and in incidence. It has changed from orange to yellow, which means that the burden of disease is decreasing in this macrogeographic region. If we come to Asia, East Asia, the high income Asia Pacific region, Australia, and Southeast Asia all have shown an increasing trend in the burden of the disease. The 3-decadal average that we calculated was the prevalence was 64 per 100,000 and incidence was 5.2 per 100,000 population. Now, if we stratify this disease burden by SDI, I'd like your attention on these areas. The middle and low SDI countries all have shown an increase in the prevalence and incidence of IBD. But on the contrary, those still high SDI and high middle SDI countries are contributing much, but their prevalence and incidence is on the decline. If we look at the age categories, the pink is 1990 data and the yellow is 2019 data, and we can see that the incident ages for IBD at both the time points is 30 to 35 years, but with passage of time, the IBD population has become older and the prevalence has become higher in the elderly age groups. Now, this is again important. If we classify the regions based on SDI, this blue is 1990 and this orange is 2019. If we look at the low SDI countries where the disease is emerging, the burden is highest in the elderly population. In the middle SDI countries, the disease burden is increasing in both younger populations and in the elderly populations. But if we look at the high and high middle SDI countries, the disease burden is declining in elderly, but it is increasing in younger populations. So high SDI and high middle SDI countries have their disease established before the low middle and low SDI countries. So this is an epidemiological trend that disease begins in the elderly population and over a period of time it starts involving the younger populations. The number of deaths from IBD have increased. The global 3-decadal average death rate is 0.6 per 100,000 population, and this is highest in high SDI countries. The age standardized death rates have declined across the SDI based quintiles, but for the high SDI quintiles. This is again important if we look at the disability. The high and high middle SDI countries have high dailies. Now we know that dailies is contributed by both YLDs and YLLs, that is years lived with disability and years of life lost. So the majority of the contribution to dailies is because of years lived with disability in high and high middle SDI countries, whereas in low and low middle SDI countries, the contribution of years of life lost is higher to the total daily. So it means that in low and low middle SDI countries, there is increased mortality or premature mortality because of the disease rather than the contribution coming from years lived with disability. If we look at the region-specific 3-decadal trends, the global prevalence incidence is declining. This is against the 2017 estimates which showed that the disease burden is increasing. But if we look at Asia, Africa and the Middle East, the disease burden is increasing. So the major point highlight was that SDI was the major modifier of the disease burden, and though this data is derived from a well-established GBT database, it has a few limitations because of heterogeneity in quality of studies, and for regions with scarce data, there are wider uncertainty intervals and separate analysis of UC and CD is not possible. So current analysis gives a snapshot of the things that we can expect in future, and greater attention needs to be paid on the impact of IBD in low to low middle SDI countries, in which India part falls, because the burden is likely to increase in the coming years, and we need to look at the treatment as well as prevention aspect of IBD in these regions. Thank you.
Steve, thank you very much for a wonderful analysis of a global burden disease data. Dr. Simon Travis.
Thank you. Nicely presented. Have you considered comparing the trends in other immune-mediated inflammatory diseases? Because you're well known in Lydiana for your interest in the microbiome, but there's some evidence that may mediate other inflammatory diseases, thinking rheumatoid arthritis, because there you may get a comparison.
So there is analysis for rheumatoid arthritis based on the GBT database, but we have not compared the IBD prevalence and trends with the...
Quite a good idea to think of inflammation across tissue.
Thank you, sir.
Dr. Agrawal.
Dr. Singh, very interesting data. I don't have any question. I have a comment. Somehow the word takes GBT data as very accurate data. But the method by which things were calculated in 1990 and the way they are calculated in 2019 are absolutely different. And it becomes very difficult to draw any conclusions. Secondly, in countries such as ours, where the life expectancy has changed a lot in the last 30 years, even if incidence does not change, though even that, because of estimation methods have changed, could change, the prevalence would increase because people live longer. And one of the basic principles of epidemiology is prevalence is equal to incidence into number of years that a person would live with the disease. From when I was a student, MBBS student, the prevalence of diabetes has increased markedly, several folds, but not because diabetes occurrence has become more common. People are just living longer and the prevalence of diabetes has gone up. So I think we need to be very cautious in thinking that there is a doomsday scenario as far as we are concerned for IBD.
Thank you, sir.
Last question, Mr.
See, the Sustainable Development Index is correlated to the diet, standard of living. I mean, are these factors playing a role? Fecal microbiota, like Simon asked.
Sir, SDI has 3 components. One is per capita income, the education, that is literacy, and the fertility in females.
No,
so
about the westernized diet that we are consuming right now.
So it is nationwide, national SDI. So the SDI for India will be based on India's per capita income, and the SDI for another country will be based on their per capita income. Diet is not a part of SDI.
Thank you, Harshdeep Singh.
Thank
I think we had a 6 wonderful paper. We're going to come to close.
Yeah, so thank you for all the questions and comments from the house, and we would close this session and we'll start with the next important session.
And this session has been judged by Dr. Acharya, Dr. B.S. Ramakrishna, and Dr. Epan. Thank you so very much for judging these papers. Thank you so much.
Thank you. Thank
Before
you,
we
sir.
start with the next session, one announcement. The book which was released yesterday is available to the ISG members at the ISG counter. So anybody can go and collect the book from the ISG stall in the exhibition hall.
Right. Thank you so much. For the next session on Young Investigator Award session, can I please invite Dr. S.P. Mishra and Dr. Sameer Mohindra to please chair this session. I would also request Dr. Anup Saraya, Dr. Jayanti Venkatraman, Dr. B.C. Sharma, and Dr. Akash Shukla to please judge this session. We also will have the lunch break at 1 o'clock, while the lunch is being served from 12 to 3. Thank you.
We have a equally important research session as the Young Investigators Award. And may I invite Dr. Ashish Kumar Vyas to present his paper on the myth of hepatotropism of hepatitis B virus, placenta, a new home for the virus, from the All India Institute of Medical Sciences, Bhopal.
Good afternoon, everyone. I'm thankful to ISGCon for the opportunity. Today I'm going to present my research work entitled The Myth of Hepatotropism of Hepatitis B Virus, a placenta new home for the virus. So hepatitis B is a DNA virus which is non-cytopathic to the cells. And as we know, it can cause acute and chronic infection, and worldwide approximately 400 million chronic carriers are available. HBV infections depend upon the chronicity depend upon the age, and adults have less chance getting chronic infection compared to adult, which is only in 5 to 10%. And the chronic hepatitis B infection further lead to the severe chronic infection of hepatitis B, like it can lead to the cirrhosis and hepatocellular carcinoma. And the severity we can assess that every minute 2 people dying because of hepatitis B related problem. And India has around 3 to 4% carrier among 1.5 billion population. And the prevalence in pregnancy will vary. In India is 1.2 to 1.6%. So our question is, HBsAg positive mother, why few mothers transmit the virus to newborn? Not all. Is their maternal immunity playing any role, or the placental immunity playing any role, or the newborn immunity playing any role? So first, what we have done initially, we look for the maternal immunity. So these are we followed we screen hepatitis B surface antigen pregnant females. So this work we have done in collaboration with Institute of Liver and Biliary Sciences initially. So we divided in 2 groups: transmitting mother, non-transmitting mother virus on the base of HBsAg positive HBV DNA and newborn at birth and after following the 6 months. So what we have found, the baseline characteristics, we found difference in HBsAg quant only in the transmitting and non-transmitting mother. We didn't find difference in the HBV DNA level and HBV positive. But we know that high HBV DNA and HBV antigen lead to the vertical transmission. But we have selected the patients on the basis of because we want to look what is the immunological differences. So what we have done, first we look for the hepatitis B B cell subsets in this pair, mother and baby. The red color is mother and the green color is the baby. And we found the mother who is transmitting, they have decreased B cells number, CD19 positive, but they did, and their baby did not show difference. But when we look immature B cells number, that means they are not mature, it's transmitting mother high and their newborn show the same. But the plasma B cell which required for the antibody secretion were defective and decreased in the both mother and the newborn pair. And when we have done the correlation with HBsAg level, and we found the mother who is have high HBsAg titer, they have high immature B cells, and they have decreased plasma B cells number. That's why the antigen level is. Then why this maturation has not happened? So we know recently the T Folca helper cells play major role in the maturation of hepatitis B, and we found the mother who is transmitting, they have decreased TFH number in mother and baby pair same. And we also look for the activations marker which also decrease in the mother baby pairs who is transmitting the virus or the infected with hepatitis B antigen. So when we have done RNA-seq analysis, I'm not going detail, there are a lot of analysis were there, and we look for the T cell response pathways. Most of the genes who are down regulated, which is required for the T cell function in transmitting mother. The similarly, TLR pathway gene was also down regulated, and this we recently published in Hepatology Communication. Now we want to look what happened the placenta. So our challenge was what is the root of and the mechanism of vertical transmission, or the hepatitis B is only hepatotropic or just replicate in the placenta or not. So we want to look what happened to placenta thing. So what we have done, this is the work we have exclusively done in the AIIMS Bhopal. We screened 27,000 pregnant females and we followed them till the delivery, and we excluded or we divided into high viral load and low viral load
mother, and we have taken the control. We look for the HBV surface antigen, core antigen, cccDNA detection by digital PCR, and we have done histochemistry analysis in the placental tissue. What we have done, we first identify the HBV DNA in the placental cells. Then after that we look for the HBC antigen. This is not new. Already from 90 people have seen that antigen of hepatitis B have found in the placental sample. Then we first time look for pre-genomic RNA. This is what new. We have found the transcript of hepatitis B in the placental cells. Then we have done the digital PCR analysis for the cccDNA. So this is the preliminary data which I am assuming recently published in scientific report. This further validated in 30 sample, and out of 30 we found 20 sample of placenta contains cccDNA. Then we have looked for HB antigen. So people can say this HB antigen in the high viral and low viral load may be coming from the circulation, but we have taken the 50% of sample from mother who are negative for HB antigen in circulation, but they are present in HB antigen in the placental tissue. This shows the HB replication in the placental cells. Then we have looked for the first time this NTCP expression. Till now, no single study or not available that placental express NTCP. This is the receptor for hepatitis B, and we found the high viral load mother have high NTCP expression compared to the low viral load, and they have compared to the healthy. And what we suspect the Mercurelex is a drug recently approved for the HBV treatment in the Europe. So we get suggesting that the Mercurelex entry receptor may be used for the prevention of mother to baby vertical transmission. We also look for the other host factor which required for hepatitis B replication such as HNF4 alpha, PPR alpha, and R alpha, and this recently published in Presence in the scientific report. So another study we have done that acyloglycoprotein, because NTCP also required acyloglycoprotein receptor, and we found this acyloglycoprotein receptor directly related from the vertical transmission. And the mother who is transmitting the virus, they have high ASGPR expression compared to mother who is non-transmitting and the low viral load mother. And we'll also look for the co-localization of HBsAg with ASGPR, and we have in the placental sample, and we found that there is a localization. So that we are suggesting that hepatitis B mother to a vertical transmission may be happening due to the NTCP and acyloglycoprotein expression and directly correlated with vertical transmission. So in summary, mother, baby, and placenta, the unholy trinity for hepatitis vertical transmission, effective maternal immunity and decreased expression of viral entry receptor on placental cells are involved in the protection and mother to baby vertical transmission. And this is the diagram which we are saying the mechanism for protection and for the prevention of mother to baby vertical transmission. Thank you.
Rakesh, before you start, this work was done at ILBS, you said?
Sir, this half part of the study in ILBS and other in exclusively AIIMS Bhopal. There are two parts.
And you don't have any collaborators?
Sorry?
And you don't have any collaborators?
I have a phone in there.
Yes, only your name is there. Only your name is there.
No. Exclusive.
Never mind.
Dr. Vyas, very interesting data. I have 2 comments.
Yes.
Firstly, your slide 4, where you will look at the characteristics of. Can we leave the slides on while the questions are on? Helps actually.
Slide रखा रहे, slide लगाए रखिए please जब तक question खत्म ना हो जाए.
So you looked at the group, those that transmitted, those do not transmit.
Yes.
Let's focus on HBV DNA.
Yes.
There is a huge difference between the two groups, but
Yes.
the p-value is shown as not significant.
Yes, sir.
And I had a concern about that. And especially the range in those mothers that transmitted is mentioned as 0.85 to 8.
Yes.
The general understanding is that mothers who have that low HBV DNA cannot transmit infection.
Yes.
Now, second question is the main question, and that is related to staining for pre-genomic RNA that you did.
Yes.
Those methods are known to be non-specific at times.
Yes.
And I would like some evidence. So what were your negative controls for those? And especially when the amount of DNA present is so different, there could be just false positives.
Agree, sir.
What were your negative reports?
Yeah, so actually, sir, we agree that I told that HBV DNA and HB antigen lead to the vertical transmission. So why this DNA difference is there? Because we have taken HBV DNA at the time of birth. We are not analyzed, sir, before in third trimester or the second trimester.
It shouldn't change.
Yes, sir, it changed, sir. We have done recent study. We are so that the time with in pregnancy different semester. First, second, we have seen the dynamics of hepatitis B DNA.
How about the specificity of the pre-genomic RNA?
Yes. So, sir, what we have done, we have this is a real time based assay. What we have used exonuclease enzyme there. So this is RT-PCR based technique. So we have to use RT and non-RT, and we have taken the primer from the pre-genomic from the polymerase region and the core region. So there's overlapping.
So if you see these results, this gel,
yes,
the bands are not very
agree, sir.
sharp.
Yes,
and
sir.
I don't know what to make of these results.
Yes.
And
So this is not telling on the basis of gel, sir.
I'm sorry, we are running late. We are running late.
So we have extracted the gel, sir, and we have confirmed by the sequencing, sir.
Did you find any difference in
Madam.
vertical transmission between cesarean section births and vaginal births?
Sir, ma'am, we have not differentiated this.
Because
I'm sorry, please. We have no, I'm sorry, sir. Yes, but the time is up. You can't produce time. Whosoever comes first come first, sir. Thank you very much.
Thank you. Thank you so much.
Okay, housekeeping, can we have this lights off, please? They are glaring and we can't see the slides from here.
So the next presentation is
Thank
from
you.
Healpan Hospital, Delhi, and I would request Dr. Hardik Ahuja on the stage and present, and he'll be speaking on a double-blind, randomized control trial on role of nutritional therapy to prevent recurrence of hepatic encephalopathy in cirrhotics.
Good afternoon, everyone, and thank you, ISG, for this platform. Myself, Dr. Hardik Ahuja, and today I'll be presenting my study, which is a double-blind RCT to assess efficacy of nutritional therapy for prevention of recurrence of HE in patients with cirrhosis. So, as we all know that OHE has high risk of recurrence and is associated with poor survival. The recurrence is as high as 40% in 6 months, and there are several modalities which are available to prevent recurrence of HE, which include drugs like lactulose, rifaximin, probiotics, L-ornithine, L-aspartate, and in recent years, there are several reports of FMT being considered as an option to prevent recurrence of HE. Coming to malnutrition and sarcopenia, which are quite common in cirrhotics, and they are associated with increased risk of complications, including HE. And nutrition therapy has been shown to prevent complication of cirrhosis, improving survival, improving minimal HE, by improving sarcopenia and gut dysbiosis. But its role in prevention of recurrence of HE still needs evaluation. So this is a quick overview of pathogenesis and implications of sarcopenia in cirrhosis. As we all know, cirrhosis is a state of accelerated starvation with increased muscle protein breakdown and decreased synthesis. So the relationship between muscle depletion and HE comes from the compensatory role of skeletal muscle in ammonia scavenging through glutamine synthetase. So what we postulated is that by increasing the muscle mass, we might be able to reduce the ammonia levels and decrease the further attacks of HE. We aimed to study the efficacy of nutrition therapy in prevention of recurrence of overt HE. Our primary objectives included breakthrough episodes of OHE, time to breakthrough episode of OHE, and secondary objectives included OHE-related hospitalization, time to hospitalization involving OHE, changes in anthropometry parameters, serum cytokines, endotoxin and myostatin level at 6 months, and correlation of baseline arterial ammonia levels, anthropometry parameters, child score, MELD score with the recurrence of OHE. So we included consecutive patients with liver cirrhosis from August 2020 to November 2021, aged between 18 to 60 years, with history of recovery from one episode of OHE in the last 12 months. And we excluded patients with ongoing OHE at the time of enrollment, history of medication use like rifaximin, neomycin, metronidazole, L-ornithine, L-aspartate or probiotics in the past 7 days, alcohol intake during the past 6 weeks, prophylaxis for SBP, previous tips or shunt surgery. significant comorbidities, use of psychoactive drugs, and hepatocellular carcinoma. So the patients were randomly assigned by the dietitian to receive either nutrition therapy, that is group 1, or no nutrition therapy, that is group 2. All the other investigators were blinded. So group 1 received diet chart prescribing 30 to 35 kilocalorie per kg per day and 1 to 1.5 gram per kg per day of protein. And group 2 received diet chart of the same diet as they were taking previously. The follow-up was done every month till 6 months. Baseline routine investigations were repeated every month. So specific tests that were done at enrollment and at 6 months included blood ammonia, NCCT abdomen, critical flicker frequency, myostatin, endotoxins, interleukins, TNF-alpha, anthropometry, HRQL, and PHES. Previous treatment including lactulose and prophylaxis of varicose bleed, for example, beta blocker, was continued as before. So statistical analysis was done using SPSS 25 software, and appropriate tests were used for analysis. Dropouts from both the groups were considered failure of treatment on intention to treat analysis. And the sample size calculation, it was based on the previous studies on MHG. So basically we screened 262 patients of cirrhosis with past history of overt HE, and we excluded around 112 patients due to various reasons as listed. Finally, we randomized 150 patients with 75 patients in each group, and we followed them for 6 months. So the study groups were comparable among demographic parameters, etiology of liver disease, HE-related morbidity, lab parameters, child MELD score, PHES scores, anthropometry, interleukins, cytokines, as well as calorie and protein intake. So after 6 months, we saw that there was a significant change in the two groups. The only difference being the group 1, which is the nutrition therapy, had a significant improvement in all the parameters, and its group 2 had significant downfall in almost all the parameters. Coming to HE related outcomes, the number of breakthrough HE episodes, the number of breakthrough HE related hospitalization death, they were comparatively and significantly lower in group 1 as compared to group 2. While conducting the univariate analysis, we saw that no nutrition therapy, serum ammonia level, various anthropometric parameters like hand grip, mid-arm circumference, triceps, skin fold thickness, skeletal mass index, fat mass, and fat percent, they were associated with recurrence of HE. But on multivariate analysis, only no nutrition therapy and baseline ammonia levels were found to be the factors which predictive of recurrent HE. So this was a Kaplan-Meier analysis which shows significant difference between the two groups in terms of time to first breakthrough HE episode and first HE related hospitalization. Coming to SF36 parameters, all the SF36 parameters had a significant improvement in group 1 as compared to group 2, which showed a significant downfall in all the parameters. So we concluded that nutrition therapy can effectively prevent the recurrence of overt HE. It improves inflammation, ammonia, quality of life, and mortality. Thank you.
Yeah, go ahead. Rakesh, Akash, you have to join them.
Yeah, my question is, how did you said it's a double-blind study. So how did you do blinding for the patients? They did not know whether they are being advised for nutrition.
Yes, they did not know. As an investigator, we did not know, and patients.
That is single blind. That
No, sir.
But
Patients,
how can patient not know that?
So in both the groups, patients were receiving the diet chart. They did not know what they were prescribed.
खाना क्या खाएगा तो पता चलेगा उसको.
I have a problem.
Rakesh.
So, how much calories and proteins did the no intervention group end up receiving, and how much calories and protein did the two groups actually receive?
During the 6 months, sir?
Yes.
Yes.
Actually, I have not mentioned. They have not mentioned
The two survival curves diverged right at origin, meaning the diet started having its effect within a day.
Sir, that was a significant difference. I cannot comment on
So
that.
I'll go back to what Dr. Ashish was trying to say. To me, my ethics committee wouldn't allow me to do this study because people who are not eating adequately how can you not give them advice to eat what they are supposed
Sir,
to?
we were not harsh to the group 2. What we did is,
Please take permission of the chair, please. Just a minute. There's another question, please. Akash and Dr. Sharma, he has to answer that. This is our award session. Let him answer that. Then we can talk.
so the compliance were ensured by the dietitian every month with the help of diary. The patients noted whatever they were eating in the diary every day, and the compliance was done by the dietitian.
My question is, there were no dropouts. This is on ITT or what is it?
Intentional treat analysis, sir.
There were no dropouts.
No, sir.
He has to answer that. This is an award session, so the person presenting has to answer, not the mentor. Now you can answer it. Go ahead.
Yeah, Dr. Agrawal's question, nutritional therapy in control group, they say first we did study this kind of in MHE, then presenting this conference and sent the paper, that in every conference, from every journal, there was objection that it should be a controlled trial and it should be double blind. And therefore, that's why this time we conducted a controlled trial, double blind, and the paper was reviewed by experts, earlier paper, and they wanted this one.
No, despite your answer, sir, how do you double blind diet? Number one. Number two, the question is valid. How much calories were the non-diet people consuming? If they were drastically less, then you have problems there.
The double blind in the sense that patients, the earlier diet was recorded and then diet was modified to one group of patients, and second group of patients, diet was modified, but same calories, same protein was there. And they were prescribed the diet charts, but in one higher group, protein the high calories, in second same group, same calories.
So the blinding was to the calories, not to the diet.
And protein and this thing, protein and calories. And what was the second question?
Thank you very much.
So next presenter is Dr. Srilakha Chintaluri, and she is from AIG Hyderabad, and she will be speaking on crosstalk between the host gut microbiome, fungome, and bacteriome with their glycemic status in chronic pancreatitis patients.
Thank you. Good afternoon, everybody. First, I'd like to extend my gratitude towards ISGCon for giving us.
Can you have the lights off, please?
For
You can't see the slides here.
For giving me the opportunity to present my study here. So, to begin with, the Indian gut microbiota is different from that of the West. But what makes up our microbiome? We have viruses, bacteria, fungi, and archaea as a part of our microbiota. Our study here focuses on the fungal component of the microbiome. It's also known as the rare biosphere. We are studying the fungome with respect to the chronic pancreatitis disease. Previously, gut bacterial dysbiosis has been documented with respect to chronic pancreatitis, but the other components of the microbiota have not been studied well. As a result, we performed a pilot study where we have observed altered microbiome with diminished alpha diversity that has led to our current study, where we aim to evaluate the gut fungome with respect to chronic pancreatitis, the interactions between the fungome and the bacterium, as well as its implications with respect to the host glycemic status. This is the first study that evaluates the gut fungal alterations as well as the interaction between the fungome and bacterium in context of chronic pancreatitis. Coming on to the study design, we recruited patients with documented CP along with matched healthy controls and procured medical, nutritional as well as demographic data along with biological samples. We isolated bacterial and fungal DNA from the stool samples as well as performed routine biochemical analysis using the blood samples. The DNA samples were further sent for V3, V4 and ITS1 sequencing, following which we performed microbiome analysis as well as statistical and bioinformatic analysis. Coming on to the results, we screened 52 participants, of which we had to exclude 6 as we did not get any biological samples from them. We have recruited 28 CP patients who were further categorized based on their glycemic status, which is 16 patients with diabetes and 12 CP patients without diabetes. We have recruited 18 healthy controls. Going on to the demographic data, as you can see, the gender distribution, this disease is prevalent in males as compared to females, and the overall etiology suggests that there is an idiopathic type of chronic pancreatitis that is observed, followed by the alcohol and smoking type.
We use the modified Kuppuswamy scale for the Indian population to understand the socio-economic distribution of our participants. They belong to the upper middle class category, and majority of our patients were from the northeastern part of the country. On analyzing the clinical data, we observed a significant difference with respect to the BMI, the HbA1c levels, and the random blood sugar levels across the 3 study groups. Going on to the microbiome analysis, for the alpha diversity, we observed significant alteration with respect to both the richness and the diversity for bacterial as well as the fungal species across the 3 study groups. We analyzed the beta diversity for the same, where we observed that in the bacterium you can see a significant clustering between the disease group as well as the healthy control group, whereas for the fungome we do not see any discrete clustering. This has been confirmed using the hierarchy dendrograms. Going on to compare the relative abundances of the phyla for the gut bacterium, we have observed a significant decrease in the levels of the Firmicutes and the Bacteroidetes from the healthy control groups to the disease cohort. This indicates that there is a diminished diversity as the disease has progressed. Similar relative abundances were checked at the species level, where we have observed 44 significantly altered bacterial species across the groups, of which Lactobacillus ruminis, Veillonella dispar, and Prevotella copri were the most significantly altered. Coming on to the gut fungome, at the phylum level, we have observed that there is a difference with respect to Basidiomycota as well as Zygomycota, as it is more prevalent in the healthy controls as compared to the disease cohort. At the species level, we have observed 3 significantly altered species, which is Cladosporium cladosporoides, Candida albicans, and Candida tropicalis. Going on, we performed correlation analysis between the bacterial and fungal phyla, where we have observed that Actinobacteria phyla has had a significant positive correlation with Basidiomycota, Zygomycota, as well as other unidentified fungal phyla. We performed Spearman's correlation analysis for the top 25 bacterial and fungal species. In the Indian diaspora, we know that Prevotella copri is one of the most abundantly found species in our common gut microbe. In the healthy control groups, we can see that the Prevotella copri as well as Lactobacillus ruminis show no significant correlation with any fungal species, whereas in the disease cohort, we see that Prevotella copri shows a negative correlation with Aspergillus niger, and Lactobacillus ruminis shows a negative correlation with Ascomycota phyla. This helps us indicate that due to the decrease in the commensal gut microbes, there may be an increase in the opportunistic pathogenic microbes that are present in our gut. Following which, we performed correlation analysis based on the diabetic status. We performed a correlation of the CP diabetic group with the clinical parameters, where we observed that Prevotella stercorea has had a negative correlation with the random blood sugar levels, which indicates that the commensal bacteria has decreased as the random blood sugar levels have increased. Certain bacteriodale species have had a positive correlation with the random blood sugar levels. In the CP non-diabetic group, we have observed Oscillospira and Bacteriodales to have a positive correlation with the HbA1c levels, and Candida albicans to have a negative correlation with the HbA1c levels. With this states that the fungal species the relative abundance of this fungal species may decrease as the HbA1c levels have been increasing. Moving on, Megamonas species has a positive correlation with the random blood sugar levels, and Prevotella, Erwinia, and Veillonella species have had a negative correlation with the random blood sugar levels. In conclusion, for the first time, to the best of our knowledge, we have reported dysbiotic fungome with respect to chronic pancreatitis, its interactions with the altered bacterium, and as well as it may potentially contribute to the alterations with respect to the host's glycemic status. To aid our current findings, we are performing metabolomics, inflammatory cytokines, as well as plasma endotoxin analysis to comprehend these interactions on a functional level. As a clinical implication, we can use the above outcomes for newer treatment targets as well as develop novel personalized therapeutic strategies for chronic pancreatitis. As an acknowledgement, I'd like to thank the SCRB India as well as the Wellcome Trust TVT for funding our project and MedGenome Labs for their sequencing technology. Thank you.
Thank you very much. Rakesh.
Excellent work, ma'am.
Thank you, sir.
I have 2 questions. One
Yes.
is, did you check for delay in gut transit in these patients, any of these patients, because they were diabetics?
Yes, sir.
And secondly, did you check for small intestinal fungal overgrowth or small intestinal bacterial overgrowth in
So,
CP?
sir,
And
to-
last is, were there any drugs which these patients were
Yes,
taking
that's
which could
important.
have effect on the gut transit, which can impact the overgrowth of fungi?
So, sir, to answer your first question, for the transient part, is it? I'll first answer your second question, which is, have we done anything with respect to the small intestine? No, the study is particularly only for the pancreas, so we haven't studied anything with respect to the small intestine. And if you're talking about the medicines, we have not checked the effect of the medicines as yet, because we're waiting for the metabolomics to occur, after which we will be studying the effect of any medication with respect to the fungal growth. And could you please repeat your first question?
I think Rakesh you can go.
Assessment of gut transit.
That is something we need to study.
Dr. Sheila, 10 to 30% patients of chronic pancreatitis are known to have bacterial overgrowth. So, I mean, this question becomes very relevant.
Yes.
Dr. Shalika,
Yes, sir.
in your analysis, I did not see any correction for multiple hypothesis testing. If I go to your slides 18 and 19, those checker boxes,
Yes, sir.
if you see the number of boxes is about, or maybe 1 or 2 slides earlier than this. Yeah.
Yes.
So there are about 20 into 20 metrics.
Yes.
400. With a p less than 0.05, I would expect 20 values to be below 0.05.
Yes, sir.
And that's possibly the number that we see there.
Yes.
Does this really mean anything? Because this could just be a multiple hypothesis testing issue.
So, sir, at this particular timeline, this study is in its initial phases. So, for now, we're only reporting as it is.
This won't change.
So, for now,
my suggestion to you
yes, sir.
is that you might want to use correction.
Yes, sir.
So, without that, I don't think we'll be able to conclude much.
Yes.
Thank you.
Thank you, sir.
Again, the conclusion and your projections are far beyond the scope of the study. This is a preliminary study, and just hear your conclusion. This can do that, that can do that. So this should be avoided, basically. Thank you very much.
Thank you so much.
And now I invite Dr. Piyush Kumar to present his paper on low albumin. Low serum albumin is the strongest predictor of non-response to anti-TNF therapy in biological naive inflammatory bowel disease.
Good
from
afternoon,
the
everyone.
All India Institute of Medical Sciences.
Good afternoon, everyone. Firstly, I would like to thank ISG for giving this wonderful opportunity. So, I'll be presenting a paper on the title Low Serum Albumin is the Strongest Predictor of Non-Response in Anti-TNF Therapy in Biological Naive IBD Patients in Resource-Constrained Region with Inaccessibility to Drug Pharmacokinetics. This is our team at AIIMS Gastroenterology Department which have put in work for this paper. So as we all know, anti-TNFs are now the standard of care for moderate to severe IBD patients. We all have been using them day in and day out. Being the pinnacle, the best drugs available for treatment in IBD, they still are not 100% effective. Almost 1/3 of the patients, they lose response as early as 3 months, and 50% develop secondary loss of response. So we see a 13 per patient year loss of response with infliximab. So looking at this loss of response and the adverse events that are associated with these drugs, it is imperative that we look in for the predictive biomarkers for our subcontinent to stratify and treat patients effectively. So with this aim, we move forward and we planned a study to identify clinical, biochemical and mucosal biopsy molecular markers to predict primary non-response at 14 weeks and secondary loss of response at 54 weeks in anti-TNF naive IBD patients. So the methodology that was involved is, it was an ambispective study. We included all the patients who were following at IBD clinic from 2005 till October 2020. There were almost 6,500 patients, of which 186 were on biologicals. These patients, the clinical and the biochemical parameters were retrieved from the database, and mucosal biopsy specimens were extracted from the biorepository that has been started to be maintained since 2018. So these molecular markers that we have seen were oncostatin M and oncostatin M receptors and IL-7R. Briefly about them, oncostatin M is an IL-6 family of cytokine receptors which is seen to be expressed highly in patients who show non-response to biologicals, and IL-7R codes for alpha-4 beta 7 integrin, which is very important for recruitment of immune cells into the lamina propria. So as you see the results on the left, ulcerative colitis, of the 68 patients who were recruited, almost 25% had primary non-response, and the 50 who had response at 14 weeks when were followed till 52 weeks, almost 26% developed secondary loss of response. When these two groups of response and non-response were compared on baseline variables like age at anti-TNF initiation, BMI, disease severity, disease extent, and type of biological use, they were more or less comparable. There was no such difference. But when we started looking at the biochemical parameters, patients who developed primary non-response at 14 weeks had significantly lower albumin levels and significantly higher CRP baseline levels. Similar results were seen with Crohn's disease, 118 patients, in which almost 14% had primary non-response. When 98 patients were looked at 52 weeks, almost 45% patient developed secondary loss of response. These patients, these two groups of response and non-response, were more or less comparable on the basis of age at anti-TNF initiation, BMI, disease phenotype, disease severity, and type of biologicals. But here also we see baseline low albumin was a significant predictor of primary non-response. The molecular markers were evaluated among 50 patients of this 186 in ulcerative colitis. As you can see here, 8 patients of this 35 patients had primary non-response. Oncostatin M stromal cells and oncostatin M receptor were expressed, had a higher expression in patients who went on to develop primary non-response, but this do not show any significance for secondary loss of response. When we looked into this predictors for Crohn's disease, oncostatin M did not show any significance, but IL-7R, a baseline high IL-7R expression in the mucosal biopsies, was significant predictor for primary non-response in this patient. So these are the histopathological slides demonstrating the IHC expression of oncostatin M and oncostatin MR. The densities of expressions were higher in patients who showed non-response as compared to the ones who had response. When this data was plotted onto, when Cox proportional hazard model was used on this data, for ulcerative colitis, we had serum albumin, low serum albumin, and CRP, high baseline CRP, clinically significant on univariate analysis. However, only low serum albumin could carry this significance in multivariate analysis. For Crohn's disease, on univariate analysis, post-op recurrence and low serum albumin was statistically significant, but on multivariate analysis, it was only low serum albumin, which helped us in predicting primary loss of response. When we plotted Kaplan-Meier plots for this data, we found that when we look in for primary non-response, in ulcerative colitis, a baseline low value of less than 3.6 predicted primary non-response with an area under curve of 0.8, and for Crohn's disease, a baseline low value of less than 3.3 predicted primary non-response with an area under curve of 0.8 with a sensitivity and specificity of 70 and 75%. Similarly, a low serum albumin of less than 3.6 predicted a secondary loss of response for Crohn's disease patients with a sensitivity and specificity of 62.7% and 56.5% with an area under curve of 0.70. There are a few limitations to this study. The molecular markers were seen in only 50 patients, which is a relatively smaller sample size as compared to the clinical and biochemical markers. There was a lack of availability of the drug pharmacokinetics in our patient, and response assessment was purely clinical. The study lacked endoscopic healing and transmural healing for ulcerative colitis and Crohn's disease patients. However, if we look at the strength of this study, it is one of the largest cohort in resource-constrained country like ours, where we have lack of availability of drug pharmacokinetics. Cost is a very big issue, and we often see drug adverse effects with use of anti-TNF. It is first of its kind of study from Asia as per our knowledge. So to finally conclude, what can we draw from this study? When we are treating patients with ulcerative colitis who are bio-naive, and we are looking whether this for primary non-response, a low baseline albumin of less than 3.6 can tell us that this patient could develop primary non-response. However, on mucosal biopsies, high oncostatin M and oncostatin M receptor showed some significance, but the data is too preliminary to comment on that. However, for Crohn's disease, a low serum albumin of less than 3.3 and a low serum albumin less than 3.6 predicted primary and secondary loss of response. Higher IL-7R expression does predicted primary non-response, but was not clinically significant on multivariate analysis. So the take-home message would be a low serum albumin is a single strongest predictor of non-response for anti-TNF therapy in Indian settings. Thank you.
Thank you very much. Ashish, please take permission of the chair. We should go ahead, please. Rakesh, go ahead, Ashish.
Dr. Piyush,
Yes, sir.
so in your opinion, is this albumin a cause of low albumin is a cause of non-response, or is it just an association? Because patients having low response would also naturally have low albumin level. So it's just a surrogate marker rather than a strong case
Sir,
prediction.
that's a very good question, sir. Sir, in severe, moderate to severe disease, we do see patients with low albumin. That is basically reflecting the increased disease activity. And when we talk of biologicals and talk of their pharmacokinetics, it is more or less insulin dependent. So when we look for response, patients with low albumin have higher chances of showing non-response because of the drug pharmacokinetics.
So you agree with
So
that?
yes, I agree, but still, so this is what this data is telling us. So low serum albumin can predict. So we can take steps to increase it before we target the patients with anti-TNF.
कुछ नहीं होगा.
Piyush, the disease in pediatric subgroup is more aggressive mostly as compared to adults. So will this data hold true for the pediatric subgroup as well?
Sir, for pediatric subgroup, maybe this data can hold true, but I cannot comment on that because of the adult group. It was mainly for adult groups.
Rakesh.
2 points. Firstly, I heard something that we are looking for a predictor that can be used in countries where drug pharmacokinetics are not available. But on the other hand, we heard a lot about a lot of molecular markers, which would obviously not be available in the same countries. Secondly, I think I agree with what was said about this just being a marker of severe disease. Just giving albumin and treating hypoalbuminemia is not going to change anything. It's very well known that people who have few hair like me have a higher risk of coronary artery disease. But that doesn't mean that if you do hair transplant, you can reduce the risk of coronary artery disease.
No, sir.
That's
Answering your second question
Rakesh,
first.
your analysis is sometimes very hilarious too.
Good. Small this thing. To me, it appears more of a retrospective data analysis rather than this. Because what is the retrospective data analysis and what the molecular markers are done, they are done in stored histopathology samples.
Yes, sir. In the biorepositories that were maintained.
Your cutoff value of 3.6 albumin in ulcerative colitis, that 3.6 is also almost a normal albumin level.
So,
Thank you.
okay, thank you.
We now move on, and now I invite Dr. Anam Ahmed to present paper on patients with non-celiac gluten sensitivity exhibit site-specific gut microbial differences than those with irritable bowel syndrome.
Thank you, sir. Good afternoon, everyone, and thank you, IGCon, for providing me with this platform. I'm here to present our study entitled Patients with Non-Celiac Gluten Sensitivity Exhibit Site-Specific Gut Microbial Differences Than Those with Irritable Bowel Syndrome. As we all know, spectrum of gluten-related disorders varies from a well-known celiac disease to recently emerging non-celiac gluten sensitivity and wheat allergy. Till now, there is no definite biomarker for NCGS, unlike TTG, which is there for celiac disease. The diagnosis of NCGS is purely established on the basis of response to gluten-free diet as per the Selenos criteria. Additionally, anti-gliadin antibody has been reported to be positive in half of the patients with NCGS. We hypothesize that gluten is not digested completely in human intestine because of the lack of prolyl endopeptidases. Partly digested peptidase activates the immune system in susceptible hosts like celiac patients as well. This might be a possibility. There might be a possibility that the gut microbiota, by release of endopeptidases, converts an immunogenic peptide into non-immunogenic peptide, and hence gut microbe might be playing a role in the pathophysiology of NCGS. which led to a research question that do patients with NCGS harbor different microbiota than those with IBS patients? The first objective was to study the differences in the small intestine, colonic, whole gut microbiome of patients, small intestinal, colonic and whole gut microbiome of patients with NCGS and IBS. And secondly, we also had an objective to study the effect of gut microbiome by gluten-free diet in patients with NCGS. For this, we screened 479 IBS patients as per the Rome-4 diagnostic criteria because IBS has overlapping symptoms with NCGS. We ruled out celiac, and finally, we had 477 patients of IBS. They underwent serological screening with anti-gliadin antibody. 61 of them had positive AGA, whereas 416 of them had AGA negative. This was the first group, that is the IBS group. We collected stool samples from 100 patients, duodenal biopsies from 20 patients, and colonic biopsies from 16 patients. Coming on to the patients that had AGA positive, 31 agreed to participate, and we collected stool samples, duodenal biopsies, and colonic biopsies from all these 31 patients. They were put on gluten-free diet by a trained dietitian for 6 weeks. 14 of them did not show any improvement, whereas more than 30% improvement in the top 3 symptoms was there in 17 patients. They were diagnosed as NCGS. Please note that the criteria of NCGS was purely after they were given a gluten-free challenge, and if they complained of recurrence, they were diagnosed as NCGS, and then their baseline samples were used for the analysis. Further, we asked them to agree, we asked them for a follow-up, and 14 out of 17 agreed for a follow-up sampling. Again, their stool samples and biopsies were collected at the end of 6 weeks of gluten-free diet. DNA extraction was done for stool and biopsies as per the standard criteria in these kits. V4 region of the 16S RNA gene was amplified and sequencing was done by Illumina platform. Coming on to the results, we firstly wanted to see the difference in the microbiome of IBS and NCGS. For this, we compared the alpha diversity in the small intestine. Over here, we can see that the IBS patients, the samples from the IBS patients had statistically significant more alpha diversity than the NCGS patients. NCGS is the orange bar and IBS is the blue. Also, we saw the relative abundance in the top genera. We can see there was statistically significant higher bacillus, actinobacillus, escherichia, and low prevotella and veillonella in NCGS patients. However, we did not see any statistical difference in the alpha diversity of colonic samples as well as the stool samples. We also saw the beta diversity between the IBS and the NCGS patients. Yet again, there was a significant difference in the small intestine of IBS. We can see these dots that are over there. These are the small intestine, and we had the significant difference between them, whereas we did not find any significant difference again in the whole gut or colonic biopsies. Coming on to the core microbiome that was there in the 3/4 of the patients, that is 67% patients, we again had difference in the small intestine, significant difference in the small intestine, whereas again the colonic biopsy and stool samples did not show any significant results. At last, we wanted to see the site-specific co-occurrence in the IBS and the NCGS patients. Over here, small intestine, yes, did have a statistically significant data. We had 47 statistically different taxa over here. And the signature taxa in the IBS over here, this is the network analysis where we can see the signature taxa is Lactobacillus, Bifidobacterium, Dilister, Prevotella for IBS. And Bacillus and alkali genes over here is signature taxa for NCGS patients in the small intestine. Coming on to the large intestine, we had significant results. We had 44 statistically different taxa. IBS had Veillonella, Cyclobacter, Paraprevotella. NCGS had D-sulfovibrio, Bacillus, Rhodococcus, and Actinobacter. For a whole gut also had significant data. We had 35 statistically different taxa. Signature taxa were Megasphaera, Megamonas, Ruminococcus. NCGS had Elusium microbium, Succinovibrio. All these 3 sites, whole gut, small intestine, and colonic biopsy had different taxa. The second objective that was there, we wanted to see, do gluten-free diet affect the microbiome of NCGS? Again, we had significant data, and Please note that the signature taxa that was there in the NCGS patients before GFT, that is alkali genes and bacillus, they got lowered after gluten-free diet in NCGS patients, and solibacillus, Stenotrophomonas and Schwannella was increased in NCGS patients after gluten-free diet. There were few limitations of the study. The sample size was extremely small. We had higher, patients did not agree for biopsies. They were not ready for taking gluten-free diet. All of the patients AGA positive, they did not agree. There was lack of long-term follow-up, and this was a non-randomized study. Finally, we want to conclude that the alpha diversity, the beta diversity, and core microbiota in small intestine of patients with NCGS are different, which may suggest that the small intestinal microbiota may be playing a role in the pathophysiology of NCGS. I'd like to thank Department of DST for providing us with the fund, and our team at NCCS Pune, where the complete analysis was done. Thank you.
Thank you very much. It's open for discussion. Dr. Sachdeva?
Excellent data, ma'am. There are two questions. One, what was the diet taken by the IBS subgroup? Were they taking low FODMAP diet? Because if that is so, then the difference between the two groups in terms of diet becomes less.
Thank you, sir.
Wheat is one of the component of FODMAPs.
Yes,
FODMAP लिया था कि नहीं लिया था? हाँ ना.
No, sir.
बात खत्म.
And second is, did you check for small intestinal bacterial overgrowth in either of the groups?
Sir, small intestine is there. We saw the significant difference in small intestine only.
That was in terms of dysbiosis, the type of bacteria, number of bacteria.
Okay, sir.
Two questions.
Sir.
First, did you look at the difference in the bacterial flora of patients with gluten with celiac disease versus NCGS?
So this was not a part of this study.
But your basic hypothesis was that there may be a difference in the bacterial flora which may lead to that peptidase activation and subsequent this thing. So that is very important to know that the first part is there clear first. The second question is that did you look at the HLA of these patients who were AGA positive, because some of them might be
Yes, sir.
reclassified as potential celiac?
Sir, 68.9% had HLA DQ2, DQ8 positive in AGA.
That's my question. Second question. You did a challenge in these patients. How many had problems with challenge?
Sir, gluten
Gluten challenge.
Sir, actually, sir, everyone had problem, but the thing is, it varied. Some person came to me that they have problem in 1 week of gluten-free diet. Others took gluten-free challenge. Others took a while, like 2, 3, 4 weeks, but most of them came with complaints. But the time varied.
You followed wrong criteria for diagnosis of IBS.
Yes, sir.
I wish to know how many were diarrhea predominant IBS in this study group, and did you rule microscopic colitis in these patients or not?
Yes, sir.
Number two, I want to know NCGS gut alteration in flora, gut microbiota following GFT. What was the difference between alterations in small intestine versus colonic this flora?
Sir,
Following GFT and NCGS.
Sir, colonic bacteria, sir, we did not find any significant results. It was similar to the stool samples, hence we did not show the data over here. The only significant difference was there in the small intestine.
Secondly, you have taken IBS, which themselves have a altered microbiota.
Yeah, but second, the diarrhea from IBS. And second is NCGS following gluten-free diet. There was no change in colonic flora. That is what you mean to say, na? Small intestine there was a difference, but not in colonic flora. But however, at baseline, colonic flora in IBS and NCGS was different.
Sir, even at the baseline, sir, there was not significant difference. There was a difference, but the only significant difference was there in the small intestine. So that was just the co-occurrence, the last part, not in the alpha beta diversity.
Thank you very much. We now move on to the next paper. Dr. Harsh Sheth, Systemic Estimation of Prevalence of Microsatellite Instability and Lynch Syndrome amongst Colorectal Cancer Patients in India.
Good afternoon,
Multi-centric, multi-collaborative study.
Good afternoon, everyone. I know I'm the last speaker before we all head out for our lunch, so I'll try and make this as quick as possible. Lynch syndrome is the most common hereditary cancer predisposition syndrome, and it's caused due to germline pathogenic mutations in one of the 4 mismatch repair genes which are listed out here. They are essentially spell checker genes of our genome. And carriers of these mutations have up to 50% lifetime risk of multiple cancer types, including colorectal,
upper GI, and genitourinary cancers. According to World Health Organization, the prevalence of it in the general population is about 1 in 125. That would mean that in India alone, there are more than 5 lakh carriers of this one disease. But the good thing is that now we have a diagnostic pathway which works very, very well in picking out these patients. And most importantly, we now have interventions available to us that can help reduce risk of cancer, one being regular use of aspirin, and very recently are showing that resistant starch is also good at reducing risk of extracolonic cancers, especially upper GI cancers. Now, when it comes to knowledge about Lynch syndrome in India, very few studies have been carried out to date. There are 4 retrospective studies, but they suffer from either recruitment bias or they have very small sample size. There was 1 large observational studies, but that restricted only to studying microsatellite instability, which is a surrogate molecular phenotype in the tumor. However, all studies tended to show a high rate of microsatellite instability and Lynch syndrome in India compared to the Western population. And to date, therefore, there's no systematic evaluation of its prevalence. NCCN and NICE guidelines both recommend MSI testing in colorectal cancer patients to identify Lynch syndrome. But this guideline hasn't been put into practice in India in a wider context, partly due to the lack of data, and also ICMR since 2015 hasn't updated the guideline to put it to clearly state that it should be recommended in all colorectal cancer patients. So back in 2019, we set out to carry out one of the largest systematic prospective study, whereby our aim was to assess the prevalence of MSI and Lynch syndrome in colorectal cancer patients in India. And during a period of 3 years, we recruited 207 colorectal cancer patients from multiple centers across India, and we performed 3 tests: microsatellite instability, BRAF E600D, and germline Lynch syndrome testing in accordance with NICE guidelines. And our recruitment criteria was rather simple. The patients had to have an age above 18 years. There was no specific bias towards family history, but all the details were recorded. We collected primary tumor biopsy and a pathology report that went along with that biopsy. And the tumor site could be anywhere from cecum to rectum. And lastly, even if a patient had a prior history of cancer, they were recruited on the study and all the details were recorded. And like I said, in accordance with NICE guidance, out of 207 patients, 17 patients later on recanted their consent to participate. Therefore, we could only do analysis on 190. But in all these patients, we did MSI testing by PCR, out of which 111 had microsatellite stable tumors. So we knew that they were unlikely to have Lynch syndrome. But in those who had microsatellite instability, we carried out BRAF E600D because it's a good marker to stratify whether a patient is likely to have Lynch syndrome or not. In those who had BRAF E600D mutation, they were classified as sporadic. And in those that didn't have mutation, we carried out germline mismatch repair gene sequencing, of which 48 ended up having Lynch syndrome. In terms of average age of diagnosis, our cohort had about 52 years. There was 2:1 ratio from male to female. In about 1 in 5 patients, we found that they had a family history of cancer. That is to say, either first or second degree relative had history of either colorectal or other Lynch syndrome associated cancers. In about 41% of the patients, we identified that they had MSI high tumor status, and it was associated with age in an inverse manner. That is to say that with every 1 year increase in age, there was a 5% reduction in probability of a patient having microsatellite instability. In terms of tumor site, we observed that it was highly associated with ascending colon in compared to descending colon or rectum, as you can see from the graph below. The pie chart showing red is MSI high status, and as you go along from proximal to distal colon and rectum, the probability of MSI decreases. And in terms of family history, it was highly associated with MSI high, with positive family history. In terms of Lynch syndrome prevalence, we identified that nearly 63% of the patients who had MSI high and BRAF V6 negative status, they were likely to have Lynch syndrome. And even in some microsatellite stable tumor patients, we identified that they had Lynch syndrome simply because they had a very, very strong family history. Interestingly, however, in about 80% of the patients, we found MLH1 to be the primary cause of the cancer. And the one patient who had a PMS2 gene mutation, that patient had no family history, and it's partly because PMS2 gene carriers have low penetrance and are likely to be de novo. Interestingly, however, when we further delved into the data, we saw that in MLH1 patients, there were two gene mutations that were occurring at very high rate. One is C154del and one is C306G2T. Now that's higher than expected, and therefore it suggests that there is a potential founder effect going on, but we need to carry out haplotype analysis to prove so. In terms of the impact of this work on public or healthcare, one of the work that we started doing is that in all Lynch syndrome patients that we identify, every year we follow them up on their colonoscopy or the cancer status. And this data is prospectively recorded and is shared with the Prospective Lynch Syndrome or PLSD database worldwide, which is now available to everyone. This is the first time that Indian data is there in the PLSD database. And this helps in prospectively assessing the risk of cancers. So, in conclusion, MSI and Lynch syndrome is likely to have a higher prevalence in India compared to European population. MSI high tumor status is associated with lower age of diagnosis, family history, and right-sided tumor location. MLH1 is the most commonly affected gene in India in Lynch syndrome patients, and two variants are likely to have founder variant status, and therefore it has impact on diagnostic pathway or surveillance. In terms of guidance, we are able to see that NICE guideline has is robust in detection of Lynch syndrome in Indian patients or in Indian population. And in terms of future direction, analysis of MSI and Lynch syndrome prevalence across extracolonic tumors is rather important because it can help in deploying target and cost-effective cancer prevention strategies. This work is not my own. I have had excellent mentors and co-investigators and collaborators from all across India and the world. And I'd like to thank all of them, as well as the funder, GSBTM. And thank you all for listening.
Thank you. Rakesh first.
Dr. Sheth, very interesting data.
Thank you, sir.
But I would suggest caution. At least I would exercise caution when interpreting.
I absolutely agree, sir.
You see certain variations compared to a standard genome in Indian patients with any disease, with a particular disease. Is it possible that if we had taken Indian healthy people, the two variations that were constituted the majority would have been present in most of us? Is it possible that that's a polymorphism that's extremely common in India anyway?
Right.
Just looking at patients with a particular cancer, I don't think we can come to any conclusion.
So, can I answer that question, sir? So, regarding these two variants, C154del and C306G2T, these are classified as pathogenic. They are already observed in Lynch syndrome patients. We know that they lead to or they increase the risk of cancer.
In a particular population, yes. In
No.
Wait. All those data come from the Caucasian population. Right? The fact that those are associated, to think that those are associated with malignancy in our population may not be right.
So that is my second point. That is why I'm coming to my second point. We did look into a healthy population ourselves. We have a database now which is launched by IGIB, and these are not observed in healthy individuals. We then looked at other populations throughout the world, because if it's commonly observed in other populations as well.
Or what is the odds ratio associated with these variations compared to the healthy Indian population? Because we didn't see those data. That's exactly what I'm asking for.
That means to say, if this variant is observed, what is the likelihood that it's observed in affected versus healthy?
In the Indian
No,
population.
Rakesh, I'll take up your question. What you say is right.
What is the clinical implication? You said that this is a policy decision. This would affect the policy decision for offering these patients more aggressive colonoscopic screening, surveillance rather. So these testing should be done in all sporadic colon cancers which we see and get operated.
Sir, MSI testing across all colorectal cancers is now becoming a standard of care across the world, partly because it helps in detection of Lynch syndrome patients, and that has an impact on not only the patient in terms of whether they should receive immunotherapy or not, but also other family members, because you can start giving targeted interventions, cancer preventive interventions.
Only two small this thing.
Sure, sir.
Any of the patients in your registry had metachronous CA, number one?
Yes,
and,
sir, they did.
And secondly, was there any discrepancy ever in between, say, blood levels and, say, ISC done on the tissues?
I'm sorry, I didn't quite catch you.
Blood levels of what?
Say, ISC for the tissue, tumor tissue is negative, and the blood is positive for germline mutation.
Okay. Sir, immunohistochemistry wasn't carried out. In fact, when it comes to colorectal cancer, MSI by PCR has a high sensitivity and specificity. That is why MSI was carried out by PCR across all tumors. But on top of that, we did carry out as a secondary project MSI by next generation sequencing, and we received 100% concordance in all the samples.
Harsh, just the last question.
Yes,
What
sir.
is new about it? We have been doing MSI for such a long time
Sure,
in
sir.
colorectal cancers, and we have few publications on that. What was new in your study?
Sir, there were two important things. In the very beginning,
What
like
is new? One, two.
Okay, number one. In the very beginning, like I said, MSI has been done very well across there are 4 or 5 studies.
MSI,
done,
Lynch syndrome.
But to do a systematic evaluation going from the proportion of patients who have got MSI, out of them, how many had Lynch syndrome?
That has been done already.
No, sir, it has
I'll,
never
I'll,
been.
I'll give you my paper. don't
Sure,
worry.
sir, please. The second part is how many of them have which specific type of variant. That is very important. Thank you.
Okay, thank you very much.
Sir, we thank all the
Thank all the speakers, and a big round of applause for all the speakers, please. All the presenters.
and we want to thank our panel of judges, Dr. Anup Saraya, Dr. Jayanti, and Dr. Akash Pavel. So,
And I thank my co-chair, Dr. Samir. We break up for lunch.
So I think it's post lunch. So it will take some time for people to come. Excellent talk we have. Tricky situation in IBD. I first invite Dr. Vineet Ahuja. Nobody require his introduction. And this is a very important topic, terminal ileitis. Dr. Vineet.
Thank you very much, sir, and thank you for inviting me to Jaipur. So, another slides, please. Yeah. So, good afternoon. I'm supposed to talk about terminal ileitis, like what to do. It is supposed to be a tricky situation in the sense that it's a kind of situation where you can go for the overkill or you may underplay, and both ways you may end up losing. So let me see that, and the data on it is very sparse. So let me see if I can make things simpler by the end. So clearly what we are looking for, the question we are looking for out here is, there is a group of patients with ileal ulcers which show a benign clinical course, and can we identify this group of patients while we are sitting in our OPDs, while we are sitting in our clinics, so that we can take good decisions. So the agenda is, how common is it, when to treat it, when not to treat it, when just to observe it. Summarize all available evidence. Since the data is sparse, it will be easy to do that, and a new algorithm for treating chronic terminal ileitis. Classically, terminal ileitis is defined as isolated involvement of the terminal ileum in the form of erosions, nodularity, superficial ulcers, deep ulcers, without involvement of any other areas, colon or any other small bowel. Today we are talking about that group of terminalitis which does not include acute causes like salmonella or Yersinia, something like that. And remember, one of the commonest causes is NSAID ingestion. So the moment you think about terminal ileitis, the first thing which should come to you instinctively is take history for NSAID ingestion, and if the patient has been taking NSAIDs very frequently, then you know that that is a very important point and that could be the possible cause. So as I told you, it could range from ulcers, small ulcers, superficial ulcers, deep ulcers, or just erosions. And I think it is very important that this kind of distinction is made whether it is a deep ulcer or a superficial ulcer, because subsequently you'll realize that this one factor may have a very important bearing. Biopsy obviously will not make you very wise, because biopsy often in such group of patients would be, if they do not have any specific etiology, would be just non-specific inflammation. So when we talk about how common it is, so you can view it in two settings. A patient who undergoes colonoscopy just because he came for screening for colorectal cancer, you found some terminal ileal erosion, superficial ulcers. There it has been found to be in around 0.5 to 1% of colonoscopies. These are the essentially asymptomatic group. The other is a group which has been referred to gastroenterologists for maybe mild symptoms of IBS, just abdominal pain. You end up doing ileocolonoscopy, and there you find terminal ileal erosions or ulcers. There it has been found to be in 7 to 9% of colonoscopies. And this distinction again becomes very important. So let's first deal with that. So while we start building up the algorithm which we can apply in our OPDs, so the first thing is that we are dealing with chronic causes today. Secondly is, let's first decide, when a patient comes just for screening, you pick it up, that's an asymptomatic patient versus a symptomatic patient. Can it really help you to decide how to move further? A study 10 years back showed 93 patients, a cohort of 93 patients, retrospective study, but these were patients who were asymptomatic individuals. And over a period of 2 to 3 years, what was seen was that 96% of them had a clinically favorable course. Only 2 had developed tuberculosis, only 1 developed Crohn's disease. This is very interesting. Next comes another study. This is another very large study. As I told you, data is sparse, so I will not tell that again, but that is a fact. Like, this is a recent study, 134 asymptomatic healthy subjects, where incidentally terminal ileal ulcers were detected, and biopsy just showed chronic or active ileitis or inflammation. The ulcers were 2 to 3 in number, the size range of 4 to 5 millimeters. They were single, longitudinal, shapes were round, linear and all. And repeat colonoscopy was done in this group of patients. And then a third colonoscopy was also done after another year or so. When the repeat colonoscopy was done somewhere out a median interval of 2 years, it was seen that 50% roughly the patients disappeared, 25% there was a decrease in size, 25% no change. There was no ulcer which increased in size. There was no ulcer which progressed onto some specific etiology. Clearly highlighting that when we decide asymptomatic and symptomatic, that can really be a beacon for us. That can be a very good, simple rule of the thumb for us. The study showed that most TI ulcers observed incidentally, asymptomatic patients showed no findings on biopsy, improved on colonoscopy, on follow-up colonoscopy without treatment. What they found, however, was that ulcers which are star-shaped, in other words, ulcers which seem to be a little bit more pathological, they persisted, although again, they did not cause any problem. Now, let's talk about symptomatic terminal ileitis patients who come to let's say gastroenterologist for some other symptom. So this is a very nice study by Saurabh from our department, which showed that 7% of such patients had terminal ileitis. And again, now you find out here that there was a specific etiology. Obviously, it's a referral bias, but at the same time, these are the patients who come to gastroenterologists. Every gastroenterologist would have a referral bias. It's not that only a tertiary hospital would have a referral bias for such kind of group. So 70% of them had either TB or CD. 30% of them had non-specific etiology. Whereas in the earlier studies where I showed you asymptomatic, the non-specific etiology was 96%, where there nothing happened. A nice study from Ludhiana, Dr. Sood's group, which looked at again around 74 patients, but again symptomatic terminal ileal ulcers. And here again you find that roughly 40-50% are non-specific, but rest 50% of them had a very specific etiology. Another paper from Chennai. This is the world literature I'm talking about. So these are the only papers. So this is a paper from Chennai, Dr. Jayanti's group, which showed that if you have ulcers, if you have erosions, and if you have nodules, then probably if you take a biopsy, the chances of getting a hit, that is a good result, a positive result, no, I won't say good result, a positive result, would be when the patient has ulcers. Suggesting, yes, histology should be part of your algorithm. So, till now the algorithm we have made is terminal ileitis. When we think of the chronic setting, divide them clearly, asymptomatic versus symptomatic. That would help you. In asymptomatic, you do not need any intervention, as a retrospective or observational one prospective study or cohort shows. For symptomatic, now comes the next step. Would you like to find out the etiology? Because obviously that is what your job is. So whether the patient has a specific etiology or a non-specific etiology. This is the old algorithm which I have told you, which we had made. That was Saurabh's paper, which they talked about, where we talked about colonoscopic appearance, that if a patient has deep ulcer, obviously that is more ominous compared to a patient who has superficial ulcer. If a patient has deep ulcer, then straight away go for imaging, and on imaging, and also you do a biopsy and look for specific inflammation, and subsequently you can see whether the patient has significant clinical features in the form of diarrhea, weight loss, GI bleeded fever, and that may help you, and that's how we have been managing the patients. But now, over last 1 year, we thought time has come to revisit that algorithm. Let us see that how does that algorithm behave now, and whether we have gained some understanding or not. So this is a nice study which has been done by Karan Sachdeva. This is Karan out here. He is starting his residency in USA. He's a MAMC graduate, and this is Dr. Samisha, is president of ACG. This poster got the presidential award post in the recent ACG. So obviously, Karan, being a fresh medical graduate, was very ecstatic about it and was sending his photographs. But this is a very nice data which he has compiled. So this is a revised algorithm approach to differentiate between non-specific and specific etiologies of chronic terminal ileitis, because that is what we want to know now. That is the stage we are. So this group, basically this was a cohort again, only cohort of patients who came to Crohn's clinic, not the ulcerative colitis patients, and there were 1625 patients who had a follow-up greater than 6 months, out of which around 153 patients had chronic terminal ileitis, again suggesting that roughly it comes to around 7 to 10%. You exclude out NSAID users, you exclude out patients who got malignancy on the biopsy and all. And when we looked at the univariate and the multivariate analysis for patients who had a specific etiology, that is tuberculosis with Crohn's disease or eosinophilic enteritis, versus patients who had a non-specific etiology, no etiology was found, it was seen that symptoms like abdominal pain, fever, diarrhea, GI bleed were significantly more. Secondly, importantly, as I told you before, deep ulcers were significantly more in patients who had a specific etiology as compared to non-specific etiology. None of the patients with non-specific etiology had a deep ulcer. Ileal thickening, long segment involvement, and necrotic lymph nodes was again more in patients who had a specific etiology. So this is the old algorithm which we had made, and we decided let's, in this cohort of patients, fit in the old algorithm and see whether it is useful or not, or how useful it is. And what you find out here is that overall the accuracy was ranging for around 82% of patients when you wanted to predict a non-specific etiology, and 66% if you wanted to predict a specific etiology. So it was good but not very good. Obviously, time to improve it. So Karan and Samagra from our department, they sat down and did some kind of bootstrap validation. They made nomographs, they did multivariate regression and came out with a model. And in the model, the first step again, which was very important, is that whether you can clearly demarcate a deep ulcer versus a superficial ulcer. That is the first step which should come to your mind. So, because these patients often come to you where you're doing a colonoscopy. So deep ulcer, superficial ulcer. So I'll spend a minute on this slide because this is important. Deep ulcer versus a superficial ulcer. Deep ulcer means there's a specific etiology. Superficial ulcers, again out here, histopathology will be specific in certain group of patients. Then you do imaging. Earlier we were not doing imaging as part of algorithm. Then you do imaging. On imaging you find that if there is long segment involvement or there is thickening or necrotic lymph nodes, it again segregates out a group of patients who would have a specific etiology like TB, CD or something like that. If there are no significant findings on CT, then you look at their symptoms and look at blood in stools, hemoglobin or weight loss, and these three symptoms would help you to further demarcate. Clearly, if you can pick up 97% of the patients out here who had non-specific etiology, where you could simply follow up the patients, and 74% accuracy was there for specific etiologies. When we looked at sensitivity, specificity, 98% sensitivity, overall accuracy 91%, which is not bad considering that this is something which can be done in OPDs and clinics. What about patients who did not have a non-specific etiology? What happened to them? When they were followed up, majority of them did well. Nothing happened to them. There was as I told you, there was 97% accuracy in picking up non-specific etiology people, and when followed up also, they seem to do well. So this is the second part of the algorithm, that is do a multi-modality workup, look at symptoms, colonoscopy, biopsy, then do one imaging, either CT or MR angiography. Thereafter, you can decide divide into two parts: major features and minor features. Major features are GI bleed, easy to remember. This is easy to remember. This is something, if you remember, you can treat every patient in your OPD with terminal ileitis. GI bleed, deep ulcers, specific histology on the biopsy of ulcers, long segment thickening, necrotic lymph nodes. Minor features, weight loss and anemia. What is the importance of that? If you have any major feature, 100% chances are that you're dealing with a specific etiology like TB, CD, eosinophilic enteritis or something like that. In other words, you need to look for the cause. If the patient has only minor features, weight loss and anemia, no deep ulcers, no thickening, then 65% chances are there that the patient still has a specific diagnosis. And if the patient has no features amongst these, out of these 7, none, weight loss, anemia, GI bleed are not there, deep ulcers are not there, CT does not show any long segment thickening or necrotic lymph nodes, then you know that clearly you're dealing with non-specific etiology. 3% chances are there that it could be a specific etiology, and so you can go ahead and treat the patient like that, because as I've already shown, non-specific etiology means that they are going to do well. So presence of one or more major features would rule in a specific diagnosis 100% of the times. There's a good rule of the thumb which you can use in day-to-day practice. Absence of both the major and minor features could rule out a specific diagnosis 96.8% of the times. So once you have a specific diagnosis, what you're going to do, obviously this is an algorithm we all know, I'll very quickly go through it. That is differentiating tuberculosis versus CD based on longer duration, diarrhea, hematochezia, perineal disease favoring Crohn's disease, left as well as right side colonic favoring Crohn's disease, large confluent caseating granulomas favoring TB, lymph nodal necrosis in CT, and chest CT showing pulmonary TB findings, again favoring TB. And PCR is not a good standalone diagnostic test for TB. Gene Expert is a good standalone diagnostic test for TB. And if you have problems, then you give a ATT trial. And if you give a ATT trial, then you do not look for only symptomatic response, but you also look for mucosal responses. Because if you find both responses, then only you would like to go ahead and treat the patient as tuberculosis. So when we're talking about giving ATT trial, earlier we used to say give for 6 months, then do a colonoscopy whenever there's a diagnostic dilemma to look for mucosal healing and confirm the patient has TB. But now we say that, okay, at 8 to 12 months only, go ahead and do a colonoscopy, because it's quite possible mucosal healing if a patient has TB would occur by that time, and you will be able to make that distinction whether the patient has Crohn's disease or the patient has TB. Next question is, what happens if a patient has non-specific etiology? All of you ask this question, should I give rifaximin? Should I give tinidazole? What to do? Well, there's a very good study which has come out from KIMS, Kalinga Institute. This is by Rajkumar. Dr. Acharya is the senior author. Just take 1 minute. So, and this it's a randomized control trial. I've picked it up from preprint. It has not been published as yet. And this shows that when you have superficial erosions, minor, no deep ulcers and all, what do? How do you treat this kind of patients? So they randomize into 2 arms. Rifaximin 550 milligram twice a day, plus albendazole 1 dose, plus tinidazole 1 milligram twice a day for 2 days. And the other group, they gave symptomatic treatment. 26 patients in one arm, 27 patients in an arm. It was fine. The results were same. In other words, whether you give rifaximin or albendazole or tinidazole in this group of patients, provided that you have done a stool over cyst or something, then it is really not going to make an impact. So this study, in a way, provides some answers to that question which we keep on asking. So finally, this is the revised algorithm which can be applied in your day-to-day practice. You have a patient of terminal ileitis, check out, rule out NSAIDs, see that it is chronic, okay, rule out infections and all, like asymptomatic and symptomatic. If it was asymptomatic, incidentally picked up, superficial ulcers and all, just follow up, don't do anything. You will be right most of the times. If it is symptomatic, then you have to decide whether it has a specific etiology or again it has a non-specific etiology. How do you do that? To differentiate between them, you do a multimodality workup. That is the things which you have done. Colonoscopy you have done, biopsy you have taken, you just analyze the symptoms and do imaging. That is the only thing addition I'm talking about. Look for major features, look for minor features. These 7 features, if you remember, GI bleed, deep ulcers, specific histology, long segment thickening, necrotic lymph nodes, weight loss, anemia, if they are not there, then you're dealing with a patient with non-specific etiology. You can just treat the patient symptomatically and follow up. If any of these 2 major, 1 or 2 of the major features are there, then pursue for a definite diagnosis, and the diagnostic accuracy of this would be definitely more than 90%. Thank you very much for your attention.
It's an excellent presentation in a very simplified way. He has explained. Any question?
Vineet. Is that okay?
Yeah.
Very briefly. Any role for capsule endoscopy, Vineet?
I thought I'd put in a slide. Okay, there are two things. That's a very good thing you put up. I thought I'd put in a slide. It has got missed in it. So there are two open-ended questions. One is, do you need a capsule endoscopy? No. You do not need a capsule endoscopy, as I've shown you in the results of our cohort, because if you have done a CT imaging, you are actually able to pinpoint with 100% accuracy. So you may be actually spending more money and not getting the results out of it. Second, is there a role for fecal calprotectin? Fecal calprotectin for asymptomatic, not asymptomatic patients with non-specific etiology, where you're just following up, would be a good test to do every 6 months or every year. If it gets raised, then you know that you have to repeat the colonoscopy. Thank you, Simon, for asking the question.
Sir, we can have question later on. What Govind is saying that after finishing all these talk, then we can have. You can have one question.
No, actually, response to the same question what Simon had put it. You remember that I discussed that protocol with you. Our postgraduate has done the thesis, and it has shown that in a very small percentage of cases, almost 10%, this number is small, but it does change the diagnosis if you do a capsule endoscopy. And all these patients have undergone CT enterography before as a part of the protocol.
So possible. So it will be good to look at that data.
So thank you, Dr. Vineet. So we have a next talk, a very interesting talk, very difficult also, perianal fistulizing Crohn's disease. And I request Dr. A.S. Puri to deliver his talk. It's very interesting and very difficult. I think it will make very simple so that everybody will understand.
Today, in the next 15 minutes or so, I'm just going to be touching on, because in 15 minutes, it's very difficult to cover the entire gamut of fistulizing Crohn's disease. And the first part of my talk is going to be essentially on the radiology, the imaging, classification, and the second part is going to be the management. Radiological anatomy can be very boring and most of us find it. I'll see if I can make it a little more interesting for you. But as far as the definition is concerned, the perianal fistula is an abnormal connection between the epithelial surface of the anal canal and the skin. I'm not taking rectovaginal fistulae and other fistulae. We're just talking about perianal fistulizing disease. So when the pathology lies only in the anal glands, in the cryptogranula, we call it primary. That's not what we're discussing. And secondary, by far the most common would be Crohn's disease. But in India, never forget tuberculosis, occasionally a malignancy. If we take in toto perianal fistula, it's an uncommon, less than 0.01% prevalence in the general population, predominantly young people, for some unknown reason, a male predominance, and the commonest symptoms are pain and discharge. Now, here I want to emphasize that the next patient that you see of perianal fistulizing disease, the chances are that it may not be. It's not rampant, so one shouldn't be just jumping to conclusions. The next patient that you see is going to be Crohn's disease. However, it may be considered as one of the most serious complications of Crohn's disease, and the lifetime risk of developing of the fistula is almost about 30%. Now, it can be the alpha event or it can be the omega event, and this comes from my bringing up in a Catholic school where it was compulsory to attend Mass. So literally the perianal fistula seems to be saying that I am the alpha and I am the omega, the beginning and the end, and the first and the last. So the point that I'm trying to make here is that just might be the initial manifestation of the disease, which occurs in approximately 25% or so, or it can come is what I refer to as the omega event after many, many years. And of late, I've changed my views that previously I used to think that you have very aggressive perianal fistulizing disease, and if there's nothing else, you do a divergent colostomy, divergent ileostomy. And surprisingly, I had a patient who developed that. I'll show you a timeline of that, but this is the point that I was trying to make. The initial event, it's just about, so we can label this as the alpha event. It's only about 25% of patients. And here the lag period means the time of appearance of the fistula to the other manifestations of Crohn's disease can be about 3 to 4 years later. So there would be a lag. And this is the timeline of one of my patients. So you can see as a young lady, she started before she was married, type 2 arthritis, then she has diarrhea, and then the symptoms got worse, and ultimately she failed medical therapy, ended up in ileostomy. And 3 years later, she developed perineal. So literally, this is what I was talking about. This is the omega events that are occurring at the fag end of the disease. And this slide shows you that the more you follow up the patients, the prevalence of perianal fistulizing disease is likely to increase. So if you are at 1 year at this is what is just about 14%. You follow them up for 5 years, the prevalence goes up to 17%. You follow them up even another 5 years, it goes up to almost 20%. The optimal imaging modality, there were 3 contenders. The first thing that backed out was the examination under anesthesia, EUA. And because of its limitations, of course, the surgeon was still very fond of it. They would do it. And we were ultimately left with the MRI and the EUS. And as this cartoon depicts, it's the MRI is on top because the overall accuracy is about 90% versus 80% of the EUS. And you can identify the internal opening with a good 3 Tesla MRI that we're using with a pelvic coil, almost 95 to 97% of the cases. And if you have a severe, some of these patients have severe anal stenosis, then you just can't put the EUS scope aside. I want you to remember this because I picked this up from a surgical textbook, at least 2 of them. I don't know how they picked up the figure that 75% of your continence depends on the external. But that is understood. So because if you damage the external anal sphincter, you are going to end up with incontinence. So the point is, the vast majority of your continence is dependent on the external anal sphincter, and only a small minority of the continence comes from the. So when we are doing cetons, which we're talking about, you just can't use cutting cetons here, because if you cut through the major part of the external anal sphincter, you will end up with incontinence. So at best, the cetons, the cutting cetons, is to be used in a very low fistula. That's the only place. The rest, I think, 95% of the time, we are just using draining cetons. We are not using cutting cetons. Now we come to the anatomy. So this is, of course, a cartoon diagram. And here you can see that that's the levator ani, and the levator ani comes down, and that becomes the external anal sphincter, whereas the internal anal sphincter is a continuation of the circular muscle. The longitudinal muscle stops short. So we have 2 landmarks. One is the internal sphincter, which is coming here, and that's the external sphincter, which has got 3 parts: the subcutaneous, superficial, and deep. And whenever we describe our findings, it's always with reference to the anal clock, where in the males, where the penis would be the 12 o'clock, and this is the coccyx would be the 6 o'clock. So this is T1 weighted image. You can see that that's your levator ani that I showed you, and that's coming down like this. The levator ani has got almost like wings, like an albatross. So that's what it is. And then there's a notch over here that you'll see the puber rectalis, and that's the EAS. So this is in a nutshell. This is, I showed you the earlier image, and this is an axial section. So here you can see you can see the fat. So that means it is not a fat sat image, and the muscles will be black. So what you see over here, the outer layer, that is your external anal sphincter. The inner black thing is the internal anal sphincter, and that white thing between the 2 sphincters is the transsphincteric fat. And this is, of course, in the longitudinal section. So here, coronal sections, and here you can see that this MRI image contrast enhanced. So that white thing that you see is the internal anal sphincter. Now, mind you, the internal anal sphincter, I think it's the size of the terminal phalanx. It's roughly about 4 centimeters. That's about all, 1.5 inches. So this is a big blow up. And so you can see that that's the internal anal sphincter, which is white, and that's the external anal sphincter. And the external anal sphincter almost seems to protest. He says, Radha kyun gori aur main kyun kala? why is the internal sphincter white? And the answer is, as they say, blowing in the wind, because this is striated muscle, this is circular muscle. So the two muscles, because their origin is different, that's why they're different. And all the anatomies that I'm going to show you subsequently is based on these two sphincters. There are several classifications which are used. Parkes, 1976, and then the improvement on that, St. James, but this continues to be the most popular. The AGA, for sheer simplicity, they just classify it on the basis simple and complex. That's as simple as it could be, based on if you have a low fistula, single external opening without any abscess or stricture, that makes it simple, and anything else like a high fistula, multiple openings, etc., that makes it. But I'm going to spend some time on the Parkes classification. The basis of that is the root. What is the root? And the landmarks for that I've already told you, is the IAS, which is the internal anal sphincter, and the external anal sphincter. So based on which way the pus tract is, that is going to, so you can see here, this is your internal anal sphincter. So if it goes between the two sphincters, this is the internal, this is the external, and this is going to be intersphincteric. If it goes across the two, so it's piercing. Now this, you can see this tract is piercing the internal sphincter, it is piercing the external sphincter. This makes it transsphincteric. And this is the point that I was trying to make, that if you put a seton over here, a cutting seton here, then you're going to rip through the external anal sphincter and the patient will become incontinent. So practically everywhere now we're just using draining setons. This is the suprasphincteric, so it pierces the internal sphincter, then goes up to the levator and comes down. And if this type 4, where it is extrasphincteric, so the sphincters are not involved, from the rectum itself it is going through the levator and coming to the skin. And that's type, so this is exactly the same. This is the radiological, you can see the transsphincteric, the intersphincteric, the suprasphincteric. Now, in the original paper, 1976 by Parkes, and he said in his, it's 400, the sample size was 400 patients. So 75%, so you can see that 3/4 of this pie chart is either intersphincteric or transsphincteric. So that is really the commonest two varieties that you get, and then followed by the suprasphincteric. And the least common that you're going to get is the transsphincteric, which is the type 4. And when you have type 4, then the three commonest diseases are mentioned here. St. James, as opposed to Parkes, is based on MRI, and the only addition that they did was they added the secondary extension. So here I've shown you this is a secondary extension, another second, and to compare the two, this is Parkes, nothing about the secondary extensions, and if you have secondary extensions, that's what they added in the St. James. So essentially type 1 is the same, so simple intersphincteric, this is the same as grade 3 is transsphincteric, and this is grade 5 becomes supralevator, and when you have these secondary extensions and abscesses, that's what they added. Now we come to the management. So in the management, when I was a resident as a young faculty, it was no gastroenterologist ever meddled with perianal fistulas. It was totally a surgical disease. And then the MRI and CT came on. So the domain, the surgeon started taking the help of the radiologist, and I think the paradigm shift really occurred when getting the name, Remicade and Infliximab came on into this thing. So when they came on, that's where the team. So now in most places, a team comprising of the radiologist, gastroenterologist, and the GI surgeon would be there. So this is an outline of the treatment. This is essentially a 4-step approach. The step 1 is always the drainage. And here, if you have an abscess, the first thing that you can do, perianal abscess, is drain, and you antibiotics. So that's what you would do as the first step. The stepping stone is drainage. And after about 2 weeks, you put the seton, and thereafter we go to biologics, which was the word was just slipping my mind. So then we go on to biologics, which have actually had a major impact in the management of perianal fistulas, because now actually it's my surgical colleagues who say, would you like to take this patient and give it some other medical therapy before you send it to us? So we are actually getting referred from them. And finally, I'm going to talk on something which is still there in the future, not yet available in India. That is the MSC or the mesenchymal stem cells. And surgery, of course, is always the last step. The antibiotics typically that we use is metronidazole and ciprofloxacin, and this is what has been bothering me. Metronidazole completely anaerobic, quinolones completely against gram-negative bacilli, and for some reason in medical literature in Europe, it's metronidazole always as the first step. I wonder why studies haven't come out combination of the two groups. At least there are no large studies which have compared this. The duration of therapy is 4 weeks for both before you go, but you would apply the cetones approximately 2 to 3 weeks after you've done the drainage and the antibiotic therapy. So this is just to give for the PGs, what is ceton. So here you have an opening, you put in a ceton suture here, and this is a draining ceton, it goes in, comes out, and then you would leave it there anywhere between 4 months to 6 months, and make sure that it doesn't get blocked, because this suture will always allow the pus to drain out, and by and by this should heal thereafter. And this is one of my patients. You can see it's a totally scarred perineum, and that's the ceton suture that's over there, and this is after she's had several sessions of this. Biological therapy, I think, has resulted in the paradigm shift which I was talking about since the 1990s. This is a major impact on the management of perianal fistulas, and the gastroenterologists have literally infiltrated into a purely surgical disease, and this has vastly improved the outcome. The obvious question, what is the choice of biological therapy? The first choice, I think, would be infliximab. The third choice is Usteki, because of it's a recent entrant into India. Probably last month it was launched, and it's right at the bottom because it's the cost. And the second best is adalimumab. And the reason this data is derived from, there are at least more than a dozen studies confirming the efficacy of infliximab, a few of Vido, adalimumab, Usteki. I think possibly the worst results have come from Vido. So that leaves us with infliximab as the top choice, followed by Ada and cost-permitting Ustekinumab. So a biological with an immunomodulator would be the standard of care as of 2023 now. Just 2 slides on what's new on the horizon, and that's the MSC or the mesenchymal stem cells. And it's not available in India, that I can tell you of any personal experience, but in Europe it's definitely, now it's commercially available as Alofisel, marketed by Takeda. It is derived from human adipose cells, and that's the strength 5 million stem cells per ml. And the indication is, now I've underlined this, mildly active luminal disease. So make sure that your luminal disease should not be very active. This is only for the perianal. So with mildly active luminal disease and perianal fistula who have failed at least one biological therapy, the Alofisel is injected through the internal opening of the fistula, and it's thought to act by increasing the anti-inflammatory cells such as the Treg cells, IL-10, and although the exact mechanism probably not very clearly elucidated. There are at least 2 trials, the Admire CD Part 1 and Admire CD Part 2. Because of shortage of time, this is going to be my last slides. Here, 120 million cells injected in each patient. This is versus a placebo, and the clinical response, which I thought was very high in the placebo group, was 55%. At week 52 was 66%, and clinical remission 60%. So this is where it stands now. If it turns out to be a success, I think it will be another game changer. Thank you very much, and sorry for overstepping the time.
Thank you, sir. I think it was excellent talk. We'll have a question answer in the last. The last is Dr. Simon Drevis. I think everybody knows because we are always getting his excellent talks, and we learned from him, all gastroenterologists in India. So topic is acute severe colitis. Dr. Simon.
Thank you very much indeed. What a privilege it is here to be back here. A younger me, of course, as you see in the picture. So tricky situations, acute severe colitis. And let's take a question-based approach. Does the patient have acute severe colitis? Do I start steroids if it might be infective colitis? What about feeding? How do I know if the patient's going to respond? When should I contact the surgeons? What rescue therapy should I use? And should I try again? And whether the patient has acute severe colitis, you would have thought that you know as an experienced clinician when a patient is sick. But sadly we don't, and I get caught out. And you need objective criteria which you use in clinic, the True Love and Wits criteria of the pulse rate, the temperature, and augmented by the CRP or ESR and hemoglobin, because it's so easy to underestimate severity, because patients may well have been on steroids, and we're physicians. We prevaricate. We kick the can down the road if we get half a chance. But the consequences of diagnostic delay are very material, because it's hours that matter. And if you kick the can down the road, it becomes days and then weeks, and that's when disaster comes. And an audit of UK practice in Ealing, I'll name it, it's a place about 20 miles from London, not far, not a remote place, where the mortality in the year 2000 was ordered at 24%, which is the same as the mortality in Sidney Truelove's study in Oxford in 1955. So, does the patient have ASC? The criteria, True Love and Wits criteria on the left there, AIMS criteria on the right, which where Vinita Huja and colleagues looked at their patients with acute severe colitis to say, well, do the True Love and Wits criteria apply in India, where the hemoglobin is materially lower than in the West? And in fact, in their group of 59 patients, you could get rid of the hemoglobin without making a misdiagnosis. And I think that you can use those criteria, or you can just use done some further work looking at fecal calprotectin alone, but it's still worth using those objective criteria. And getting the diagnosis right matters, because whilst acute severe colitis usually complicates ulcerative colitis, it also complicates Clostridium difficile associated diarrhea, CMV, amoebiasis, which you see here far more than I do, as well as infective colitis ischemia. So send off the cultures, send off the hot stool, and do a flexi and send the biopsies. But do please start treatment before the results come back. Because the next question is whether you start steroids if it might be infective, and that happens every day in your practice, I dare say. It certainly happens very frequently in Oxford, and the short answer is yes. Go back to the guru of ulcerative colitis, Sidney Trulove, and he wrote up a paper with Mike Goodman before Clostridium difficile was identified as the cause of pseudomembranous colitis, and he treated these patients with membranous colitis, as it was then called, with steroids. And guess what? They got better, and that was the time when you could get a 6-case report into the BMJ and get it published. But it's better to treat both rather than either alone. But after 30 years of trying to educate my infectious diseases colleagues in Oxford, I have to admit failure, because they still just treat with antibiotics and then say it's not working and then think, oh, could it be ulcerative colitis? Much better to treat both rather than either alone, unless it's that. No prizes for guessing, but it's a hot stool to the lab, and you'll pick up the hematophagous macrophages very easily with the experienced eye. So this is standard practice in 2023 of the management of acute severe colitis. Hospitalize the patient, do a plain abdominal film flexion biopsy, start steroids. We give rectal steroids as well as intravenous steroids, antibiotics if they're high risk, and I suspect that applies to all patients in India. Although in the UK we don't give antibiotics routinely unless we seriously suspect that there may be an infective exacerbation, or if they're the first attack, venous thrombosis prophylaxis, and then after that, the general process towards a surgical decision is a matter of days, between 5 and 8 days. You heard from Vinita Gujja yesterday about venous thromboembolism and the importance of prophylaxis, even in an Indian population, because the risk is threefold higher than other hospitalized patients. Do your pre-screening tests and start enteral nutrition, because that answers the third question, which is, what about feeding? And traditionally in Oxford, we have tended to keep people nil by mouth, give them intravenous fluids, not because we feel that it improved the outcome, but because it helped decision making. Now, then last year, there was a randomized controlled trial driven from AIMS, which compared patients with acute severe colitis who who were randomized in an open manner to receive either enteral nutrition with an oligomeric feed or standard diet. And what was really fascinating about this was that although all the patients had a standard of care of intravenous steroids, then the outcome was steroid failure as defined by need for escalation therapy or surgery. You can see that the patients who had the enteral feed in the green bars, they had a lower steroid failure rate on the ITT analysis, which almost reached significance compared to those who just had the intravenous steroids and standard diet alone. On per protocol analysis, because 3 patients couldn't tolerate the enteral nutrition, then there was a clear benefit of enteral feeding, and this has changed our practice. So in Oxford now, we give patients enteral nutrition when they come in because it improves the steroid response rate. You'll notice that there was no difference in the colectomy rate, but numbers were small, and the great advantage of the enteral feeding was that it also had a sustained effect with improvement in biological markers and also reduction in readmission rate over the following 6 months. And so will the patient respond? And I draw attention to some further work also between Oxford and India, which is still online in Gut, which was the Admit ASC index. More indices, yes, more indices, but you need these to guide your judgment. And it's a really simple index. Sanghi in Oxford was driving this, and where patients were given a score out of 4 points. That's 1 point if their CRP was notably elevated on mission over 100, 1 point if it was less than 25, and 2 points if they had very severe ulcerative colitis endoscopically, UCIS of 7 out of 8, and only 1 point if it was 4 or more. And you'll see that the discovery cohort there, compared with the first validation cohort we did in the Gold Coast in Australia, and then the second validation at Delhi when Gut rejected the paper on the first occasion, and this works in Delhi just as well as it works in Gold Coast, just as well as in Oxford. And if you've got a score of 4, then there's 100% chance of steroid failure, and this is applied on admission. And that is a great advantage, and therefore if you score 4 or more, you know that the patient is not going to respond to steroids. You need to move on. And what was interesting about this was that since we first published our we did the first predictive index, the Oxford Index, back in 1996, it's interesting to compare subsequent iterations of the Oxford experience in the proportion of patients who had rescue therapy. And back in 1996, it was a quarter who had rescue therapy, largely cyclosporin. Here in 2015, 2019, with Vipin Gupta from AIMS also contributing, a half patients had rescue therapy. And if you look at the colectomy rate on the right, it was 29% in 1996, and it's halved in recent years. And that must be a relation. Double the rescue therapy rate, halve the colectomy rate. Yes, on the third day you can use the Oxford or indeed the AIMS index, which is an ulcerative colitis and UCIS index. But the point that I'm making here is that whatever index you use, for goodness sake, keep it simple and apply it, because you've got to help it to make decisions. So what about the surgeons? Clearly, if they've got a dilated colon admission, you need to get the surgeons involved, because only half of those respond to steroids. And clearly, if the admit ASC index is 4, you get the surgeons involved at an early stage. You have a chance on the third day using the Oxford Index or the AIMS Index and get them involved, but that's a cue to start rescue therapy. And you've got to consider the pattern of disease. Is it first attack, relapse of established disease? And remember, if you are on your own in a peripheral hospital, then you're only a phone call away from getting advice. And get advice on that third day. Don't wait until 5 days a week has gone past, or often more, before calling for advice and help. And you do need a place where you can get surgeons, because you need to decide, the longer you delay, as this study from Canada shows, the longer you delay the number of preoperative admission days, the mortality goes up. And it's all about saving lives. It's not about saving colons. Which choice? It's either cyclosporin or infliximab. Lots of factors come into that. Affordability comes into it. But bear in mind tofacitinib in India, because it's cheap, it is available, and at a dose of 30 milligrams daily. Then Peter Higgins in Ann Arbor has suggested that that it is effective. It hasn't been subjected to a randomized controlled trial at the moment, though that's in progress, and it's all based against historical controls. But it's a useful, cost-effective choice if patients can't afford the biologic. Just as an illustration of the real world, we had a patient who was barely fit for a haircut, let alone a colectomy. She was 67, but biologically much older, non-response to steroids for acute severe colitis. She had already lost response to cyclosporin and vedolizumab. She had diabetes, breast cancer, and we gave her tofacitinib, and hey presto, that was next. It all came crashing down. This is the simple clinical colitis activity index, came crashing down, and then we changed to 5 milligrams twice daily, got worse again, we doubled the dose, and she's been fine ever since. So, do you try again? Retrospective studies suggest that there is no benefit in doubling the dose of infliximab. This is a meta-analysis amongst 3 centers in the US, in that there was absolutely no difference in the colectomy rate between standard 5 milligrams a kilogram or higher doses, and no difference in colectomy in subsequent events. And I don't think there's an advantage in just giving more infliximab. And if you add in a second rescue therapy, I think the bottom line is simple. Don't try and rescue the rescue therapy on the same admission. Fair enough if it's a subsequent admission, but don't rescue the rescuer, otherwise one of them will not work and come to colectomy. So, just to conclude, the messages I hope to take home are: use objective criteria for making the diagnosis, use an index to predict response, the Admit ASC, look it up, and or you can use the Oxford or Ames index. We validated it in India, and it all relates to the severity of the disease. Do add enteral nutrition to your intravenous steroids, but act on the results. Get a surgeon involved. Cyclosporin works as well as infliximab, and don't rescue the rescuers. Go for colectomy at that stage. Don't duck difficult decisions. Pick up the phone and phone a friend. Thank you very much indeed.
Thank you, sir. Thank you very much. We can have questions for all three speakers. Yes. You can have mic. Yes. You can come here. You can come. Yes.
Can I ask the question?
Yes, please.
This is for Simon. Yesterday you spoke about us choosing drugs, the biologics, in the order in which they have been manufactured. In your setting, would you want to change? Would you think of anti-integrins?
Yeah, I think it's a fair question. We use infliximab because it works rapidly, and yes, it's chronologically the first one which was licensed. Although cyclosporin before that, the tofacitinib really does appeal as an option, but I must say in Oxford we use cyclosporin as long as the patient hasn't been on azathioprine. If the patient's already been on azathioprine, then we use infliximab. But people feel confident with infliximab. Trouble is, it doesn't get out of the circulation quickly, whereas both tofacitinib and cyclosporin do. So I'm looking forward to the first RCT in tofacitinib.
Because you don't know whether the drug has failed or the patient was going for a colectomy anyway.
Yeah, I think that and there can be a circular argument if you send in the colectomy, but it comes back to the question that it's colectomy that saves the life of the patient, and if physicians delay, they contribute to the surgical complications. And then, of course, when the patient has surgical complications, you blame the surgeon, and you say, well, I said surgery was dangerous anyway. Actually, it's the physician who was at fault.
A question?
While you're still there, can I ask you another question? Right? Yeah.
One for you, Dr. Puri.
Sure.
I think we need to escalate the dose of infliximab in perianal fistulizing disease. So would you want to make a comment on therapeutic drug monitoring when we are dosing a patient for perianal? I think the doses are, we have to give more infliximab that works better than the routine.
Okay. Am I audible?
Yeah.
So the first thing is that the availability of TDM in India is so limited that that becomes a problem. The point that you made is absolutely correct. Those patients who have perianal fistulizing disease, they are the ones who probably need more aggressive, and they are the ones who need proactive, not for the sake of doing it. So they are the ones that we would actually recommend. And yes, if you have that facility, by all means, you would need to either to step up the dose or to shorten the interval.
Would you then suggest a higher dose upfront? Instead of 5 milligrams, would you suggest a higher dose?
Here again, you'll have to balance the fact that in India, there is the risk of tuberculosis and other, opportunistic infections is much higher than the West. I think the figure for India is somewhere between 8%, 9%, or just short of 10%, somewhere in that range. So if you step up the dose of the Remicade, and it's not recommended. I don't think I've read it anywhere that upfront you start just because of the fact. Yes, if the patient is not doing well, then you can do a TDM. That point is well taken.
Thank you.
Yeah, we find about 25% of our patients using TDM, and we can do it, and we have easy access to it. We do it before every infusion. We find ourselves escalating, and so for perianal fistulae, we aim for the top quartile. Surely, given the expense of biologics here in India, that since TDM is accessible, you can measure infliximab levels. You may have to send them away. They may be expensive. Isn't it cheaper to do that when a patient is failing before just routinely escalating the dose?
Vineet can correct me if I'm wrong, or maybe.
Yeah.
But I think there are probably only 2 centers doing TDM.
Oh, is that the case? I didn't
In
know
the whole
that.
of India.
Okay.
So the sample has to go,
Yeah,
and
yeah.
it goes to Mumbai that I know,
Yeah.
and one goes to Hyderabad probably. So they are the only 2 centers. I don't know if AIMS is doing it as yet. And the other thing is that the TDM that Dr. Salanke was asking, we want to keep the Remicade level around 7 microgram. I'm using the word Remicade. Infliximab level around 7 microgram. But in the patients with perianal fistula disease, the level has to be slightly higher.
Yeah.
So we'd aim for a much higher value as compared to 7, where 7 is good enough for the, most of the conditions where we're using infliximab.
And make sure they're on azathioprine, of course, as well, because protect what's working is such an important
Yes,
principle.
I'm a great fan of the Sonic and the Success. So I would use practically everywhere. I wouldn't. My patients wouldn't be on monotherapy with the biologicals. But I wanted to ask you, Simon, since yesterday you had said one of them, possibly in terms of volume, the biologic that you're using maximum is Vido.
Yeah, that's for ulcerative
And what
colitis.
is the what is the success rate of Vido with perianal fistula?
Oh gosh, now you're asking. I think that the vedolizumab is gut specific, but tell me where the gut starts and where it ends. But I think perianal fistulae and vedolizumab, the results are very poor. I think that there was a signal from their original registration study that it might work in Crohn's disease. Actually, in experience, Takeda pulled the study, which was looking at randomized controlled trial, and we reckon fistulae and Vido don't go. I like vedolizumab for inflammatory Crohn's disease, not for visualizing Crohn's disease, and I'm afraid ustekinumab looks as though it's going the same way. We hope that upadacitinib or filgotinib, the JAK1 inhibitors which are effective for Crohn's disease, we hope that they will work for fistulae, but we don't know the answer just yet. There's massive unmet need, as you indicated.
So in fact, you validated my slide when I said the first choice for first choice biological would be infliximab followed by ADA.
Completely. Absolutely agree and with everything you say. And I think that MSCs is a triumph of hope over expectation, but we shall see.
Clinical trial with MSC. We finished a clinical trial with MSC, 10 patients.
Yeah.
So till now the results have been good, almost like 50 to 60%.
Really?
And these are the results till for 24 weeks, 52 weeks, and 104 weeks. So
Yeah.
104 weeks we have not completed, but till 52 weeks the results are decent, 50%, 60%.
Well, that's encouraging because perianal disease seems to get more common as you go east, and certainly you mentioned the paper by Zai and so on. I mean that's East Asia. It's very common indeed. There's massive unmet need.
I think one point again needs to be stressed out here while you're talking about giving more biologics is that most of the times we start biologics without treating infection in perianal disease. So
Yeah.
that is an equally important facet. So infection should be treated, a ceton thread should be put in, and you should give an antibiotics. Biologics are never going to work even if you give super high doses if there's an infection in the patient.
That is the point that I had clarified very clearly. That and Simon, your take on this. So the perianal flora abscess is essentially a GNB, a gram-negative bacilli, and the anaerobes.
So no, we use metronidazole augmentin actually. I liked your point about using dual antibiotics. Great, leave it. But we actually, for what it's worth, use augmentin coamoxiclav rather than ciprofloxacin. But we use metronidazole and something against gram-negatives as well.
I can see there's a microbiologist sitting here, but to the best of my knowledge, augmentin has very little activity against anaerobic activity. And
Oh no! But then you got your metronidazole.
you're using a combination of
Oh yes, of course, yes, absolutely. No, that's why I liked what you said about using combination. Completely agree with that.
Okay, sir. Anything? Any question? I want to ask simple question. IBS you will consider in symptomatic or asymptomatic? Second thing was how many ulceration, how long you should go in ileoscopy to see, and whether that length of involvement with ulceration make your difference.
IBS will consider in symptomatic because there would be abdominal pain or diarrhea. That's what you would be doing a colonoscopy. Number two, it's a superficial ulcer versus a deep ulcer. Number of ulcers and all does not matter. Obviously, if the florid ulcers, it's a different.
Thank you.
Sir, my question is to Dr. A.S. Puri, sir. Sir has mentioned that perineal fistula may precede Crohn's disease many years before. So, all perineal fistulas do merit investigation of colonoscopy if there is no obvious cause is found. And second question is
the short answer to that is yes, because if I have a perineal fistula, the first thing I want to find out is the rectum involved or not. Because in perineal fistula, rectal involvement is seen in 75% of the patients would be there if it's Crohn's disease. Whereas if you have ileal Crohn's disease and you have perineal fistula, that probably would be a very, very small minority. So definitely you would want to know whether the rectum is involved. It's just an isolated cryptograngular perineal fistula and abscess, or is it something more sinister? So does that answer your question?
Yes.
Okay.
I think we should finish this session. Thank you very much to all speakers. I think it was a very wonderful experience we had. Thank you very much.
Thank you for the session. Thank you, chairpersons, and thank you, speakers. For the next session on functional gastrointestinal diseases, may I please invite Dr. Chetan Bhatt, Dr. G.S. Lamba, and Dr. Manish Palwal for chairing this session. I would like to also announce that the alumni meeting will take place from 7 to 7:30 in hall number D for SDMH Institute, Vellore in U1, AIMS in U2, PGI in U6, and SGPGI in hall E. I repeat, from timing 7 p.m. to 7:30. Thank you.
Good afternoon, everybody. We start the session on functional gastrointestinal diseases, and we have 3 lectures in this, and the discussion and the question answers will be after all the 3 speakers have finished their talk. So because last session overtook the time, so I'll start straight away. The first lecture we have is by Dr. M. Srinivas, and he's going to speak on multi-dimensional clinical profile of IBS, its importance in management, including the mantras. Right, Dr. Srinivas, please.
Thank you, chairpersons. Thank you to the organizers for giving me this opportunity. So without much ado, I'm going to talk to you today about multi-dimensional clinical profile of irritable bowel syndrome and its importance in management. I put there, at the end of it, we want to know, is it too many cooks or is it strength in numbers in management? So I have a disclaimer before I start. I'm not going to bore you with Rome 1, 2, 3, 4, 6, 8, all those things. So as we all know, irritable bowel syndrome has a population prevalence of anywhere between 4 to 9%, and in India it's around 5 to 10%. Many patients actually have overlaps between fecal evacuation disorders, functional diarrhea, functional constipation. So we have to look at all of these together as functional GI disease. And actually in a non-complainant population questionnaire study on IT group in Chennai, we actually found that in 957 participants, about 18% actually felt that they had functional constipation and only 5% felt they had irritable bowel syndrome. This is a non-complaint population. And I would say that Indian data on interventions is lacking. We have a lot of studies on clinical profile, prevalence and all those, but interventional data is lacking. So, are we meeting the expectations of our patients? We all see a lot of pain, like I showed in those cartoons. Sometimes we feel like telling them those things. So there was a survey of 1,000 plus irritable bowel syndrome patients. 90% of the patients want the doctor to listen, answer their questions, provide treatment information. Now we can all introspect. I will tell you the answers. 64% patients felt listened. Less than half felt supported. And only 40% felt that they were given satisfactory information. So, okay, this is what the patient thinks of us. we are actually giving them so many happy pills and various medications. Are these interventions working? There was a symptom improvement survey in 102 functional GI disease patients. This is all comers, not just IBS. This was done 1 year after specialist GI consult in a study from Melbourne. Only a third felt better at the end of 1 year. Dyspepsia patients were worse off than the other FGIDs, with 21% only feeling better. Of the diet that were used, FODMAP group did better, but even that only 50% felt better. PPIs, the bulwark of any gastroenterologist and any pharmacist, the results were very, very poor. Only 22% patients at the end of 1 year felt that the intervention was working. So clearly, our interventions are not working. So I put it to you, and I'm sure most of you will agree with me, that functional GI disorders are a GI physician's Waterloo. The IBS is the pin waiting to prick the balloon of our self-esteem. So why are we struggling? We need to know. For that, we need to understand the complexity of the pathophysiology of this disorder. This picture you see is common for most disorders of the gut. Yes, environmental factors, genetics, we talk about that even in IBD. Gut physiology, we talk about that even in IBD. Psychosocial factors, we talk about that in IBD. But in irritable bowel syndrome, it is not just these factors, but also how the patient perceives their symptoms, how they react to these symptoms, and what they feel is happening to their quality of life as an interplay of what is happening in their lives dictates what happens. And of course, in other disorders, we have structural problems. With treatment, these improve and the patient may feel better. So in order to treat these, we have various things in our armamentarium, ranging from laxatives, antidiarrheals, anticholinergics, and then some other ones like lubiprostone, alosetron, and then various, I call them happy pills, like amitriptyline, venlafaxine, sertraline, and also some other centrally acting agents like gabapentin. While we may be able to use all these things to manage the patient's gut physiology and to an extent use the dietary modifications, there is this big elephant in the room, which is psychosocial factors, which cannot be managed with medications. And that requires various other modalities, as you see there, which include psychodynamic therapy, hypnosis, stress reduction, psychiatry consult, etc. So clearly, this is a multimodal disorder. Will multimodal approach work in irritable bowel syndrome? The answer to this question is we do not know. Why do we not know? Because we did not have any RCTs on this. Everything was anecdotal. So now, this study, the MANTRA study, published in 2020, was the first RCT. Do not ask me the expansion of the acronym MANTRA, because that doesn't have any bearing on the actual title of the study. I tried my best to read the paper and I couldn't find. So this study has been done in Melbourne. It is an open label, single center, randomized control trial. Who are these patients? These were patients who have functional GI disorders. They were divided into two arms, a standard gastroenterologist treating as he normally does, and then a multidisciplinary treatment with 1:2 enrollment. The MDT included a GI physician, psychiatrist, dietitian, pelvic floor physiotherapist, and a hypnotherapist. The study cohort, although they screened 1,632 patients, after applying various exclusion criteria, they ended up with 46 in the standard arm and 98 in the multidisciplinary treatment arm. And the primary outcome was a symptom improvement on Likert scale of 4 to 5 out of 5. That is, I feel very good or excellent. So what did they find? So if you actually look here, the functional GI disorder group with the MDT approach, actually there was significant improvement in symptoms irrespective of what outcome you looked at. Further, when you did a subgroup analysis, it was found that the patients in the IBS group were more likely to feel better with a multidisciplinary approach. So of course this study had its limitations. It was, for starter, a single center study. and it was not powered adequately to compare subgroups. So therefore, this subgroup analysis has to be taken with a pinch of salt. And we thought we had only one study, but then there is another study which was published last year in the Digestive Disease and Science, again a matched cohort study coming from Australia, but another center, Gerald Hodgman Center in Brisbane, where they looked at functional GI disorders, that is functional dyspepsia and irritable bowel syndrome. Standard care non-responders were recruited in this study. A 12-week multidiscipline integrated care program involving a GI, primary care physician, psychologist, dietitian, and an exercise physiologist. They compared this with a standard care group. Again, it was 1:2 randomization, so 52 versus 104. Pre and post treatment, the patients were evaluated for their GI symptoms with a SAGI score and for anxiety depression with a HAD score. So what we find here is the patients did in the multidisciplinary team, in the multidisciplinary treatment arm, did well after treatment in their SAGI score and also in the HADS, anxiety and depression score. So there was significant improvement in the MDT approach. However, there is a caveat. Patients with higher SAGI score who were 28 in the MDT arm actually dropped out. So if they had been included, we do not know. It might have looked much more spectacular or worse. We do not know the answer to that. And again, of course, this is a single center study. So now we have two randomized controlled trials which suddenly tell us that multidisciplinary team approach works. So what are the roles in an MDT approach? Obviously, as GI, they come to us first. I would say that we confirm that it is a functional GI disorder by doing some basic investigations, and we can manage the gut regulators like the loperamide and the cholestyramine and all those things. And a psychiatrist will assess and optimize the central agents, which is amitriptyline, pregabalin, and all those medications. The physician is very, very important in this because many patients have multiple other comorbidities which need to be addressing. And the dietitian will do the food manipulation with FODMAP diet, lactose-free diet, and all other dietary manipulations. The psychophysiologist will work on cognitive behavior therapy, general counseling, and a committed physio can work on biofeedback if the patient has fecal evacuation disorders, exercises to strengthen the pelvic muscles, yoga. So we have all these people who can do all this. Now I am going to tell you about something. See, this lady you see there was Avaya. She was a 10th century saint poet, the Chola dynasty. And of course, the other gentleman there is Henry Ford from the 20th century, who revolutionized the car manufacturing industry. Avaya said, Kattrathu kaiman alavu, kalladadu ulagalavu, which translates as, what is learnt is a fistful, what is not learnt is universal. So basically what it tells us is, recognize limitations. Yes, I have recognized my limitations. I know I can only give some medications. I cannot manage the other aspects of the patient. So what can I do about it? I can't stop there and tell the patient, go and see a better doctor. So that is where Henry Ford said, coming together is a beginning, staying together is progress, working together is success. This can apply in various fields, including a good married life. But here we are talking about teamwork. So we need to recognize limitations and overcome it with teamwork. So ultimately, it is the patient satisfaction that matters in any functional GI disorder. If it is a structural problem, me, a gastroenterologist, is sufficient. But having said that, nowadays, in structural diseases like inflammatory bowel disease, celiac disease, we have to work as a multidisciplinary team to make the patient feel better. But when it is a multimodal problem, such as functional GI disease, the gastroenterologist may at best be a hub with multiple spokes. So these are my spokes. My psychiatrist, Dr. Siddhika, my physician, Dr. Ashwin, my dietitian, Dr. Lakshmi, my psychotherapist, Ms. Sayanthini, and my physiotherapist, Dr. Uma. You actually see my dietitian and physiotherapist have doctors in front of their names. They are PhDs. So clearly, they know what they are doing. And each one is a specialist role. And my patient expects to see a master for each of them and not me a jack of all arts. So now coming back to my balloon. So the irritable bowel syndrome is ready to prick the balloon, but when I place this shield of MDT approach, I not only retain my self-esteem, but more importantly, my patient regains his self-esteem. So a healthy patient makes a happy doctor. So ladies and gentlemen, I tell you, irritable bowel syndrome has multi-dimensional stimuli and effects. It requires a multimodal approach. One-step functional GI disorder clinics are the future. So I asked you in the beginning, is it a case of too many cooks or is it strength in numbers? I'm sure you'll agree with me that strength in numbers is a definite yes. So every conference that I go to, I aim to take something home with me which I can work on the next 12 months. So if there is something that you want to take away from this meeting, I put it to you, go back to your centers, your standalone clinics, your hospitals, start looking around for interested psychiatrists, general physicians, dietitians, psychotherapists, work with them, set up a team, and you act as the hub and they can be your spokes. Thank you.
Thank you, Dr. Srinivas, for your excellent talk and finishing well in time. So I'd like to invite the second speaker now, and he's Dr. Kevin Shah, and he will be talking on diet in IBS, what to eat, what not, and how difficult it is to adhere. A very important clinical talk.
Thank you. Thank you, organizers, and thank you, chairpersons, for introducing me. Before we can set up a multidisciplinary team, let's see what we can do in our clinic for maybe 5 minutes or 1 minute if you dare talk to the patient about diet. Why do we need to talk about diet? Well, I was given this topic, that's why I need to talk about it, but why do you need to talk about it? Because the patients will think that diet or food is the cause of their problem anyway. If you don't talk about diet, a lot of them will not think that they can cure. Although stress is not a big issue, but food definitely is the major reason or trigger for a lot of our IBS patients. What else? A lot of patients are doing self-control or self-dietary modification that may not be safe. So without guidance, they may have problems. It is not something that is without harm. So how many patients actually, when come and see you in your clinic, have already tried diet plan? Actually, most of them could have tried. And they have tried different things. Whether it's the right one or not, we're not sure. For example, a lot of them would have tried lactose-free diet, gluten-free diet, a low-FODMAP diet, or vegetarian diet, various diet. But are they the right one to try? And are they doing it correctly? So that's why we need to talk about diet in IBS. And we also need to talk about it because many studies have shown that patients who have IBS actually lower intake of many foods. For example, milk, yogurt, fruits, and a lot of micronutrients. So if we leave it to patients to do it themselves, we will have problems. So just now we talk about what Dr. Srinivas talked about, the needs of having a multidisciplinary team to help us with IBS treatments. It is true for that you need a good GI dietitians, but not all of us have the luxury of having a GI dietitian. I bet of all of you here, maybe less than 5% will have a good GI dietitian in your hospital. That's what I think. This is the same in other countries that I have talked to and different people I have talked to. Unless you're a tertiary hospital, it's really unlikely that you have a GI dietitian that is, that can talk about lactose intolerance, that can talk about low-FODMAP diet with your patient. Most of the time, you are the one that need to spend time with the patient. And most of us don't have the time. And what about the major societies? What do the major societies tell us about diet control? So, for the recent guidelines from UK, USA, Canada tell us that, traditional dietary advice works. And some of them, they don't recommend gluten-free diet, but a lot of them also talk about low-FODMAP diet. That's probably why the patient, pick up low-FODMAP diet, and this is also a very popular diet things in a lot of the magazines. So, I like to use 5 minutes normally to talk to the patient about what to eat. And the real things after we look through is actually there's quite little thing that you can recommend them what to eat. One thing that we all know is fiber. But it's important to let the patient always tell you that, oh, I have a lot of fibers already. But the important thing is, are they the right fiber? So I don't need to tell people here that there are insoluble fiber and soluble fiber. And it is the soluble fiber right now that is important. Although there are increasing studies that says that we also need insoluble fiber for the gut microbiota. But most of the time, you have to remember that if they don't have enough soluble fiber, it is the one that we need. But recently, this is just within the last 1 month. A study in American Journal of Gastroenterology shows that if you consume 2 green kiwi fruit daily, you improve your constipation and abdominal comfort. And this is a multi-center study. This is the only study or only fruits that I found that you can recommend them to eat. But I'm not sure about you. In Singapore, kiwi fruit is pretty expensive. So it's good for FC, IBS-C, and combined combination, and they compare it with psyllium, which is your soluble fiber. So an apple a day keep you closer to your IBS doctor because it's high FODMAP. Maybe 2 green kiwi a day keep you further away from your IBS. New Zealand didn't sponsor me for this study, although it's supported for other study. More important than what to eat, actually, is how to eat. I think this is what all we can do. This is a chart that I did myself. It is important to know how to eat. The timing of meal is very important. Having regular meals and take time to eat. Fluid intake. You shouldn't have 8 cups of water. I don't know where it's from, but 8 cups of fluid is what is recommended. I'm trying to find the evidence. I still couldn't find. If you know the answer, please send an email to me. And food and fiber intake, probiotics, refer to a dietitian. But the simple thing is that what we can do is timing of meals. How do you have regular meals? But I realized that I was in Calcutta and now I'm here in Jaipur. The time of dinner is really not that ideal for your stomach. People eat at 9, 10 o'clock and then you go to sleep with full stomach, then it's going to have problem. Well, it's just an observation anyway. And what not to eat. This is the part that actually, what we know most and what the patient knows most about as well. And this is also the part that, is important because a lot of people think that they have trigger food. So normally we tell the patient to eliminate trigger food, for example, fatty food or fried food, and those kind of things. These are very common. But they are also the food that is very delicious. So it's difficult to tell the patient to stop. Maybe you just stop eating all the things that you like to eat. Just eat things that you don't like to eat. Then within 1 minute, you can solve your problem. But no, it's difficult. Gluten. I always say that no crime is not equal to low crime. So although a lot of us, gluten is not really recommended, but there are also another entity called non-celiac gluten sensitivity. We don't have time to explore on that, but there are some patients that may be benefit for it. But although a lot of the IBS guidelines doesn't recognize and do not actually recommend it, and it's actually very difficult to do gluten-free diet anyway. And those, even the product with gluten-free packaging on them, some of them actually have gluten in them. So be careful. FODMAP is has been a very cool thing for very long, and we don't want to spend too much time. You know about the mechanism. Professor Uday there is experts on talking about mechanism of FODMAPs, but what we want to talk about is it easy to do? The answer is no. Why? It is not easy to do it properly. The principle is important. You need to reduce the dose of FODMAP consumed over a day to achieve the symptom control and replacement. High in FODMAPs can then be replaced with dose is low. So these are the main principle of doing a low FODMAP diet. But you don't have time to talk to the patient the whole thing because you can't do it within 5 minutes. You need someone who is trained in low FODMAP diet to do that because it includes FODMAP introduction and FODMAP personalization. This is a long process. A lot of us don't have time to do that. Well, in my clinic, I normally just look at them and do you eat a lot of mango? Do you eat a lot of apple? Maybe you cut that down first. That's the main thing. But when you say that, what are you actually talking about? So these are the full challenge schedule. Nobody can do. Very few people can do it in your clinic unless you have a GI dietitian that helps you. And the reason why, because FODMAP is in a lot of food, right? Just now we have a lot of roti, naan. These are all full of FODMAPs, high FODMAPs. And this is the problem if you want to ask a patient to cut it. And a lot of our popular food actually have very high FODMAP diet, like curry, stews, other countries, pho, and wasabi noodles. And if you ask someone to cut their diet, they're not going to be able to do it. Just like when you ask someone to stop smoking without giving them the advice or having another route to do it, at least on smoking they have a patch. We don't have a low FODMAP patch. Maybe you should have a FODMAP patch. That would be a good idea. But the problem is we don't have a replacement. When you don't have a replacement, the patient just ask the patient to do it, they won't do it. And when we talk about a lot of Apple things, because there are still other things that we don't recognize. For example, there may be sucrose intolerance in IBS, lactose, maltose, glucose, and fructose. And there are hidden sources of all these sugars that we may not know. For example, in Milo, Horlicks, or in milk. And how many people can tell how much lactose in this fruit? I didn't know it fell, and I had to find out. So, for example, yogurt have 9.3 grams. Your ice cream, just now the ice cream was quite nice, 5.7 grams and 2.6 grams. And your burger, even the burger have 1.1 gram. So there are a lot of hidden sugars or lactose that we didn't know. And if the patient do practice diet control now, and the hottest topic in the world, globally now, is ARFID. ARFID is not a person's name; it's actually a problem. It's a problem that is caused by avoidant restrictive food intake disorder symptom. Some of the patients that you see them, they'll tell you that I've already stopped eating this, eating that. You probably have seen them and recognized some of these patients in our clinic, but it's just not that obvious. But now, this is the main problem that some of us are seeing in clinic. Patients stop eating certain food, and then they are suffering from ARFID. Nice? Anyway. And with ARFID, then the patient, you need to understand what are they facing, not just food. They may have problem with psychology, they may have problem with social behavior, they may not have problem, may not be able to spend time, and they have fear of eating. And we published a paper looking at problems of patients who, when they stop doing normal diet as well, so they may have suffered from macronutrient deficiency. So with patients who have a lot of extreme diet, vitamin B1, B2, calcium, iron, and zinc may be a problem with this patient. And we don't normally check this kind of macronutrient deficiency all the time. And then we come to dietitian. Dr. Sindra very nicely talk about the role of a multidisciplinary team. It is important to invest in a good dietitian. But the problem is, most of the dietitians, they prefer to see diabetic patients. They don't like to see IBS patients. Why? Because at least my dietitian, my GI dietitian told me that, well, most of the time, the dietitian themselves become a psychologist also. So they don't like it because they found out that instead of talking about food, nutrient and stuff, they have to be counselors, just like any of the doctors here who see a lot of IBS patients. You become the counselors. And the dietitian feel the same. So they prefer to see hypertension, diabetes patients who actually want to change, rather than seeing IBS patients. It's tough. And that's why, although we have multiple circles, and that's why we need to understand the role of dietitian ourselves, and maybe having 1 second just to talk about them. But do not worry, even if you don't know about low FODMAP, we don't have a GI dietitian. If you know about, for example, a good dietary habit, just talking about when to eat, how to eat, and this kind of normal thing, study shows that the effect is just as good compared to low FODMAP. Obviously, more study will be done, and this is just a few good to tell that if you can talk to patients about importance of good meal time, good traditional IBS diet advice that's been there for very long, it does a good result. You don't need to have sophisticated low FODMAP diet to do that. We have that the IBS guideline dietitian, very nice one. I did it myself, and it's on my own Twitter account, Kevin Sia, a bit of advertisement. And we know that the Indian Neurogastro Society also have a very nice Twitter account that gives you a lot of information. So we could go and visit the Twitter account. So you'll see this, if you want to take it down and copy it during your clinic, you can give it to your patient. It's very easy, one piece of paper. And with that, I thank you very much, and welcome to the question later. Cheers.
Thank you, Dr. Kevin. Very interesting lecture. In India, most people ask the first question is what to eat. Now, I would request Professor Ghoshal. Sir is head of Department of Gastroenterology, and he is the pioneer in India looking at gut-brain axis disorder. Professor Ghoshal.
Thank you, Chairperson. I like to thank the organizing committee and ISG again for giving me this opportunity. I'll be talking about a new entity, so therefore it is very important that carefully listen to it. You might be seeing this patient in your clinic every day. Now we all know that acute gastroenteritis, that is infective diarrhea, always don't recover forever. And this was shown long ago by Truelove. So in ulcerative colitis, he used Truelove criteria. So Truelove was a British gentleman, and he was very observant. He found that patients who develop acute gastroenteritis, a proportion of them actually continue to develop chronic GI symptoms like IBS. And this is a meta-analysis by Madhusudan Grover from Mayo Clinic, Rochester, who showed that 10% of patients who develop gastroenteritis due to a GI infection actually develop post-infection irritable bowel syndrome. Now let me tell you, in Rome 4 we have changed the name. We have said, just stop calling them this as functional gastrointestinal disorder. These are not functional gastrointestinal disorder. Functional gastrointestinal disorder is now an obsolete entity. We gave a new name to this. This is called disorder of gut-brain interaction. Here I have written brain-gut interaction, which is the old terminology, but we have further changed it to gut-brain interaction. Why? We wanted to stress much of their problem is in gut and not in the brain. So therefore, what I have written here is brain-gut interaction. You just reverse this. This is gut-brain interaction. This is what we call this in the room. Now this COVID and IBS actually reiterates that further. That actually we have in these patients much problem in the gut and less in the brain. Because we know that coronavirus not only affect the respiratory tract but also GI tract, because GI tract also has angiotensin converting enzyme 2 receptor. Now coronavirus is known to cause a lot of things. It increases gut permeability. It causes ulceration in gut mucosa. People have shown that mucosal serotonin is increased in COVID patients. There is also inflammation in the gut as evidenced by increase in the fecal calprotectin. We know there is immune activation. So even there is an hypervigilance, there is panic. So therefore, enteric nervous system involvement. Based on these, we put forward a hypothesis. And this hypothesis we put forward long ago. In 2020 early part, we put forward this hypothesis, which was published once in American Journal of Gastroenterology and once in Gut and Liver, which is a Korean journal. What I want to convey from this to you is India should be signed. You know why? You can put forward hypothesis and even prove that in India. Today nation is talking about make in India. I request you to make knowledge in India, research in India, and I'll show you this has happened. Now we wanted to prove this, that does coronavirus disease 19 lead to IBS? So what we did is we launched a study. I had a limitation in SGPGI because we did not have access. I did not have access to the COVID data. No problem. If you have a problem, there are solutions. So I asked 3 medical colleges in Bangladesh to contribute to data. One was Mugda Medical College, one was Sheikh Russell Gastro-Liver Foundation, and Dhaka Medical College. So these 3 medical colleges contributed to a lot of data. Also, Dr. Ujjwala is sitting here. So she was backbone for this project of mine. Whenever she diagnosed a patient of COVID-19, I got the information with help of one of our senior resident, Dr. Akash Mathur, we called all these patients and followed them up telephonically. So what we did is we took 280 patients, followed them up telephonically 1 month, 3 months, and 6 months, and we had healthy control data from our previous study, and we compared this data. What did we find? What we found is at 6 months, a proportion, about 10%, develop IBS or functional dyspepsia or its overlap. So what we did is, this is what the Kaplan-Meier curve are, and you can see there is a higher proportion of FD, there is a higher proportion of IBS, there is a higher proportion of FD-IBS overlap. And this is the first study in the world proving that hypothesis, what we did in the early part of 2021, that COVID can lead to post-infection FGIDs. We also looked at the predictive factor. What are the factors which predicts that whether a fellow will have PI, FGID or PI DGBI following COVID-19? What we found is, if the patient is asymptomatic, he has a 0% chance of developing post-COVID-19 DGBI. GI symptom particularly predisposes them to more. If somebody has anosmia and ageusia, he has more chance of developing post-COVID-19 IBS. Presence of psychological comorbidity, yes. And if somebody continued to have GI symptom, even after 1 month after recovery from COVID-19, he is the one who is more likely to develop. You'll see how do you explain this. Actually, look at anosmia. Anosmia during the early COVID illness means that fellow, his olfactory nerve is involved. And we all know olfactory nerve is the direct extension of the brain. So olfactory nerve involvement means brain got involved by the virus. Similarly, we know that the taste sensation is carried by facial nerve in a small extent, but largely by the glossopharyngeal nerve, because we know facial nerve is a motor nerve, but it has a sensory component, and this one is taste sensation. And the glossopharyngeal nerve is a mixed nerve, it carries the taste sensation. So that means if somebody has taste loss, this glossopharyngeal facial got involved. If somebody has smell loss, the fellow's olfactory nerve is involved. That means the fellow's brain is involved. And we all know from the brain comes the enteric nervous system, parasympathetic and sympathetic, and enteric nervous system controls GI motility. So this is the reason why it got involved, and our study reiterates that these are disorder of gut-brain interaction and not functional gastrointestinal disorder. We also looked at psychological factors, and we found those at 1 month, those who had more, this we use DASH questionnaire. So DASH questionnaire, those who had anxiety and depression at 1 month after COVID-19 are the ones who developed this disorder more often. Now subsequently, this study came from Vineet Ahuja's group. It is also a very important study. So look at this, the third study in the world on post-COVID-19 DGBI was again from India. So hypothesis from India, first study in the world from India, and the third study in the world is from India. They took a lot of patients. In fact, they took 320 patients with COVID-19. They took 2 groups of control. One group of control, 320 patients. Those were healthy control, but they also took another 280 seronegative control. So they became very particular that there is no asymptomatic COVID-19. And what happened is their result was very similar to ours. That means about 10% following COVID-19 developed post-COVID DGBI. And this is the Rome survey. And much of the Rome data came from multicentric survey. Obviously, India and Bangladesh were part of it. I represented this from India. And this is published just a week ago in the Gut. And we found similar results. So that means about 10 to 15% patients following COVID would develop post-COVID-19 DGBI. This particular study also used a predictive model, which I'll not go to because of shortage of time. This is the second study on post-COVID-19 DGBI. It came from US, and there is an author from Mexico, Max Mulson. And what they found was very, very interesting. They found 66% patient who develop COVID-19 develop post-COVID FGID. Can't believe. But look at this study design has a problem. In the phase 1, they took 1,114 patients of COVID-19 to look at their symptoms, that is GI symptoms. And then in the phase 2, they emailed questionnaire to all of them. So this was an internet survey, and only 164 responded to their questionnaire. What does it mean? There is a recruitment bias. Those who had persistent GI symptom are the one who responded to their questionnaire, and that is the reason why they got a figure of 66%. So the data of 10% post-COVID DGBI is very similar to a development of post-viral enteritis IBS, and this is also 10%. So 3 questions. Can prolonged excretion of the virus, that is SARS-CoV-2 virus, explain development of post-COVID DGBI? Can gut microbiota dysbiosis explain? And third is, can continuing GI inflammation explain? This is our data. What we did is, Dr. Ujjwala is sitting here, she did much of the work. What happened is, we took patients with COVID-19, she did their nasopharyngeal RNA initially at baseline between 0 to 2 weeks, subsequently at 2 to 4 weeks, then 4 to 8 weeks, then 8 to 12 weeks. And we wanted to look at, is there a prolonged viral excretion among these patients, that is SARS-CoV-2 virus? And what we found is no, the prolonged virus excretion is not there. Can you see here, 8 to 12, none had fecal virus, though about 50% had fecal virus in the early part of illness. So therefore, prolonged viral excretion cannot explain. Can gut microbiota dysbiosis explain? This is the data from Si Wenji from Hong Kong, where she found that gut microbiota dysbiosis does occur in COVID-19 patients, and it can last up to 1 year. So therefore, this may be one of the pathology. The third study is one of the MD student thesis under Dr. Ujjwala. So what she did is COVID-19 patient 30 and healthy control 30, and they looked at fecal calprotectin at 1 week, 2 week, 3 week, 4 week, 5 week, and the serum marker of inflammation such as interferon alpha, serum IL-4, IL-6, IL-8, and IL-10. We all know IL-10 is an anti-inflammatory cytokine. IL-6, 8, and 4 are pro-inflammatory cytokine, and they found yes, IL-6 and IL-8 remained high in the serum about 5 weeks after COVID-19, so it may be one of the explanations. So how do you treat this patient? Because you get this patient in your OPD if you carefully look at them. Treatment of IBS, we all know, depend on the subtype. Constipation IBS has different treatment than diarrhea. And here, let me show you data from 2 studies. One study, our group, other study, Mexican and American group. And what it showed is most patients has diarrhea predominant IBS. So how do you treat diarrhea predominant IBS? This is the treatment of all diarrhea predominant IBS. But let me tell you, there is no randomized controlled trial on post-COVID-19 pharmacotherapy. If any postgraduate is here, please take this as a note. And if you do it, he will be the first in the world. But let me tell you one thing. People have shown that there is increase in the mucosal serotonin in the gut mucosa in patients with COVID-19. So what I do is I treat this patient with either remocetron, which is a serotonin blocker, or ondansetron, which is a serotonin blocker. And from the clinical experience, 100% of my patients responded. So till we get more scientific data, you should treat with serotonin blocker, ondansetron and remocetron. This is a Chinese study where they use probiotic. It is an open label pilot study, and they found good result. This may be because of gut microbiota dysbiosis. So now, as our first speaker, Dr. Srinivas, gave you a message, here is the message. Today in your clinic, you get a patient of IBS or FGID, ask the patient, did you have COVID-19? If the patient says yes, ask him or her, did you have your GI symptom before having COVID-19? If the patient says no, then ask, If not, did this start only after suffering from COVID-19? If the patient says yes, you are dealing with a patient of post-COVID-19 DGBI and treat them accordingly. I acknowledge all the supporting people like Dhaka Medical College, Mugda Medical College, Sheikh Russell Gastro Liver Foundation, DBT for giving me the research grant, Dr. Ujjwala, Dr. Giovanni Barbara from Bologna, Italy, Dr. Masood from Bangladesh, Dr. Max Mulson from Mexico, and students such as Radha Chauhan, Ankita Pandey, Shruti Vasan, and Akash Mathur. Thank you so much.
Thank you, Dr. Ghoshal. That was excellent as always. I think we've had 3 very good talks, and now we have time for discussion. So the house is open for any questions from the audience. I think to set the ball rolling, I'll start. Dr. Ghoshal, in your trial, you took controls. Were they historical controls or?
So very important question that you asked, because we realized that if we take asymptomatic control at that time during the pandemic, how many is COVID asymptomatic? We don't know. 5% of the COVID patients are asymptomatic. So this is a historical control of another study of ours, which was published in American Journal of Gastroenterology at around 2017. At that time, COVID did not come. So these were historical controls.
And, according to Rome 4, if you have to diagnose functional dyspepsia, you have to exclude after endoscopy any structural disease. So in your patients, did you do endoscopy in these patients?
That's why initially we wanted to call this patient as functional dyspepsia. Reviewer objected. So therefore we called them as uninvestigated dyspepsia. According to Rome 4, you are supposed to look for Helicobacter pylori present, eradicate, then only FD can be diagnosed. So we said uninvestigated dyspepsia.
I would like to ask Dr. Srinivas, do you do any psychotherapy in your clinic, or you straight away send to the psychologist? Because I think the Indian mindset is they come to a doctor and you straight them send to a psychologist or a psychiatrist. Then the problem is they feel the doctor is thinking that they have a psychiatric disorder.
Very, very important question. See, on an average, every week I will have at least 2 patients who come into my room, spend some time. Initially, they are very, very cagey. They bring some relative with them. We start talking. They want to please me. They want to tell me, doctor, I have IBS. Or they will tell me, doctor, I have GERD. Something they want to tell me. Then I tell them, look, I don't want diagnosis. If you know the diagnosis, you could treat it yourself. So please tell me, what is your complaint? That's how we start. And then as the conversation goes, invariably at least 2 people in a week will end up crying in front of me, and that's because either things are not going well at work. Mostly it will be an IT professional. He's not having a work-life balance, or he's upset that nobody has done a root cause analysis for his GI problem. So they cry, or it is something going on in the family, some domestic issue. And once they cry, they start feeling better. And then that is the time when I kind of tell them that it is important to have some help. And not only just these 2 people, there are many other patients who are already thinking in terms of a psychiatrist, but they just live in denial. They don't want to believe that there may be something psychologically wrong with them. So therefore they feel, oh well, I'm getting some stomach pain, I'm getting some loose motion, why not go to a gastroenterologist? He'll be able to solve the problem for me. So when they hear it like from the horse's mouth, that it is what they think it is, but not what they want to believe it is, they're more willing to go and see the psychiatrist. And I also tell them, please don't feel that I'm shutting you away from this consultation and my clinic. You can always come back. It's not shut on you. It's not like it's one size fits all. It's not like a all or none phenomenon. Either you have GA or you don't have GA. It's not like that. All I'm saying is go and meet someone else, talk to them. If you have a problem, by all means come back.
I think there's one question from the audience.
Yeah, we had a very good guideline from Dr. Kevin about how we could credential our own dietitian That means this dietitian is good for a GI consultation or a co-consultation. So can we have something similar for a physiotherapist? Because most physiotherapists are interested in cardiology and neurology and stuff like that. And we don't even know how to tell them what we expect. So is there any guideline from, there would be a basic thing which we can ask from them? We do our own rectal manometries, but there should be something that we can look back to as to what would be credentialing for a GI clinic.
I think what you said.
Yeah. This one. Yeah.
I think what you just said is very true. We share our physiotherapists with a colorectal surgeon, and very sadly, we recently just lost our nurse, and it is very difficult to have people who are interested in doing irritable bowel syndrome physiotherapy work. Mainly because similarly to dietitians and to psychologists, because they have a lot of underlying issues and they don't normally want to come. But yes, normally you have to work with your colorectal surgeon to come up with what we call basically a good biofeedback plan for your hospital. But it's very difficult to find.
Thank you.
Dr. Ghoshal, if I may ask, you have given us various hypotheses of why post-COVID people develop a GI disorder, serotonin levels and all those things. But do you think that it's a lot of it is about simple anxiety which has increased in these patients after COVID, especially in?
Yeah, quite likely. See, what we did is we did analysis DAS at 1 month, then 3 months and 6 months. And we found those who had anxiety and depression are the one who more likely develop post-COVID FGID, not only in univariate analysis, but also multivariate analysis. And if you look at the earlier studies, for example, Cochrane published in Lancet long ago, in maybe 90s, which was a part of his PhD thesis in Sheffield, he found following acute gastroenteritis, post-infection IBS is predicted by anxiety and depression. So what you are saying is quite likely, but we all know that all these DGBIs are multifactorial, multiple factor operate. So therefore, you cannot also ignore the gut pathology such as inflammation, microbiota dysbiosis, autonomic imbalance, and all those.
Thank you, Dr. Ghoshal, Dr. Kevin, and Dr. Srinivas. Now we'll close the session. Thank you all.
Thank you, sir. For the next session on gastroesophageal reflux disease, may I please invite Dr. Shobna Bhatia, Dr. Rajesh Upadhyay, and Dr. Ajay Jain to please chair this session.
Welcome to the session on gastroesophageal reflux disease. Our first topic is going to be refractory GERD, nocturnal breakthrough and its pharmacotherapy. And I would like to invite Shajan Peter, who's now at Alabama Birmingham, previously was at CMC Vellore. Shajan? Is he here? Hi. So can I request to start talking about GERD and the role of nocturnal acid breakthroughs?
Thank you, Shobnaji. Thank you, Sir Meena, for sharing my session. Thank you for the organizing committee, Dr. Mahesh, Govind, for inviting me here. It's really good to be home here and to see all of you. My topic is on the role of GERD and nocturnal acid breakthrough and pharmacotherapy. And it sort of reminds me of, I took my DM exams more than 20 years back. And on the last session was the oral session, and they asked me this question. Okay, the last question to you is, what are the physiological changes that happen during the night? And guess what? It was just a thought block, and the only physiology that happened in me was just sweating. So I hope I can tackle this topic today in a different manner. Here are my disclosures. What are my objectives? One is to discuss the approach of patients to refractory GERD, that kind of phenotype with special emphasis on nocturnal symptoms, how and when to pursue reflux testing, what is the data behind the treatment of nocturnal refractory GERD. Let's start off with this case, a 42-year-old woman with a 6-year history of regurgitation after meals. She belches frequently, globus, minimal heartburn with nocturnal symptoms and difficulty in going to sleep. She's 50% better with PPI. Does she have refractory symptoms? Does she have refractory GERD symptoms? Does she have refractory GERD symptoms, nocturnal breakthrough, what's called the NAB, or an objective evidence of GERD? So let's revisit. How do you approach this with refractory GERD? Do we need to go back and look at the initial symptoms and presentations? What is a typical presentation? Is it heartburn, regurgitation, or atypical symptoms like a non-cardiac chest pain or sleep disturbances? Has GERD been proven or not with an endoscopic testing or pH testing? Now look at this. This is a whole spectrum of GERD syndromes. You start off with NERD and you go down to laryngitis, chest pain, Barrett's, this whole spectrum. And all these is affected by acid reflux, bile reflux, visceral hypersensitivity, motor function disorders like hiatal hernia or non-esophageal factors. The classic definition for non-nocturnal acid breakthrough was presence of intragastric pH less than 4, intragastric pH less than 4, during an overnight period for at least 60 minutes in patients taking PPI. And that's an old definition, but now patients are coming with what's called a nocturnal reflux and symptoms. What are these nocturnal symptoms? Even a survey of many patients in the US, almost 80% of patients complained of nocturnal symptoms like heartburn, fragmented sleep pattern, and external manifestations like cough. Now, this could be related to physiological changes, increased gastric acid during the nighttime, decreased salivary secretions, there's an esophageal UES, which sphincter closes, that gets disturbed during sleep. And also, there are these primary and secondary facts, peristalsis are decreased to, therefore the clearance is less. And all this relates to a more severe form of reflux, which is related to bad esophagitis or Barrett's esophagus or strictures. So therefore, nocturnal symptoms are important to realize. So the important thing is to objectively evidence GERD, and that's a new kind of thing that we have in the new guidelines, and that is basically by doing an ambulatory pH monitoring on PPI. And the classic definition is more than 2 days of acid exposure time for more than 6%. And then you keep going down to decide whether this is reflux related or not reflux related. Again, these algorithms again go in the same model of proving reflux using a pH manometry. A manometry obviously is selective based on what the symptomatology is, but at least a wireless pH monitoring or a 24-hour pH monitoring is needed, or a repeat endoscopy. You have the methods which are the 24-hour pH impedance testing or the Bravo testing. The Bravo is a much more longer period. You can use it for at least more than 48 hours. The other one is for 24 hours only. And in a pH monitoring, you can see that these are acid refluxes that you see. And interestingly, you can also see the period during the sleeping pattern also. And that shows you how the sleep pattern is affected by acid disorders. And you know that nocturnal acid is an isolated phenomenon with little correlation to acid exposure time or nocturnal symptoms. So keep in mind that this is not a very common effect when you look on pH monitoring. The impedance also helps you because it also identifies weakly acid, like for example, a pH more than 4 to 7. In addition, it tells you what kind of thing it is. Is it acidic? Is it mixed, gaseous, or solid refluxate? Now, in this study, 172 patients were monitored on PPI. Those who had typical GERD symptoms, they had a non-acid kind of testing on 48% versus in atypical symptoms. Again, there's no reflux in 78% documented, and only acid only in 3%. So you see how pH kind of differentiates between whether this patient really have acid reflux or not. And these are patients on PPI. And bringing to this Lyon classification or consensus, I bring you to this point that they use an adjunctive evidence of a thing scoring called the mean nocturnal baseline impedance, which a lower the mean nocturnal baseline impedance correlates to increased acid exposure time and reflux. So coming to now to therapies of reflux, and this is how we're going to look at how to approach it. The lifestyle modifications, weight loss, avoiding eating close to bedtime, sleep with head and elevated, PPI optimization, adjunctive medical therapy, which we can talk about, and invasive GERD management like surgeries. PPI is the cornerstone of any therapy, and therefore you know that PPI needs to be optimized. In this good study that they show, they asked patients how do they take the PPI, and you see almost more than 50% of them didn't take PPI rightly on time. You take it half an hour before meals, you see that people who take it more than 60 minutes after meal, after meals, at bedtime, and PRN. And therefore importance of optimization. And PPI therapy, let's look at these studies. And this study says a 40 milligram versus 20 milligram of esomeprazole, like 53% versus 50%. If you look at NERD and nocturnal relief with DEXA, lansoprazole, 60 milligram is 80% versus 30 milligram is 76%. So there is some benefit of doubling the dose. With a night dose of rabeprazole, there was a lower pH than with the breakfast dose. So importance of knowing these PPI therapies. Now H2 blockers play an important role, and this is an old theory that you can add H2 blockers for nocturnal breakthrough. And ranitidine helps, and obviously we don't use ranitidine now, it's famotidine, which is now used. And it can prevent nocturnal residual acid secretion. But there are two randomized trials lately which show that there's no difference at 28 days between BID PPI and BID PPI and H2 blockers. And this is related to its tachyphylaxis or tolerance. And therefore, a question of how to give a holiday period or a washout period is something that we need to think about when putting them on H2 blockers. Alginates with PPI, this is a good study again which shows that with PPI and placebo, there was a significant change in the reflux or heartburn score related to adding alginates with placebo. Lesser nights with symptoms and alginate score, and it acts like a mechanical barrier to the acid pocket, which is postprandial. The other options you think about, baclofen, this is a muscle relaxant, decreases the transient relaxation of LES. There are side effects, but there's no difference for nocturnal reflux. Prokinetics are not well studied, but if you think of a patient with gastroparetic, there's no harm trying that out. An anxiolytic like buspirone, 20 milligram a day, helps in systemic sclerosis patients which have a lax LES. And in this patient with 30 patients, it increased the LES pressure from 7.7 to 12 mercury, and the heart scores also decrease. Hypnotics, they've tried this one, which is highly selective melatonin receptor antagonist, and 16 patients RCT, just 16 patients decreased nocturnal symptoms. A GABA agonist like the zolpidem in a randomized controlled trial actually increased the mean acids clearance. It actually worsened the reflux symptoms, especially in the night. Pain modulation. Is there any effects with giving pain modulation? These RCTs, typically, it's useful to start them on an antidepressant like a tricyclics. And if you look at these on the red box, significantly imipramine, amitriptyline seem to have significant effects. Again, fluoxetine seem to be significant. So they have some effect, but the data on nocturnal reflux is still low or lacking. Now we come to the other one, which is these potassium competitive acid blockers. And this is a new kid on the block. And they're acid stable, they're active drugs. You don't need to dime dose them around meals like the other PPIs, and they're typically not metabolized by the cytochrome cycle in the liver. They appear to have a nocturnal acid suppression, and their levels immediately peak, not like they are sustained, unlike PPI, which have the cumulative action of increasing their dosage or their action. They are not FDA approved in the US right now for GERD, though they are approved for triple therapy for H. pylori, and there are no studies yet for GERD. I saw this recent study on this drug called tegoprazan, and this is another novel potassium competitive acid blocker. And this showed that if you used it, it increased the pH at night 66% as compared to vonoprazan, which is another drug, to 60%. And with omeprazole, it was only 36%. So clearly, it seems to be a benefit there. However, we don't have any correlation to acid exposure time. This is all done on intragastric pH and those values. So more data is awaited for this class of drugs, and clearly this is going to be a new kid. I think would may be a good game changer in the long term. Let me just track down to other modifications, touch base briefly on those. Importantly, lifestyle modifications like avoiding fatty meals, all the lifestyle, like smaller meals, don't lose weight. Obesity is an important player. So keep these things in mind, especially when you're talking about just not only pharmacotherapy, but important to know that lifestyle modifications are important. And this is a very good study from the Danish group, which showed that if you lie down on the left side, it's better than on the right side. And this is primarily because of the angle of His and its angulation and LES compression. And so you see the acid exposure, acid clearance time was lesser as compared to lying on the right side. And also the acid exposure time was much less on the left side as compared to the right side. So that's a good study which tells you a simple thing in addition to. Let me not touch on this because this is beyond my scope of discussion, but these are new reflux procedures that which are endoscopic and surgical available now. And but I bring you to this slide, which is a good systematic review on clinical effective interventions for the treatment of nocturnal GERD. And if you see this slide, it shows, yes, acid suppression therapy, the lifestyle modifications, again, very important. Elevation of head, for example, prolonged dinner to bedtime, left lateral position. There's less evidence for CPAP or nocturnal CPAP. H2 receptor antagonists, again, are 50-50, as I mentioned. Hypnotics, baclofen play no role, pretty much are not recommended. And as you tailor your symptoms, important to know that this is an altered physiology, and there is where PPIs are more effective. When it's an altered perception, then as the spectrum goes down to NERD, to reflux hypersensitivity and functional heartburn, PPIs are less effective, and you should start thinking of alternate end measures of treatment. And I'll come to this last slide, which is an important slide, which shows you how the treatment approaches. And if you start from each of the spectrum of erosive esophagitis to NERD, reflux, heartburn, extravascular esophageal syndromes, and you see the spectrum coming down from PPIs to medications to lifestyle behavior modification. Each one has its role in this important spectrum. So defining it is very important. So what is my take-home messages? One is important to know that there's something called nocturnal reflux, and that can be coexistent refractory GERD. Important to differentiate it from other syndromes. Emphasis on diagnostic testing, which is a new guideline. And you need to control both for other symptoms, especially if there's reflux hypersensitivity. Sending for surgery is a role, but remember that not all patients are going to benefit with surgery. It's not a guarantee. And let me stop with this. And my special thanks to my friend Jim Calloway. He's my main esophageal guy at our center. And you're welcome to, if you're there, please holler out. Welcome to see you all. Thank you.
Thanks a lot. That was an excellent talk. We'll take any questions at the end of this session. So could we invite you to come and take a seat here in the meantime? And we'll proceed on with the next talk, which is about current indications for probiotics. And I would invite the speaker, Dr. Umesh Goyal, to come and deliver his talk. Dr. Goyal is a professor of gastroenterology at Dayanand Medical College in Ludhiana. Dr. Goyal, please.
Good evening, sir. Thanks for the invitation. So prokinetics, the talk is on prokinetics. So they are undoubtedly the second most commonly overprescribed drugs in gastroenterology, obviously after PPIs, and you all must agree with this. The reasons of this, because they are available over the counter in our country. They are available as fixed dose combinations. So when a person needs PPI, the chemist may hand over a combination of PPI and the prokinetics, mainly because the lack of awareness of the efficacy of the prokinetic, and also the lack of awareness of the adverse effects of the prokinetics, that they are being used so widely by the physicians and by the patients themselves and by the all over the counter people. So today my task will be to dig in the literature. I'll discuss the GI motility regulation, how does it act, and the prokinetics, how do we classify them, and I'll talk on their adverse events profiles, which we had studied in the basic pharmacology, because we need to know that before using the drugs. Then I'll dig on the literature of the evidence of the prokinetics in GI disorders, and then I will summarize the topic. So as we all know, the regulators of GI motility include enteric nervous system, the intestinal cells of Cajal, the smooth muscle cells, the mucosal neuroendocrine cells, and the autonomic nervous system. And all these systems act through various hormones like serotonin, dopamine, acetylcholine, motilin, cholecystokinin, and catecholamines. So this diagram sums up the whole neurotransmitter going on in the GI tract. So if you see down there is the enterocyte cell, which will help to movement of the GI tract, and we see the acetylcholine when it acts here, it leads to the movement. So what affects the acetylcholine when will get released? It is affected by various factors like dopamine has a negative feedback on this. We have 5-HT4 receptor here, serotonin acting on it leads to acetylcholine release, and release here leads to peristalsis. And when we have acetylcholine esterase, and it based on acetylcholine, but when this is blocked, the acetylcholine will increase. And then we have the D2 receptors lying here, and when we have something acting on the D2 receptors, then we'll discuss these, how the drugs act at various parts. And so this is all regarding the motility. If we see the vomiting part, we have two areas in the brain which control the vomiting part also, and the 5-HT3 receptors mainly act there. So prokinetic literally means to promote movement, and in gastroenterology the prokinetics are a class of drugs that promote GI motility and increase the transit by increasing the frequency or the amplitude of contractions without disrupting the normal rhythm. This is very important. They increase the normal rhythm by their amplitude or their frequency. And the various mechanism, as I showed the slide before, here we can see the various mechanisms in detail, how the drugs act. So we have D2 receptor antagonists like domperidone, metoclopramide, levosulfide. They antagonize the D2 receptors, thus increasing the release of acetylcholine and leading to increased peristalsis. The other class of drugs are 5-HT4 agonist drugs like cisapride, tegaserod, mozapride, and the latest one is prucalopride. They are agonists of the 5-HT4 receptors, which lead to increased acetylcholine release and which further leads to increased motility. The other class of drugs are bethanechol, pyridostigmine. They are not very good pro-motility drugs, but mentioned here because they act by the same pathway. They directly lead to the increase in acetylcholine. Then we have new drugs like atošiamide, which has a dual mechanism of action. It acts on the muscarinic receptors, and also it inhibits the acetylcholine esterase activity, so that increased acetylcholine occurs, and further which leads to increased GI motility. So all these drugs will act at different pathways, but ultimately lead to increased acetylcholine, which will lead to increased GI motility in a coordinated fashion. And the adverse effect profile differs because of the receptors on which they act, because these receptors are present at other areas of the body also, especially in the brain, in the cardiac centers. So that is why the ADR profile of all these are different. So this is how we classify. Just to repeat the name of the drugs, which they all come in this class, are metoclopramide, the first drug to be discovered and then came domperidone, etopride, cinetapride, and the latest one is levosulfide. The second class which we talked was 5-HT4 agonist drugs. We have a whole list of drugs in this. Cisapride, which has been banned. I'll come to that later on. Mozapride, tegaserod again, which has been withdrawn. Then the latest newer drug in the market is prucalopride, and the lower 4 drugs are still in phase 2 to 4 clinical trials, and they possibly have fewer side effects, and they will be better tolerated in the future. Just to mention here, cisapride was a true pan-GI prokinetic drug. It acted on the esophagus, stomach, small intestine, and also on the colon. That is why it had a good action in constipation. Tegaserod mainly acted on colon. That is why it was promoted for IBS-C and chronic constipation, but it had cardiac side effects. It was withdrawn. Prucalopride, again, because it's specific for 5-HT4 receptors, it acts mainly in the small gut and the large gut. It's also used for constipation. Mozapride, on the other hand, it acts mainly on the small gut and the stomach, so it's used for dyspepsia and gastroparesis. So they are slightly the effect is due to the different receptors on which they act. The third class is motilin receptor agonist, which we know are present in the stomach. Erythromycin acts on this as a very good drug for gastroparesis, but because of being an antibiotic, its use is limited. Other class of drugs is renin receptor agonists. These also act in the stomach, and they are good prokinetic drugs. Both are in the trial phase. Other class is 5-HT1 agonist drugs, buspirone, but it has side effects and inconsistent effects on the motility, so it has not been in clinical use. Other class are cholinergics or cholinergic drugs, in which the directly acting drugs are bethanechol, which directly acts on the acetylcholine receptors, but again the effects are inconsistent, not in use. Then indirectly acting are acetylcholine esterase inhibitors. The previous ones which we know of are neostigmine and pyridostigmine. They are mainly used in Ogilvy syndrome and chronic pseudo-obstruction. We all know that. The latest drug in this class is acotamide, which has a dual mode of action and it has good action on the gastric accommodation, which is being used in upper GI disorders. Other drugs like ranitidine also have gastric pro-motility action, but we cannot promote these for GI motility. Other drugs which are not in clinical use, but yes, they come under prokinetics, just a list I have mentioned here. They act mainly on the colonic mucosa, or they don't have any consistent effects, so they are not in clinical use. Now, after the classification, as I told you, ADRs are the most important thing in this discussion. So, starting from the oldest drug, metoclopramide, which we know we all must have faced dystonias in patients whom we gave metoclopramide. So, these are CNS effects occurring up to 30% patients, because it crosses the blood-brain barrier and leads to so many of CNS effects, starting from anxiety, confusion, hallucinations, EPS symptoms, NMS, dystonias, and with prolonged use for more than 12 weeks, we have Parkinson-like symptoms which may be reversible, but tardive dyskinesias are not reversible. GI side effects, diarrhea can happen. Another fearful side effect is hyperprolactinemia, which can cause gynecomastia, impotence, and male infertility. Cardiac side effects like QT prolongation also occur with metoclopramide. And so because of all these things, especially because of tardive dyskinesias, US FDA gave a black box warning that cumulative dose more than 12 weeks leads to tardive dyskinesias, so they do not advise it using more than 12 weeks. But still, it is the only prokinetic available in US. See this. So we are so lucky in India that we have the newer drugs coming soon, lucky or unlucky, I don't know, because the misuse can also happen when you get it so easily. But in US, this is the only prokinetic available till date. Domperidone, it had fewer side effects than metoclopramide, but because it doesn't cross the blood-brain barrier, but still it has some CNS effects like dizziness, headaches, and it has GI side effects. It increases prolactin, causing all those symptoms, and also it has cardiac side effects. And because of prolonged use and higher dosing of use, in US it was being used to promote milk secretion in females. So it led to cardiac side effects. It was withdrawn from the US market in 2004 because of increasing dose causing cardiac arrhythmias in women. Then a European agency said to restrict its use to when it's urgently required. Only then they can be used. But we are using it day and night in India. We don't have so many side effects because the dose till 30 mg it is safe. The side effects occur at more than 60 mg. So people were using it at more than 60 mg. So mind you, if we use a DSR combination which is 30 mg, we use twice a day, we are heading towards side effects. We should never use those combinations for twice a day. They are meant for once a day use. Levosulfuride, the newer drug in this, has all the side effects but lesser severity than the previous two drugs. It has 5 to 10% rate of CNS side effects. EPS do occur, but they are mostly reversible. It has hyperprolactinemia and 10 to 15%. We all must have faced patients coming with gynecomastia and erectile dysfunction after prolonged use of levosulfuride. QT prolongation is luckily rare, very rare with levosulfuride use. Etopride, again the newer drug in this block, has dual mode of action and has less side effects. So it is the beneficial out of all these drugs. Like it acts on the D2 receptors also, and it also inhibits the acetylcholine. So it has dual mode of action. It leads to good this motility, GI motility augmentation. So the only side effects noted are abdominal pain and diarrhea, and just headache in the CNS part. It is devoid of serious adverse CNS, cardiac, and prolactin increase adverse events. So it is a safer option out of all this class. Cinetapride, there are very less data available. So I just have one study showing that some fatigue and diarrhea occurring with this, and we need more data for the safety of this drug. Coming to the second class of drug, 5-HT4 agonist, I told you cisapride was a truly pan-prokinetic, pan-GA prokinetic drug, but it had serious side effects like headaches, like cardiac arrhythmias. Because of it, it was withdrawn from the market in 1995, and in 2000 it was withdrawn from clinical practice in most of the countries. Mozapride, the sister drug, is better, has lesser side effects, can be used. Tagaserod, as I told, acted mainly on colon. It was also withdrawn from the market in 2007 because of concerns of serious cardiac events, and also it had ischemic colitis in few case reports. Prucalopride, the new drug in the 5-HT4 category, is good, as I told, because it is a selective inhibitor and at 5-HT4 agonist, it has fewer side effects. Erythromycin can be used as a good gastric prokinetic, but it has less side effects, but less used because of antibiotics. Acoshamide, dual mode of action, better gastric accommodation, fewer side effects, is a very good drug. So now coming to the efficacy in GI disorders. So prokinetics mainly will help in disorders where the motility is the main problem, like in gastroparesis, constipation, colonic inertia, but it has some role in GERD and dyspepsia also. So in gastroparesis, we will see the literature. The ACG guidelines recommend the metoclopramide as a first-line drug for gastroparesis because it's the only prokinetic available there. Other studies show a good efficacy of erythromycin, domperidone, metoclopramide, and etopride. So we have the combination of and levosulfide. So all these drugs, anyone can be used wherever they are available. So these are the drugs. As I told, in India, we have all the available drugs. We can choose from these, or we can use prucalopride, which is not yet recommended for this condition, but can be used. And we have few drugs in the trial phase. For GERD, if you see the literature, initial literature showed that mozapride, that time only mozapride was available, that is 10 years back, was not useful in combination. Then studies came that they alone obviously were not useful, but when PPIs combined in prokinetics, yes, they led to some improvement in quality of life and symptom outcomes. Future systematic analysis and reviews also showed that combination of PPI and prokinetic is effective in refractory GERD patients, and adding them lead to some improvement in quality of life. And most of the guidelines, Japanese guidelines, also recommend adding this to the therapy. Again, a few more studies showed that adding acoshamide and other prokinetics are helpful for GERD patients. So current evidence suggests that combination of PPI and prokinetic can be used to increase the global symptom score in GERD patients and NERD patients. However, in erosive esophagitis, they have no role because it doesn't improve the mucosal healing. So this is to sum up the prokinetics for GERD available in India. We've discussed all that we can choose any one of these. And in the algorithm, just want to show that most of the algorithms show that GERD, that prokinetic should be used as a second part. So we have prokinetic lying here when we try the PPS for adequate duration. For functional dyspepsia, studies show that etopride, domperidone, mozapride, acoshamide, tegaserod, cisapride all are effective than placebo, and they can be added to this patients. And but we have differences of the data in East and West. So if you see the right side of the figure, Eastern data shows that people respond to prokinetics as a single agent in dyspepsia patients, and this may be due to various patient factors. And Japanese guidelines recommend these are the first-line treatment for functional dyspepsia. However, ACG guidelines, they do not recommend them as a first-line treatment because, first of all, more PKs are not available there, and also they recommend as a third-line therapy after tricyclic antidepressants and PPIs. So I will go with the list of drugs which are available. Again, for functional dyspepsia, all the drugs can be used, and I would suggest this algorithm by the Rome 4. If you have a patient of PDS, we can give prokinetic as a first agent. If you have a patient of EPS, we give anti-secretory as a first agent. If EPS patient is not improving, then we can try prokinetic as a second step in this dyspepsia patient. In geriatrics and pediatrics, this is a big challenge because no more studies are available in this class, but we can start prokinetics at a low dose if needed, and then slowly increase this. I'll come to the take-home messages. So current indications include gastroparesis, GERD, and functional dyspepsia. The choice of prokinetic depends on the available data on the efficacy, and some may be good in terms of efficacy, some in case of safety. Safety concerns mainly arise when they are using higher doses, longer duration, and in elderly. So monitoring of cardiac, CNS, and prolactin side effects are required. For gastroparesis, they are a first-line treatment. For GERD and NERD, they can be added to PPIs. For erosive esophagitis, no benefit. For functional dyspepsia, first-line treatment in PDS and second or third line in EPS patients. Options are available, and future research on this safety for long-term in Indian patients is required. We don't have much of Indian data here. In the end, I would like to share this that we published this book on prokinetics. Further information is available here. So under the mentorship of Professor Saraswat and Professor Bakharia, we were the authors and we wrote the manuscript, which includes all the available data on prokinetics till date. Thank you so much.
Thank you, Homesh. I'm happy you finished such an extensive piece of work in a short time. And now I have the pleasure of inviting Dr. Amit Datta and Dr. Manas Panigrahi. Hi, Manas. Amit Datta, is he there? It's coming. Okay. So, given the background, the Indian Society of Gastroenterology has given some guidelines on PPI stewardship, on rational use of PPIs, and also talking about when to stop the PPIs. So, can Dr. Amit Datta, can you come on and start? Dr. Amit Datta is from CLC Bangalore.
Thank you, Chairperson. So I'll be presenting on, about the discussions we had on the ISG guidelines on rational use of proton pump inhibitors. We are calling it PPI stewardship, like the same way we have antibiotic stewardship. So essentially, we know that PPIs are among the most commonly prescribed medications. They are available over the counter in our country, and therefore inappropriate use is sort of overuse. Inappropriate use is frequent, and this has two implications. One is obviously more cost, and also the potential of adverse effects, which more and more the list is increasing. And because PPIs are drugs not only confined to gastroenterology, they are used at different level of care across different specialities, and therefore there is a need to have a guideline talking about when we should use, and more importantly, how long should we use PPI. So with this objective, the ISC has undertaken this task of formulating guidelines on rational use of PPI. So in this short presentation, obviously I can't go through all the statements and the literature, but I'll touch upon the important aspects of this guideline. So these are the contributors, the chairpersons, then the draft committee member, and then we had members from different parts of the country. So we followed the modified Delphi process where we had the generation of statement followed by voting, and then we had an in-person meeting in Delhi where we had the final round of voting after discussion. This just shows the different events, different steps we had beginning from June last year up to October when we had the final meeting to get the final guideline. So when we presented the statements in the final meeting in Delhi, first the statement was presented, then the key literature for the statement was also discussed. The quality of evidence was done based on the grade methodology into high, moderate, low, and very low. Then we had a discussion on each of the statement, the literature which we had. This was followed by the voting on the agreement. And for a statement to be accepted, more than 80% of the members had to agree, either strongly agree or agree for the statement to be accepted. And then we also made recommendation, whether it's a strong recommendation or a conditional recommendation. Now for statements where the evidence is not applicable, those statements we call it as a good practice statement as witnessed by the grade group. So moving on to the recommendations per se, the first section dealt with the overuse of PPI and how to optimize overuse of PPI. So the statement was, inappropriate use of PPI is quite common in clinical practice, and there is a lot of evidence to show, as I'll show in subsequent slides. And therefore, the good practice statement which we generated was that PPI should be discontinued if there is no definite indication, and it was a strong recommendation by the group. So there are several studies which have looked at inappropriate use of PPI. This is just one set of them, and this includes study from India as well. And as you can see, that in majority of them, overuse or inappropriate use is more than 50%. This is just one set of studies, the second set of studies, and the third set. So huge number of studies showing so much of PPI actually used inappropriately. So we actually did a meta-analysis, we pulled the data together, and what we found is that almost 55% of the PPI which is used actually doesn't have a proper indication. Either indication is not correct or it is used for longer duration than it is required. So therefore, we need to be aware about this, and when somebody is getting a PPI, we should ask, does he really need a PPI, and how long does he need a PPI for? Then what about deprescribing? What about stopping the PPI therapy? So we have two options. One is you can abruptly stop, or second is you can taper the dose. As of now, there's not too much of literature available on which strategy is better. There's one RCT only which has looked at that and found it to be similar. So the group said that the strategy needs to be individualized and flexible. As of now, we can't recommend one over the other. Use what you're comfortable with and look at the patient scenario as well. So that was about the overuse and how to stop using PPI. The next set of statements discussed about the common indications, the GERD, dyspepsia, peptic ulcer disease. So if you look at GERD, most of the patients in OPD which we see are patients who are having reflux symptoms. They don't have alarm symptoms. So we don't do any endoscopy. So these are called the uninvestigated GERD. Even among patients who have endoscopy, majority of them endoscopy is normal. They are the NERD group. Among the esophagitis also, the grade A and B is more common than C and D. So they are the mild esophagitis. So therefore, the majority of GERD patients you see are uninvestigated GERD or NERD or mild esophagitis. So for these patients, a standard dose of PPI is sufficient for about 4 weeks, and don't go beyond 8 weeks based on the literature available to us. So about 4 weeks should be sufficient, and the level of evidence is high with a strong recommendation for this. So majority of GERD patients which we see in a clinic, we should give them a finite therapy for about 4 weeks, maximum up to 8 weeks. Now obviously when this is the literature again, I'll skip the literature because of lack of time. Now obviously when patients come to us with GERD symptoms, a proportion of them will have a recurrence of symptoms when you stop therapy. Now in them, we have two options. One is that you continue therapy, second is you give it on demand. That when the patient gets symptom, you give therapy, otherwise you don't give. So there are several trials now and a meta-analysis which has shown that on-demand therapy, that is giving it when the patient gets symptom, is as good as continuous therapy. And with on-demand therapy, you minimize the cost, you reduce the chance of side effects. So the group says we should recommend on-demand therapy in patients with NERD or mild GERD for recurrence of symptoms. Obviously, patients who have severe esophagitis or a peptic stricture, those patients who need a long-term continuous PPI. This again is a meta-analysis talking about which showed that on-demand is as good as a continuous therapy for NERD and mild erosive esophagitis or uninvestigated GERD. So that was about GERD, one of the frequent reasons for using PPI in practice. The next is dyspepsia. Now, in dyspepsia, again, the majority of patients we see are patients who don't have any alarm symptoms. So we don't do endoscopy. These are the uninvestigated dyspepsia. Among patients who have endoscopy also, majority of them, endoscopy is normal. These are the functional dyspepsia. So among patients with uninvestigated dyspepsia or functional dyspepsia, standard dose PPI is the recommended therapy but should be used for 2 to 4 weeks. So don't use for longer than 4 weeks in this group of patient. Again, a moderate level of evidence and a strong recommendation. Just the data from Cochrane meta-analysis backing this statement. For H. pylori, there's not much of a controversy. PPIs are an integral part of therapy. So standard dose PPI in twice daily dose is an essential component of drug therapy for the eradication of H. pylori. Peptic ulcer disease, again, PPIs have been the drug which have changed the landscape of management of peptic ulcer disease. So in somebody with peptic ulcer disease, if there is H. pylori, you treat H. pylori. After eradication, if somebody has a DU, you can give it for 2 more weeks of PPI. If it is a gastric ulcer, 4 more weeks of PPI can be given. So that was about the indications, the GERD, dyspepsia, H. pylori. Obviously, we have more in the statements when it comes in the print, but because of lack of time, we're discussing the important ones. The other situation where a lot of PPIs uses prophylaxis, to prevent the risk of ulcer, to prevent the risk of bleed, like when somebody is on NSAID, antiplatelet, anticoagulant, etc. Now, even in those, when we review the literature, we realize that everybody who is on antiplatelet or NSAID or steroid does not require PPI prophylaxis. So look at the setting, look at what are the additional risk factors the patient has, and then you prescribe the PPI. So for example, for antiplatelet, if somebody is on a dual antiplatelet, or if it is single antiplatelet plus some risk factors, then you should give prophylaxis, not single antiplatelet alone. Even for anticoagulants, they have to have some additional risk factors for them to get a prophylaxis. For NSAID, routine prophylaxis for PPI is not recommended for everybody on NSAID. Only if you do a risk assessment. And if there is a risk factor, I'll show in subsequent slide. If you have the risk factors, that's when you start using a PPI prophylaxis. Same for critically ill. Now, the stress ulcer prophylaxis is one of the frequent reasons for overuse or inappropriate use of PPI when we review the literature. So every patient who is in ICU, every patient who is sick does not need a prophylaxis with PPI. In critically ill patients, prophylaxis is useful if there are additional risk factors for GI bleeding in that patient. So for example, for NSAID, I was telling you, if somebody is on NSAID and if they've had a previous ulcer, whether it's complicated or uncomplicated, if they are on high dose NSAID, they're taking anticoagulants, if they have ages more than 70 years or they're on steroids, if these risk factors are present in addition to NSAID, then you should give prophylaxis in that group. Same for stress ulcer. Somebody who is critically ill, who is in ICU, who is sick, and he's on a ventilatory support, or there is coagulopathy, that platelets are low, INR is prolonged, or there's a prior history of ulceration or bleeding, if somebody has a brain injury or a spinal cord injury, or significant burn. These are the group of patients where you need prophylaxis, not everybody who is in ICU. So again, I'll just skip the literature due to lack of time. Now, another important statement we had is on the chronic liver disease. Now, more and more data is emerging that PPIs can have a harmful effect in patients with chronic liver disease, and we know PPIs are prescribed quite often in patients with CLD. So in patients with cirrhosis of liver, PPI should be used with caution. And there is data showing that there is a higher risk of SPP, higher risk of decompensation when you give PPI, especially for longer duration in patients with chronic liver disease. We also had statements on children just to make the guideline more complete. So for children, we said if in children over 1 year of age, for a short-term treatment of symptomatic GERD, we can use PPI. We can also use PPI for healing of erosive esophagitis, peptic ulcer disease, and H. pylori. But in those below 1 year of age, use of PPI discouraged, and that's because the data for efficacy in that group, so there the PPIs are mainly used for extraintestinal manifestations of GERD or some atypical symptoms of GERD. And there the correlation of symptom with GERD is not very good. So for less than 1 year of age, I think the PPI use should be discouraged. We also had statements on about PPI in pregnancy. So in the pregnant patients, if they are in the second and third trimester, you can use a standard dose of PPI if there is an indication. But in the first trimester, you should avoid using PPI until there is a strong indication. So there was this NEGM study, which is a large retrospective study, which showed that there was no added risk of birth defects when you give PPI. But then there are subsequently more further cohort studies, more case control studies, which show that there is a risk. And therefore, the group felt that we should err towards caution. And birth defects are a concern in the first trimester. So therefore, in the first trimester, we should probably discourage use of PPI and use it only when there is a very strong indication, no other option is available. Next, we talk about the co-prescriptions. PPIs are sometimes given with other drugs to reduce the side effect of the drug, for example, antibiotics, etc. So here again, we just had a talk on the prokinetic before this session, before my presentation. And what the group felt is there is a lot of prescription which contains a fixed dose of PPI with prokinetic. And actually, if we need prokinetic, we should think about them individually and add them as a separate prescription. We should not use a fixed dose combination, because then what happens, as long as you continue one drug, both drug keeps getting continued. So fixed dose combination of PPI and prokinetic is not recommended. Also, for patients who are antibiotics, there is no need to prescribe PPI routinely, except for like somebody who got H. pylori therapy. Their H. pylori PPI antibiotics are a part of therapy. There is no rationale for adding probiotics to PPI. So again, somebody who's on a sort of a PPI probiotics, do not add PPI to that therapy. SSRI. SSRI can have some impact on platelet function. There is an increased risk of GI bleed, but there is no data showing that PPI is reducing the risk of bleed. So therefore, even for SSRI, we should not use a PPI prophylaxis or a co-prescription. For bisphosphonate, again, which are known to cause ulcerations in the upper GI tract, even low GI as well. There also, there is no evidence showing efficacy of a benefit of PPI. So PPI should not be routinely used while prescribing bisphosphonate, should not be routinely used for patients who are on SSRI therapy. And just showing an example, so why we are talking about this. So, antibiotics, the risk of C. diff infection increases. And if you give this patient's PPI, then the risk becomes even more. And therefore, we should think of the side effect profile. And if there is not much of evidence showing a benefit, then we should not use the combination or a co-prescription. The final set of statements which we had dealt with the adverse effect. I mean, that's why one of the main concerns is why we should have such guideline, because obviously it's cost of therapy to the patients, but also the risk of adverse effects. So a lot of adverse effects have been sort of reported in the literature. So use of PPI is associated with an increased risk of certain GI infections like Salmonella, Campylobacter, C. difficile. We know that PPI already, there is a risk of renal injury in patients who are taking PPI. So use of PPI among the renal thing, acute interstitial nephritis is something where there is a very good sort of got the maximum evidence for. So use of PPI is associated with a small increase in risk of acute interstitial nephritis. About the risk of bone fracture, a lot of studies are talking about the bone fracture risk. But when we look at the literature, it's actually mainly in the very old age group patients. They have other risk factors as well for fracture. And it's difficult to pinpoint what role PPI alone is playing in that group. So therefore, for this, the statement we suggested was that there is uncertainty about the increased risk of bone fracture after use of PPI. Use of PPI is also associated with an increase in risk of hypomagnesemia. The long term of PPI, use of PPI is also associated with a small increase in risk of vitamin B12 and iron deficiency. So adverse effect wise, infections, acute interstitial nephritis, hypomagnesemia, and B12 and iron deficiency. This again the meta-analysis showing the infection. In fact, infection. If you look at the meta-analysis about infection, if you look at Campylobacter, the odds ratio is about 5, which is quite high. Same is true for Salmonella. Here also the odds ratio is about 4. So significant increase in risk of these infections when you take PPI. Same for C. difficile, although the absolute risk is lower than it is for Salmonella and Campylobacter. This is the meta-analysis showing risk of interstitial nephritis. B12, something that meta-analysis done by our own group led by Dr. Vishal Sharma, the guideline formulation group, and this is what we found that for with patients on PPI, there is again a risk of B12 deficiency. Although the absolute risk is not that high, it's about 1.4 with the CI of 1.16 to 1.73. So this is my final slide. So what I have shown is that when we looked at the literature, overuse of PPI is quite frequent. So almost 50% of PPI prescriptions do not have proper indication in terms of indication as well as the duration. So therefore, there is a need for PPI stewardship. And I think so when we are giving PPI, we should ask 2 questions. Does the patient really need PPI? And second, very important question is how long should the patient take? And we should advise the patient about duration. Otherwise, because drug is available in the counter, they will go buying themselves and they'll keep on taking it for long term. And therefore, there should be special emphasis on duration of therapy. Thank you for your attention.
Thank you very much. I think we've had a very important and a very practical session relating to gastroesophageal reflux disease, use of prokinetics and PPI, which is of everyday clinical practice importance. I'm sure there'll be a number of questions for the speakers. So if there are any questions, please go ahead.
So can I start the questions, Dr. Upadhyay? So my first question is to all the 3 speakers. Okay, we had Dr. Shajan Peter who talked to us about refractory GERD. In his entire slide set, I did not see the word prokinetic. Correct? Then we had Umesh telling us about GERD and
I could find a prokinetic there in the slide. I was looking for that.
And Amit Datta who told us that FDCs are not good enough. So Umesh, about the combination of prokinetics and PPI, do you think they should be given individual drugs or the fixed dose combinations?
Definitely, ma'am. First of all, again, as you emphasized, as I told in the slide also, it should be used only in refractory GERD, not as a first-line drug at all. So because of fixed dose availability only, they are being used over prescribed. So I think fixed dose combinations, if at all they have to be used, again it has to come down the line as a refractory GERD. But dosing individual is still far better because we don't know what would happen to the efficacy of drug, whether a drug given as a 75 mg of Leukosulfate suppose in the morning, whether it's acting truly over 24 hours or not. So I completely agree that they should be given as separate doses, and that too as a refractory GERD patient, not as a start alone therapy.
Shajan.
You're absolutely right, Shobhaji. You did spot that on. But I didn't mention that. I fully agree that prokinetics have a specific role. But when you look at the literature for nocturnal kind of reflux, or that we don't have data. That's to point out one. And then the second thing is that more pointed out that the prokinetics in US is not that freely available. So the studies are much lesser in the US compared to other countries. In my practice, for example, it's like a case by case basis, and I do a lot of Barrett's. I ablate them; they don't respond to therapy, and lo and behold, I see a lot of food in the stomach, and I can be assured that they have some form of gastroparesis. That's the kind of indication I would proactively start them on a prokinetic and see the benefit, because that's refractory GERD and Barrett's in that sequence.
So, what you suggest is that if patients have evidence of gastroparesis, that means if they have bloat, if they have early satiety, or if they have nausea, then you would add a prokinetic rather than straight off starting a prokinetic for GERD. Okay.
Can I add, ma'am? Or on a pH impedance testing, if we have a acid reflux, volume reflux coming, then prokinetics will help.
Yes.
So we
volume
need to
reflux has to be proven.
Yeah.
Correct.
Why so much of combination is being used in India is primarily because of the compliance. The patients love to have a single tablet and get away with it, but that is certainly not the way to practice. But the other point is why it is more commonly being used prokinetics is because we see in India 30 to 40% overlap of GERD with functional dyspepsia or bloating type of symptoms. So the use of the combination is more common.
Yeah, correct. But at the same time, yeah, we have some questions from the audience. Yes.
Yeah, Dr. Shiva Prasad. I have a couple of questions to Amit. Number one, I think you should have also mentioned about the use of parenteral PPIs, intravenous PPIs in practice. Your entire guidelines were only to
No, there is a statement, but this is because of lack of time we didn't present here. There is about IV PPI for the GI bleed, especially we have a section.
No. The overuse and misuse. You'll find that any patient reaching any nursing home or any hospital with any chest or any abdominal discomfort, the first drug he gets is an IV PPI. So this kind of highlighting of inappropriate use of IV PPIs, because it has serious implications. The patient gets diverted into thinking that he has a GI, some upper GI non-specific dyspeptic symptoms rather than what he really has.
No, that will be covered. That the first three, those studies I showed you, the 3 slides with all the literature about overuse. Some of them are IV overuse as well. So when we discuss in discussion, we'll put that.
The second point is about co-prescription of PPIs with dual antiplatelets. Earlier, it was felt that the risk would kind of justify because we still didn't have, compared to single platelet and versus dual platelet therapy. But today, almost every patient is on dual platelet therapy for long periods of time. And even if they have no symptoms, and even if they have had an endoscopy done which shows no GI pathology, still they continue to remain on PPI. I think we should be coming up with guidelines because a vast majority of daily maintenance PPI is those who are diabetics, cardiac patients who are just co-prescribed because they happen to be taking an aspirin, a clopidogrel, or even a statin. They just are told that this goes along with that. We have to find some way to get over that barrier also.
But there the problem, Dr. Shiva, is that there is evidence showing the benefit in that group. The problem is until we have something saying that it doesn't help in that group, we won't be able to make that statement. Because for dual, so single active we know the NNT is very high for single, so that's why we said we don't use it for single. But for dual, until we have some
define the guidelines, say that okay, if the patient has had an endoscopy which is normal, if the patient has no significant, some risk factors also should be added.
Yeah, but I'm saying whether can we say that if somebody is endoscopy normal today, dual can we say that he will not develop a complaint in future? So that statement is difficult for us right now to make. But for single, that's why we specified, I think. But if you look at the practice, even single antiplatelet people are on PPI. That's what.
So Dr. PK.
Sir, I've got a comment and a question. All of us, in fact.
Your mic is not working.
Hello.
Yes.
Day in and out, we are in fact, using PPIs as said by you all, sir, and as well as Dr. Shiva sir said about it. So injudicious use of PPI in fact has been too much there in our country, just for sure. Number one. Number two is that, sir, do we really need to give in combinations, the fixed combinations which are there available? As ma'am you pointed out, don't we need to really give it in a titrated method? Number three, sir, most of our elderly patients are addicted to PPIs. So is it more of a psychological, physical dependence which is there on them? And how do we get rid of it? And Dr. Umesh, what about the use of impedance pH manometry? Number one, it is very difficult to read.
So PK, one moment. Let Dr. Amit answer the first question.
Sure.
You can come to the second question and please have short answers because we have other people who want to ask questions.
So I think in fact, so what you said is what we made the statement. So we are also discouraging, that's one of the statements, the strong thing is that we should not encourage use of fixed dose comps. So what you said is what we have said in our guidance. So we agree with you on that. Yeah.
Okay. Your second question.
So yeah,
in a patient, how good is impedance?
In a patient of GERD-like symptoms, let's say we do an endoscopy. Suppose after 4 to 6 weeks of therapy, if he's not improving, we do endoscopy. Endoscopy is normal. The patient still has symptoms, not improving on double dose of PPIs. This is the investigation of choice. This plus manometry, if available, both can be done together so that we can classify the patient whether he's having reflex hypersensitivity, whether he's having functional heartburn. So then after the classification, if he has reflex hypersensitivity, suppose whether normal reflux patient is having symptoms, we need to put on antipsychotics. He doesn't need a prokinetic, doesn't need a PPI. If he has a volume refluxer, we add prokinetics. So this is the answer. I mean,
I've got a comment.
So can I just answer that in the same context? When you are looking at a patient of GERD, you have to first decide whether the symptoms are really reflux or not reflux. If a patient has never been investigated in the past but has been put on PPI and does not respond, your endoscopy is normal. You're really not sure whether this is gastroesophageal reflux disease. In that case, the best investigation to do is to stop all the drugs, wait for 10 days, and do a plain pH metry. It's cheaper.
True.
However, if you want to see whether the patient also has regurgitation, then in that case an impedance can be added.
Ma'am, I have got one reservation here. I do all the three things: a simple plain pH metry, an impedance, and a 24-hour pH metry where there is no impedance. And to my surprise, normally what happens is in most of the places we have got variation of results coming up. On a simple 24-hour pH metry, usually a D-Mister score doesn't give you any answer. On an impedance, you find a very high impedance coming up more than 200s. The D-Mister score come up. And on a manometry, you basically are confused whether it's a real reflux or not. So how do we go ahead?
So I think that's a separate topic for discussion. It's a really long topic, and I can sit with you personally and
Ma'am is a
take
master in that.
Yes,
and
Professor Brewer.
My question is that in some patients there is a genuine indication for using PPI for a long time. Now we have been hearing about these adverse effects for almost last 25 years. Are there any guidelines? How do we monitor these patients for these adverse effects? How frequently we should check their B12 levels, iron levels, magnesium levels? After we should do the DEXA scan to check for osteopenia, etc.
Yes, Dr.
Any
Amit.
guidelines by our society or by any society?
So that's a very important point. Actually, in our literature review, we looked at all that. Is there any recommendation for monitoring? As of now, no recommendation for doing a DEXA. No, not for even a B12 level actually. So although the deficiencies are known, but the recommendation to monitor frequently is not yet there for these conditions, especially for bone also. There's no need for DEXA as of now. So you have to get some evidence if it's beneficial. You have to show evidence is there that we do a DEXA or this thing that is beneficial for the patient, it has benefit effect. Then only we can. Until we show beneficial effect, we can't make. It will be like again an expert opinion.
So we have the last question because he's been standing.
It's in continuation with the adverse effects. Is there any relationship between the dose of the PPI that the person is taking and the adverse effect? Because as discussed, it is practically not possible to stop PPIs in all the patients. And if suppose a patient is taking half the dose and still is the risk of adverse effects high?
So the adverse effect has been more looked in terms of the duration perspective. Dose has not been that much investigated, and what we found definitely for most of the adverse effect, duration is a very important concern, and actually the dose part.
Bhatia ma'am, one question.
Yes.
On-demand therapy, when the patient recurs after stopping the treatment, so it has to begin again for 8 weeks, or there is some guideline for on-demand therapy.
So,
So-
Amit is going to answer that question. Yes.
So on-demand therapy doesn't have to be full 8 weeks. Actually, there is no set pattern, but people will use for 1 week, 2 weeks, but you won't use for full 8 weeks. Even the initial therapy is about 4 weeks is what we say, but maximum 8 weeks actually. But about a week or two, you can even use half dose if you want to. You can use it. So there is a lot of flexibility in that therapy option. If you look at the trials for on-demand, they are not similar in what they have described on-demand as actually. So sometimes they describe it as giving it for about 2 weeks. Sometimes it's flexible.
I think we are running out of time now, and I would like to thank all the speakers for their excellent job which they have done. I think we have gone through a very extensive discussion. There is a lot of interest, but in the interest of time, I would like to close the session now.
Thank you. Thank you, everybody.
Thank you, sir. Thank you, ma'am. For the next session on upper GI, can I please invite Dr. E. Peda Viraju, Dr. Kunal Das, and Dr. P.K. Agarwal to please chair this session.
Before my other co-chairpersons come up on stage, this is the last session for the day, and thanks for your patience. The first topic is on Chicago 4 classification of esophageal motility. I request Dr. Nitesh Pratap to.
Thank you, sir. So today I'll be talking on Chicago 4 classification, how different it is from Chicago 3, and including its clinical implications.
So we know that high resolution manometry has simplified esophageal motility disorder testing as well as diagnosis. And the Chicago 4.0 was published in 2020. So what is the Chicago classification? Chicago classification basically helps us to categorize esophageal motility disorders based on the parameters we get from high resolution manometry using an algorithmic approach. So the manometry protocol, normally we place the catheter transnasally. After we place the catheter, we have a 60-second adaptation period followed by a 30-second basal recording in which we look at the lower esophageal sphincter, and this is followed by 10 swallows of 5 ml at 25 seconds apart. Now in the new version, they have included a positional change. Suppose you give 10 swallows and the conclusion is not coming on manometry, you can do 5 swallows in upright position. Even if that doesn't help, then they have recommended for provocative testing, which includes multiple rapid swallows in which you give 2 ml swallows every 2 to 3 seconds, or a rapid drink challenge in which you give a 200 ml liquid bolus, or pharmacological provocative tests using amyl nitrate or cholecystokinin. So basically, if you are doing a manometry and you have done a provocative test also and you are not coming to a conclusion, then they have told that you should use additional tests like a time barium swallow, flip, or impedance should be used. Now, how does a normal manometry look like? So if you look at this is upper sphincter esophageal body motility and you are looking at the LES. So in the LES, we take the first 4 seconds of maximum deglutative relaxation after a patient swallows, and this is what we call as the integrated relaxation pressure. Normally, it should be less than 15. The other thing we look is for the esophageal body, we are looking at the contraction amplitude. Normally, the contraction amplitude is between 450 to 8000. Less than 450 is suggestive of a weak peristalsis, while more than 8000 is suggestive of a hypercontractile esophagus. The last thing what we look at is the distal latency, which is the point from which the upper esophageal sphincter has opened to the contraction deceleration point. Normally, this should be less than it should be normally more than 4.5 seconds. If it is less than 4.5 seconds, it indicates that it's a premature contraction or a spasm. Now, based on these parameters, Chicago has classified these into disorders of esophagogastric junction outflow and disorders of peristalsis. The EJG outflow obstruction includes eclasia and EJG obstruction, and disorders of peristalsis include absent contractility, ineffective esophageal peristalsis, distal esophageal spasm, and hypercontractile esophagus. Now, if you see eclasia, we all know is characterized by absence of body peristalsis and impaired swallowed induced LES relaxation. So eclasia based on the pressure has been classified into type 1 or classic eclasia, in which all the swallows have a failed peristalsis. In type 2, 2 or more swallows have got a panesophageal pressurization, and in type 3 eclasia, 2 or more swallows are spastic. Now, if you compare Chicago 3 to Chicago 4 for type 1, type 2, type 3 eclasia, there has been no change. But the new thing which has come up is the inconclusive diagnosis for eclasia. This is almost in 20 to 25% of the patients. And inconclusive means these are those patients, especially type 1 or type 2, who do not have a contractility and IRP is upper limit of normal. So they suggest that in this group of patients, you should do supportive tests like a time barium swallow or a flip to prove outflow obstruction. The other group is the inconclusive for type 3 eclasia. This is those group who have an abnormal integrated relaxation pressure but evidence of peristalsis. In these patients, you shouldn't label them as eclasia. They should be labeled as EJG obstruction with spastic fissure. The second group is the EJG outlet obstruction, which is almost around 10% of the patients undergoing HRM. This is previously we have always considered this as a clinically inconclusive diagnosis. In these patients, you have got an elevated median IRP, and there is evidence of peristalsis. These patients may evolve to eclasia, may represent a eclasia variant, or in one third, that is around 30% of the patients, they might be clinically irrelevant, might be because of some benign causes or opioid use, or it can be an abnormal artifact because of pressure measurement. That is the reason that Chicago has brought a major changes in the diagnosis in this group. So for us to diagnose a patient as esophagogastric junction outlet obstruction, you should have manometric findings, that is elevated IRP. More than 20% of the swallows should have an increased intrabolus pressure, and a peristalsis should be present. And these with clinically relevant symptoms of dysphagia and non-cardiac chest pain. So apart from these two, you should have a supporting evidence of obstruction with time barium swallow or flip showing obstruction. So EKG obstruction, you have to have manometric findings, you should have clinically relevant symptoms and supportive evidence of obstruction in either time barium or flip. Additional criteria which will be supportive are outflow obstruction on RDC or solid test meal or pharmacological provocation test. So the main reason why there has been so much change when compared to type 3 is, if you see in type 3 it was just manometry, but now in type 4 they have added symptoms, they have added confirmatory non-HRM supportive tests to look for outflow obstruction, because they have seen that in these group of patients, if you spontaneously follow them, there is a resolution of symptoms in almost 15 to 70%. So mainly the aim of making this guidelines more stringent is to prevent overdiagnosis and unnecessary therapy with myotomy in asymptomatic individuals. Chicago usually recommend that botulinum injection can be first considered in these patients with severe symptoms, especially obstruction symptoms, because they have got a beneficial response. So coming on to the disorders of peristalsis, the distal esophageal spasm, for us to diagnose clinically relevant distal esophageal spasm, you should have a manometric diagnosis. That means that when you give a swallow, more than 20% of the swallows should have a premature contraction with a distal latency of less than 4.5. The IRP should be normal and DCI should be more than 450. And you should have a clinical relevant symptoms of dysphagia with or without non-cardiac chest pain. So when compared to type 3.0 Chicago, 4.0 has added symptoms of dysphagia and non-cardiac chest pain. And also in patients in whom inconclusive diagnosis is there, if the premature contractions are associated with a DCI of less than 450, then you call it as an inconclusive. This is very commonly seen in patients with GERD. The next disorder of peristalsis was hypercontractile esophagus. For us to have a hypercontractile esophagus which is clinically relevant, you should have at least, when you give a swallow, 20% of the swallows should have a hypercontractile pattern, that is a DCI of more than 8,000, and clinically relevant symptoms should be there of dysphagia and non-cardiac chest pain. And it is very important that they shouldn't meet the criteria for eclasia, and mechanical obstruction should be ruled out, because mechanical obstruction can also have similar presentation as a hypercontractile esophagus. So when compared to type 3, they have added now that dysphagia and non-cardiac chest pain has been added in the 4.0 definition of hypercontractile esophagus. So apart from this, they have divided the hypercontractile esophagus into 3 groups. One is the single peak group. The second is the multiple peaks, that is what we call as the jackhammer esophagus. And the third one is a vigorous LES after contraction. Vigorous LES after contraction is usually not associated with any obstructive features, but jackhammer has got a very high DCI and has got the worst symptoms. The main implications is that almost 72% of the patients with hypercontractile will have symptom resolution in a 2 years period. And the predictors of patients who will have continued symptoms include patients who have dysphagia and a DCI of more than 30,000. So the Chicago group says that because there is a resolution in majority of the patients, they say that you should have a cautious approach while treating these patients, because initially you should try for conservative therapy before looking for any endoscopic or surgical therapy. The third group of patients in disorders of the esophageal peristalsis include the ineffective esophageal peristalsis. This is the most commonly reported esophageal motility disorder and seen in around 20 to 58%. Now, for us to diagnose ineffective esophageal motility, you should have a manometric diagnosis of at least 70% of the swallow should be ineffective. That means weak. So the DCI should be more than 1000 but less than 40,000, or they should be 50% failed peristalsis. If it is 50 to 70% ineffective peristalsis on manometry, then you should do supportive tests like poor bolus transit on barium esophagogram or multiple rapid swallows showing a lack of contraction reserve in these patients. So when compared to the Chicago 3.0, Chicago 4.4 has changed from 50% ineffective swallows to 70% and added more than 50% failed swallows. The term fragmented peristalsis has been removed. Ineffective esophageal motility is a very common thing, almost seen in 50% of the patients with GERD. It is very commonly seen in patients with Barrett's esophagus, diabetes, collagen vascular disease, and also in healthy patients in almost around 10% of the patients. Now, the main implication is for those patients who are going to undergo surgery, GERD patients who have got dysphagia planned for surgery. If ineffective esophageal motility is there, there is an increased risk of post-fundoplication dysphagia, especially if you are looking at a 360-degree fundoplication. And if there is ineffective esophageal motility, magnetic sphincter augmentation should not be done. Now, the last group is the absent contractility group. This is the group in which the IRP is normal, both in upright and supine position, and these patients have got 100% failed peristalsis with a DCI of less than 100. So this is most commonly seen in collagen vascular diseases, especially in scleroderma. So this is almost synonymous with what we call as scleroderma esophagus. In a study of patients with scleroderma, they found out that almost 56% of the patient had absent contractility and 33 patients had both absent contractility and very low LES pressures. So it is a very common thing which we see in patients with scleroderma. And other part is few patients will have an absent contractility but borderline high IRP. So this is the group of patients in whom you have to rule out type 1 eclasia, because type 1 eclasia has to be ruled out. So in these patients you have to do supportive test with a time barium swallow and or flip to show that there is an outlet obstruction to document. So when compared to Chicago 3.0, Chicago 4.0 definition, there is not much of change in absent contractility. So I conclude by saying that Chicago update has improved the precision of previously characterized motility disorders. Chicago 4 deletes infrequently encountered diagnosis, or it has removed clinically irrelevant data. It has given patterns where you can go for a very conclusive diagnosis of motility disorders. If you are doing a HRM and if you are not able to come to a very conclusive diagnosis, then you have to add symptoms as well as supporting tests for diagnosis of these disorders. Esophagogastric outlet obstruction, distal esophageal spasm, and hypercontractile esophagus are motility patterns, and these must be accompanied by esophageal symptoms of dysphagia and or non-cardiac chest pain to be considered clinically relevant. Thank you.
May I call upon the next speaker, Dr. Shobna Bhatia ma'am, to deliver something very important about the resistance to antibiotics in H. pylori, choosing the right combination in view of the emergent resistant strains or the new emerging strains which are resistant to. Shobha ma'am, and we'll have the discussion at the end of Madam's talk. Have your questions ready, sir.
So, good evening, everyone. I have a small thing to say that this is not a topic I normally talk on, and Dr. Usha Datta has kindly helped me with some of the slides. Some of the slides do belong to her, and I acknowledge her for that. So, first is whom should we treat? So, if you look at all the guidelines in the world, they say that all patients who have H. pylori infection should be treated. There was an editorial almost maybe 20-25 years ago which said the only good H. pylori is a dead H. pylori. Of course, we know that removing all the H. pylori from the world is not easy, and especially in India where more than 50% of the population may have H. pylori, it's going to be a difficult job. However, there are certain situations where the patient may need H. pylori eradication. If you want to start long-term PPI, we're going to convert a chronic gastritis maybe into an atrophic gastritis with H. pylori infection. Patients who are on long-term NSAIDs, who can have higher incidence of bleeds, and patients who are on anticoagulants, a family history of gastric cancer. It's better to prove H. pylori before you start treating. So the first thing is always prove, and even with mild fuzzy symptoms, don't go and treat H. pylori just because the Western guidelines say so. So how to treat? Which are the drugs? What dose? What duration? And how do we tailor it to India? I'm not going to talk about the routine treatment, and we'll be coming to resistance in a short time. So first, what are the drugs? Let's look at these drugs. You have the PPIs which suppress the H. pylori. Metronidazole causes free radical damage. Clarithromycin inhibits the protein synthesis. Amoxicillin inhibits cell wall synthesis. Quinolones inhibit DNA synthesis. Rifabutin inhibits the RNA polymerase. And bismuth inhibits almost all the enzymes that it has. I'll be coming to probiotics at the end when I'm discussing the other drugs. So same here, PPIs suppress the H. pylori. Now the H. pylori enters into a replicative state at a pH of 6 to 8. For the residents, go back to the seminal paper by Mary Marshall, where he tried to prove the Koch hypothesis. He drank the H. pylori culture, nothing happened to him. Then he took the cimetidine and he drank the culture and he got infected. So H. pylori can only be in the replicative state when the pH is 6 to 8, which means an alkaline stomach is good for H. pylori replication, but the antibiotics are also acting when the H. pylori is in a replicative state, which is why you use the PPI. Why do you use it in a double dose? You just heard from Dr. Amit Datta all about the PPIs. Single dose PPIs usually act for 16 to 18 hours. Very few of them can go to 24 hours, and therefore you need a 24-hour coverage when you're giving these antibiotics. So which PPI? We know that higher dose beta acid suppression is better to kill the H. pylori, get the H. pylori killed. Newer generation PPIs have higher efficacy. The best one is vonoprazan, which is not a PPI but a PCAB. And also esomeprazole is not affected by the CYP2C19 polymorphism. This is a very great paper to be read by all the residents. David Graham published this paper in 2018 in Helicobacter. Also, a similar one came up in gastroenterology, where he showed that the efficacy of the PPIs as compared to omeprazole 20%. Esomeprazole and vonoprazole are far superior. And when you add it to H. pylori eradication, then in that case, the eradication is much better with vonoprazan, because here it provides almost 24 hours of coverage. So what is vonoprazan? It's a potassium competitive acid blocker, inhibits the hydrogen potassium ATPase, rapid, profound and sustained acid suppression. And dose is 20 milligrams BID. As Dr. Shajan Peter told us, vonoprazan is available for H. pylori eradication in the US, but it is not yet approved for gastroesophageal reflux disease. It is not yet available in India. Amoxicillin, I already mentioned, it inhibits the cell wall synthesis, and in combination with clarithromycin, it prevents clarithromycin resistance. The dose is 1000 milligram BID. Most of us are scared to give this dose because the patient comes up with diarrhea, but the best dose to give is 3 grams per day. Same with clarithromycin. We have to give 500 milligram twice a day, and most of us do use that dose, but quite often it has a lot of resistance, which we'll be coming to. Quinolones inhibit the bacterial DNA synthesis, and there are 2 quinolones which we can use, levoflox and moxiflox, in the respective doses as mentioned here. Metronidazole is a very good drug for many things. Unfortunately, there's a very large amount of resistance. The active form, it prevents the free radical, it gives free radical damage to bacterial DNA, and it does not get metabolized in any human cell. It is metabolized only by the bacterial cell, and that is how the bacteria DNA is destroyed. Rifabutin is a rifampicin analog. The mechanism of action, it inhibits the expression of the beta subunit. The dose is 150 milligrams twice a day. Importantly, it is highly concentrated in the stomach, almost 10 times, and is stable even at a higher pH. Therefore, it's a very important drug to have local concentration. Importantly, it is a very low resistance in H. pylori infections. As far as bismuth is concerned, bismuth has been revived in the last few years. It was not available for so many years, and inhibits all the enzymes of H. pylori, that is the urease, the catalase, and the lactase. It also inhibits adherence of H. pylori to the surface epithelial cells, and more importantly, it overcomes clarithromycin and levofloxacin resistance. It is not affected by metronidazole resistance, and also the effect of bismuth lasts 7 days more than the duration that the drug is given. That means the drug levels are present in the blood and continue to act on whatever remnant H. pylori is there. It decreases the bacterial load of the H. pylori and makes the other antibiotics easier to work. There are 2 formulations available. The one that we most commonly use is the colloidal bismuth subsalicylate 1 to 20 milligrams QID. The patients always complain of black stool, so they have to be told about it. One more thing I have to say, remember, I don't know, there are some seniors in the audience, we used to use a mixture of bismuth and kaolin to treat diarrhea in the past. It was a very good mixture, and therefore, in some patients, it may be useful with antibiotic-associated diarrhea when the bismuth is added as part of the regime for H. pylori. Looking at probiotics, they secrete antibacterial-like substances, mainly lactic acid, short-chain fatty acids, and bacteriocin. Now, these lactic acid also decreases the pH inside the cell and therefore can kill the bacteria. They may inhibit the pro-inflammatory factor expression and many other functions, and destroy the biofilm, which is an important aspect of antibiotic resistance in H. pylori. Now, I had to show this work because this was my first international paper, which is very widely quoted now. Lactobacillus inhibits growth of Campylobacter pylori in vitro. We did this when the H. pylori was actually called Campylobacter way back in 1989, and we showed that Lactobacillus acidophilus helps in the culture medium. It inhibited the H. pylori, but when we gave it to patients of H. pylori, there was no difference in the eradication rates. And now there are studies of lactobacillus coming back as treatment in patients with resistance. So this is the antibiotic resistance mechanisms. There is the DNA stability, DNA replication, RNA transcription and translation, which may be affected. The beta-lactams are acting here, and for some reason they stop acting when there is a change in the bacteria. I will not be able to explain some of these because I don't understand that. Coming to clarithromycin, resistance is extremely high in the world, almost 30 to 40% in some parts, and in India it is close to 30%. That's here, and that's why we call it a area of high clarithromycin resistance. So what is the pattern of antibiotic resistance in Asia. The primary antibiotic resistance to H. pylori is quite high, and the resistance is going on rising. These are three drugs that have been looked at. If you look at clarithromycin, which is in blue, from 7% before 2000, going up to almost 21% in Asia. Levofloxacin has gone up to 27,000, and metronidazole at 36% to 45%. This is the whole of Asia. When you come to India, it's slightly different. Why am I mentioning this? First, the WHO Aware Committee has said, limit the use of antibiotics, especially because many of these antibiotics are useful in other disorders which are much more important. As far as India is concerned, the most important one is tuberculosis. If we increase the use of antibiotics in H. pylori, we are going to have some problems when treating tuberculosis, which is a much more fatal disease than H. pylori. And here we have clarithromycin, ciprofloxacin, levoflox, and rifabutin. There's a long list in this, but I've mentioned only 4 drugs which we use in H. pylori, and these are also useful in tuberculosis. So please limit the use unless we need it. So this is an important paper done by Halur et al. From Karnataka, where they looked at 113 dyspeptic patients and looked at the resistance. And what did they find? Levofloxacin 65%, metronidazole 81%, amoxicillin virtually no resistance, 3%, and clarithromycin is at 35%. And all across India, if you look at the papers, the multiple papers starting from the first paper in 1997 from KEM Hospital, where we showed metronidazole at 100% resistance, 91% resistance to clarithromycin. And subsequently, the most recent one from Odisha, which is showing almost similar results, except clarithromycin slightly less. There is one multicentric study in India with more than 200 patients, where again metronidazole is at 73% and clarithromycin was 45%. So which regime and what duration? So the drugs used, quickly go through them, everybody knows these. The standard, so we have 2 types of therapies, the standard and the sequential, first week and second week. For the standard ones, the first and the second week treatments are same. That is, here there is the PPI, amoxicillin, clarithromycin, with or without metronidazole. In the sequential therapy, you give amoxicillin in the first week, clarithromycin and metronidazole go to the second week. Concomitant therapy, again same. Hybrid therapy can be started, and quadruple therapy, which is 4 drugs with a PPI, metronidazole, otinidazole, bismuth, amoxicillin, clarithromycin in various combinations. When you look at these, you find that the results of therapy, eradication is standard is 76% today. This was 95% when it was first started almost 15 years ago. Sequential therapy is 83%, and hybrid therapy is again 80%, concomitant is 85%. So when a patient has clarithromycin resistance, concomitant and the bismuth quadruple resistance therapies are the best, because when you add bismuth, you automatically take care of the clarithromycin resistance. So durations, all these, as I said, are superior to triple therapy. 14 days is the most optimal, and the definition of an optimal regime is you should have more than 90% eradication. If you look at the previous table, none of the therapies had more than 90% eradication. So what are the guidelines? I've combined the guidelines into one because they almost say the same thing, that in areas of low clarithromycin resistance, that is less than 12%, its triple drug is even fine. But moment there is high clarithromycin resistance, add either metronidazole or a bismuth to overcome the clarithromycin resistance. Remember that clarithromycin alone will cause a problem, but when these are combined, the drugs are more effective. And with dual resistance, again, bismuth is the one to be added. More recently, the Indian guidelines said that nitroimidazole, that is metronidazole, should not be used for H. pylori. And in areas of high clarithromycin resistance, in both cases, it said that bismuth should be the drug of choice to be added when a patient has suspected to have clarithromycin resistance. So how do we improve the efficacy? By adding bismuth, we improve the efficacy by 10%. By adding probiotics, more than 10% to any of the regimes, replace clarithromycin with cleomethasone, or replace PPI with vonoprazan, and that is the best combination so far. I'll be finishing in about 1 minute. We talked about probiotics, adding probiotics to improve the results, and when you add these, there's one study about 3 years ago which said that yes, adding a probiotic improved the efficacy by about 8%. Subsequently, 3 more randomized controlled trials using different probiotics in different combinations have shown absolutely no difference in the efficacy with H. pylori eradication. So the Indian perspective, real life eradication rates are about 86% with 4 drugs. Resistance rates are high. This is the latest paper just published in Indian Gastroenterology Online from Mumbai, where concomitant therapy was similar to sequential therapy, 87% versus 81%, and they concluded that in spite of resistance, the in vivo responses are much better, though the in vitro antibiotic resistance is high. So what we can do is add bismuth, choose a high dose PPI, choose new generation acid suppression, that's vonoprazan, choose a higher dose of amoxicillin, going to 2 to 2.5 grams a day, ensure compliance with 14-day antibiotics. And in conclusion, we know that antibiotic resistance is common, maximum with metronidazole and clarithromycin. Use antibiotics judicially and add bismuth. And my only request to all of you, please do not overuse antibiotics for diarrhea, cough, so that we can limit the use and improve the resistance of H. pylori. Thank you.
Thank you, ma'am, for a very comprehensive and very good lecture. Now we have a third final lecture of the session by Dr. Amit Datta, a very common topic, and all of the gastroenterologists do see functional dyspepsia. So Dr. Amit Datta will tell us what is the functional dyspepsia standard of care in 2022 or 2023 now.
So thank you, chairpersons, and good afternoon again. So I thank the ISG for inviting me to give this presentation. So I'll be speaking on management of functional dyspepsia. So dyspepsia basically refers to the group of symptoms. It's a conglomerate of, it's not one, it's a conglomerate of symptoms originating from the upper GI tract, and the cardinal symptoms include epigastric pain, epigastric burning sensation, fullness in upper abdomen, bloating, early satiety, belching, nausea, etc. And also about 20 to 30% of patients have symptoms of other conditions like GERD, IBS, and they have dyspepsia. It's a very common condition in the population, affecting about 25-30% of them. And although it doesn't affect the patient's lifespan or longevity, it has a negative impact on patient's quality of life, and that is why we have to diagnose this condition, manage it appropriately. So as I said, dyspepsia is basically not a single but a group of symptoms, and also it is not a single causative factor, it's a heterogeneous condition. In a small proportion, we can find a cause like a peptic ulcer disease, infection, etc. But in large majority, that is about 2/3 of them, no cause is found on upper GI endoscopy. So these are called as patients having functional dyspepsia. And also nowadays called a disorder of gut-brain interaction, or DGBI. And here again, although endoscopy is normal, these patients have been found to have abnormalities like dysmotility, hypersensitivity, etc. So the definition of dyspepsia has been given by the Rome group, and it talks about the presence of cardinal symptom, at least one of the 4 symptoms, which is epigastric pain, epigastric burning sensation, early satiety, bloating. One of them should be there, and then they have the duration criteria, frequency criteria. If somebody's symptoms are more of satiety and bloating, then they are called as having PDS or postprandial distress syndrome. If the symptom is epigastric pain or burning, then it is epigastric pain syndrome. But in clinical practice, up to 1/3 of patients may have overlap, And actually data from India shows that overlap may be seen in more than half the cases actually. So overlap is more common than having an isolated PDS or a EPS. Now these criteria are good for clinical research when you want to have uniformity of recruitment of patients and follow-up, etc. But in clinical practice, we don't have to go strictly by the criteria, we have to use a clinical judgment. Even the Rome committee, if you look at a paper published last year, what they said is that for clinical purpose, we should relax this frequency and duration criteria. So for dyspepsia, basically the symptom is important and how it affects patients' quality of life. And as I told you that in Indian scenario, data has shown that about half the patients may have actually overlapping than isolated PDS or a EPS. The other entity which has been there for last few years is called H. pylori associated dyspepsia. So what is this entity? So if somebody has a dyspepsia and he also has H. pylori infection, you treat the H. pylori. After treating H. pylori, if the symptoms improve and there is a sustained response, that is patient is okay for at least 6 months after that, then only you call it as H. pylori associated dyspepsia. So anybody with dyspepsia and H. pylori does not automatically become H. pylori dyspepsia. And I must tell you, this happens only in about 1 in 12 or 1 in 15 patients. So out of maybe 12 to 15 patients with H. pylori dyspepsia, only 1 will show a sustained response in symptoms. So therefore, H. pylori associated dyspepsia is not as common as H. pylori infections. Now, we have an issue with that definition, say at least for our population. So if you look at the randomized control trials done from India on H. pylori and dyspepsia, we have 3 RCTs. The first one came more than 20 years ago. There was a smaller RCT and there was a benefit of H. pylori therapy. But subsequently, we had much bigger trials. The one in 2013 from Kashmir, which had about 400 subjects. There was no difference in response rate between H. pylori eradication versus the control. Again, a more recent study in 2021 from Delhi, where they had again H. pylori therapy versus PPI, and there again there was no difference in response. So therefore, what we are seeing in our country is that among patients with H. pylori and dyspepsia, eradicating H. pylori is not giving us the therapeutic response. So therefore, this test and treat strategy which is proposed by the Western group may not be very applicable, and it will only increase the resistance of H. pylori, and then somebody who really needs it will probably find it difficult to treat those patients. So therefore, if you look at the ISG guidelines on H. pylori, while there is an association between functional dyspepsia and H. pylori, the data to support eradication isn't sufficient from India. So we, at this point, we are not in a position to follow this test and treat strategy as the initial part of management of dyspepsia. Next question always comes is about endoscopy. When should we do endoscopy in dyspepsia? If there are alarm symptoms, not much of an issue. But what about the age cutoff? So if you look at the Western guidelines, they talk about 55 years of age. But data from India and from other Asian countries have shown that cancer occurs at a bit of an earlier age group. So our age cutoff cannot be same as the Western age cutoff. So this is the data from our own center, CMC Vellore. We had studied more than 2,000 patients with dyspepsia. And what we found is that we have to take a cutoff of 40 appears appropriate. If we make it 45 or 50, then we'll miss a significant number of patients with gastric cancer. Same was the finding from a study from Bangalore, where they found that the age cutoff should be about 46. Even the American guidelines, we look at the fine print, they say that for the Asians, this 55-year cutoff may not hold true. So for our patients, if you're doing endoscopy and age is your factor, probably 40 to 45 years is the good cutoff, not 55. Next is about dysmotility. So we know one of the mechanisms, one of the proposed physiological abnormalities in patients with dyspepsia is a dysmotility. Up to 20 to 40% of patients have been shown to have abnormal gastric accommodation. And you need specialized tests like Verostat or SPECT, etc. to prove this actually. So, but the issue is that the correlation between the gastric emptying and the presence of symptom is not very consistent. So therefore, how it will guide us how it will guide therapy initially is also not very clear. So right now, therefore, even though this abnormality is seen, the routine testing is not recommended, and this has been also endorsed by the Canadian and the American group in this guideline, where they say that we recommend against routine motility studies for patients with functional dyspepsia. The next point I would like to emphasize upon is the overlap. So as I told you, these disorders don't always come in isolation. In fact, about 20 to 30% of patients will have coexisting symptoms of IBS, GERD, or some other disorders. So when you see somebody with a dyspepsia, also ask for history for these coexisting conditions. So just to summarize what I have said so far, that dyspepsia, you have the subtype, the PDS and the EPS, but looks like in our population, the mixed variety probably is more common than the isolated ones. For H. pylori, the RCTs are not showing benefit of therapy over a PPI alone. So therefore, the test and treat strategy which the Western people propose may not hold true right now in our country. For endoscopy, I think an age cutoff of 40 to 45 seems appropriate. This motility, although present, we don't need to do it in every patient, and look for overlapping features of other disorders of dyspepsia when you're seeing these patients. So moving on to the management of dyspepsia. Now we have both pharmacological and non-pharmacological management. A few important points to remember is that when you see a patient and you have done say an endoscopy is normal, but still we have to make a positive. We cannot say that everything is normal, you don't have any problem, there's no condition. Because as I told you, it's a real condition, patient has symptoms, it's affecting quality of life. So we have to make a positive diagnosis, tell the patient there's a real entity, there is a therapy required for this, patient has to be on follow-up. So we have to empathize with the patient, acknowledge the presence of symptoms, educate the patient about the benign nature of the condition, at the same time also getting treatment and what the realistic goals of therapy are. We should also find out about what treatment the patient received in the past, so that will help us deciding the therapy which the patient will need, what he responded to, what he didn't respond to. Now we don't have any biomarkers in stool or blood like we have on IBD to follow up the patients of dyspepsia, because they don't have any abnormality in those parameters, most of them. So here we have, for them we have what we call this scoring system for quality of life, for symptom severity. There are quite a few of them. These are mostly used in research tools, but it's good to be aware about them. I think more recently, a Hindi version of this short form Nepean Dyspepsia Index has been validated in our country and published recently in Indian Journal of Gastroenterology. So if somebody wants to do a research on this topic, then this may be some scoring which they may use for their purpose. So among the non-pharmacological thing, the strong evidence for any specific dietary or any specific avoiding diet is not present right now, but it may be beneficial in some patients. So one thing we can tell the patients is that you can avoid food which is triggering symptoms. For those who suffer with this postprandial distress syndrome, early satiety, bloating, then we can tell them to take small frequent meals. Again, promoting healthy lifestyles may also be useful. There was again this recent paper published from India on low FODMAP diet for functional dyspepsia. Now, this did not find any difference in the overall dyspepsia group per se, but when they looked at the group with postprandial distress syndrome, in that group there was some benefit of a low FODMAP diet in dyspepsia. So next we talk about the pharmacological therapy. Now this network meta-analysis shows most of the trials with drugs done for dyspepsia. So the big circle is obviously the placebo, because most drugs have been compared with placebo. Majority of trials have been done with acid suppression, standard dose PPI or low dose PPI, high dose PPI, H2 receptor. Some trials with prokinetics like domperidone, etoposide, mosapride, and some with drugs like amitriptyline, SSRI, SNRI, etc. So among these medicines, what we see, the drugs which have good efficacy are the acid suppressants, the tricyclics, whereas the prokinetics don't have as much efficacy. So PPIs, there are a number of RCTs showing the PPIs have a good effect and they are the standard of care for initial management of dyspepsia. Whereas for the drugs which affect the sort of motility, the evidence is not that much strong. So therefore, for pharmacological therapy, PPIs are generally the first line therapy for dyspeptic symptoms. They have more efficacy than H2RA. But again, remember, the duration of therapy should be 2 to 4 weeks. I was just talking about it in the PPI guidelines that we should limit the duration of therapy, tell the patient the medicine should be taken for about 2 to 4 weeks. And then after that, if the patients have recurrence of symptoms, you can use on-demand therapy. And the subtyping of dyspepsia, which again we are talking about, as of now, there is not again very solid evidence saying that if you have a postprandial distress syndrome, you use a prokinetic, or if you have a pain syndrome, use PPI. There are very few trials comparing that to subgroup, and those trials, the results have been equivocal. So right now, based on evidence, we cannot say that if somebody has a PDS, you give him prokinetic as a first line of therapy. There also probably PPI may have efficacy. Now having said that about PPI, H2RAs are still very relevant in therapy. They still have about 20 to 30% more benefit than what a placebo has. So somebody has a very mild symptom, in them you can use this as an initial therapy also. They are good choices as a step down. So somebody on PPI, you're not able to suddenly stop it, then you can step down to a H2RA and then you can subsequently stop the therapy. Antidepressants are the second line therapy. Those who don't respond to PPIs or acid suppression use antidepressants, and the common drugs are amitriptyline and mirtazapine. Obviously, psychotherapy like CBT, hypnotherapy, etc., have been shown to have benefit, but the problem is these are not very freely available, and therefore, practically, we don't have access to these therapies for most of our patients. So we have to rely on pharmacological therapy. So the agents affecting motility. So first line is acid suppression. Then you use your antidepressants. If somebody has the coexisting psychiatric symptoms, then antidepressant can be used in the first line itself as an add-on to PPI. Now, if these therapies don't work, then you can consider adding the prokinetic as an add-on therapy. So here also, acosciamide is one drug where there has been benefit, but this has been mainly the trials have been done in Japan. There's not much of evidence outside of Japan. For etopride, mosapride, the evidence is not that much strong. So if you summarize the pharmacological management of dyspepsia, then proton pump inhibitors are probably the first drug of choice. The standard dose PPI for 2 to 4 weeks is what we would recommend. H2RS can be used as an initial therapy for mild symptoms or as a step-down therapy in patients. Antidepressants, second-line therapy, or if somebody has a strong coexisting psychological symptoms. For drugs affecting motility, the evidence for benefit is not that strong. It can be used add-on therapy, especially in somebody who's got a postprandial distress-like symptoms. Now, H. pylori, I said at the beginning that RCTs show no benefit of H. pylori over PPI. So, should we completely shut off? I don't think we should completely shut off this option to our patients. Now, somebody with dyspepsia, you've tried PPI, H2R, you've tried antidepressants, and patient is not responding. In those patients, there may be a case of checking for H. pylori and treating, because then this patient needs some therapy. And so, it may be in the select group of patients where the routine therapies are not helping, we can consider doing this H. pylori testing and eradication. Just want to touch upon this MDCP or multi-dimensional clinical profile, which has been proposed by the Rome group. So it divides the patient's condition into different dimensions, like the diagnosis, the clinical modifier, how it impacts patient's daily activity like his work, life, etc. In the psychological comorbidity, physiological disturbances. So the benefit of this is that once you break it down into different components, then you are able to address each of the components. You can provide a comprehensive care, and also you can provide individualized therapy and follow up the patient accordingly. Now, the one important aspect of management we should not forget is the follow-up. So as I told you, when you start therapy, everybody may not respond to your initial therapy. So in these patients, that's why when you treat somebody with dyspepsia, please have a follow-up plan. If they are not responding, they may need a change of therapy or further evaluation. And also it gives us a chance for de-prescribing PPI or any medication. So because patient may be continue to take the medicine what we prescribe, so when we see on follow-up, we can tell them they are better, they can stop taking the medication to prevent overuse of the medication. So with that, I come to the end of my presentation. Thank you.
Thank you, Amit, and all. Please join us on stage. Thank you, Dr. Amit, and all the speakers for their very comprehensive talks. The topic is open for discussion. Any comments, questions? Shiva Prasad.
Thank you. Shobha, just one question as far as metronidazole resistance is concerned. And there have been studies all right from the beginning that the lack of correlation between in vivo and in vitro actually applies most to metronidazole, and that may give the reasons why there's so much conflicting data. So what do you suggest we do? Now we know that you look at the guidelines that ISG has recommended, you're saying go straight to 4 because clarithromycin resistance is widespread. Correct. So we cannot use the standard kits which are available now. So should we go for 4 drugs with bismuth, or do you think we can still say 4 drugs with nitramidazole in our scenario? I mean, first is 4 drugs recommended for all patients eradication in India. You think that should be the way forward? And if it is 4 drugs, can we still try nitramidazole with PAC, or should we only go for bismuth? I'm asking this because bismuth is not that easily available, and especially in this combination form.
So, as far as combinations are concerned, I suggest not using any of them because they're usually underdosed. Yeah, and use the best PPI available. That's the first. So if you look at metronidazole resistance, I didn't bring out the Japanese and the Korean data, where in patients with metronidazole resistance, if you use vonoprazan, which is going to give you 100% acid suppression, that metronidazole resistance can be overcome. So from 70%, it will go to 90% eradication rates. But we don't have that drug. So if you can increase the PPI or increase the acid suppression. So I had mentioned about David Graham's study in Helicobacter. There's a similar one in gastroenterology where he talked about the efficacy of PPIs versus omeprazole. So on a dose by dose basis, If you take 20 milligrams of omeprazole as the standard, then in that case, pantoprazole is just about equal to 4.5 milligrams of omeprazole. So using that in an H. pylori regime is usually not useful. Use esomeprazole or rabeprazole, where it is equal to 30 or 32 milligrams of omeprazole. So use that twice a day rather than the other this thing. I'm not saying pantoprazole is bad, but at least in H. pylori, use a stronger PPI. Using that will overcome clarithromycin as well as metronidazole resistance. Unfortunately, there are no studies from India about metronidazole because data has said that there's 80 to 100% resistance here. And coming to your question about using 4 drugs straight away, I do that. At least in patients with indications, that is ulcers, bleeds.
Before the secretary ISG asked somebody in the back.
So, question to Shobna ma'am. Ma'am, regarding the dosing of amoxicillin, when you are giving a quadruple therapy with bismuth, is there a option to reduce the dose as you prescribe, like 3 gram up to 3 gram? Whether we can reduce the dose of amoxicillin with bismuth?
So, as I mentioned in my talk, we are scared to give 1 gram twice a day. Many people will still go to 750 milligram twice a day. Most of the kits have only 500 twice a day. But use the higher dose if the patient can tolerate it. Okay, 750 is what you would use in a slide, which I use quite frequently because again 1 gram I'm worried about the diary. Yeah.
Any the relation with the meals with the bismuth, whether we can give after meals, before meals, is there anything?
No,
before
you can
food.
give, yeah,
Bismuth is
bismuth
before.
is before food, the rest of them after food. Yeah, but tell the patient you get black colored stools. That should be bond.
The president of ISG, Govind.
Yes, sir. Yes.
I just want to emphasize one point that once we think of H. pylori, we write very loosely drugs.
I'm sorry, I didn't get
Once it's a comment, it's not a question to you. So once we think of writing H. pylori, the first question that do you want to treat it? You want to treat it, then treat properly.
Yes.
Give proper doses, proper duration, and confirm eradication.
Yes.
Many times we just want to write drug and we're not serious about it. Therefore, we write non-serious treatment.
Yes.
Combinations, low doses, using metronidazole or so. So I think the good practice would be to first ask yourself that yes, I need to treat this patient and choose right combination, right duration, and right
antibiotics.
doses.
And yeah, I think Govind, that's a very important point that you've made because many people test for H. pylori for any patient that comes with mild dyspepsia. They find it borderline light pink, give the treatment, and it's more like a placebo. These patients are never retested. Patient gets the feeling he's been treated but never retested. So yes, retesting is important and prove eradication. At least for the financial reasons, I recommend testing for eradication in patients who had a bleed, who have a peptic ulcer, a maltoma, and patients who are at a risk for getting gastric cancer. The other patients, it is sometimes telling them to have a second test after the eradication therapy is little difficult. Yeah.
Madam, can I ask you a question on this?
Yes. So there are 2 other speakers.
yeah.
Please remember that. Yeah.
Wait, I think we're over with the session today.
No, there are 2 other speakers whom you can ask.
Yep, of course. Even Dr. Amit also. Sir, you know, I mean, as H. pylori is considered to be a normal common cell, 30% is found to be normal in a body, right? So, what do you think, you know, I mean, regarding about the position of H. pylori when we test, say it's positive, and what kits do you really recommend, you know, for a normal testing, and when do you say that it is really eradicated? Because it keeps on coming up with our Indian diet system. Dr. Amit, please.
I think the important thing is to decide when you should treat. That is a very important aspect. Actually. If you decide that, then you will not have too many patients. So, but once you treat, then you have to check for eradication as well. Now, whether it's coming back, that part data is not. There is a little bit of reinfection rate, but it may not be a major. More probably a primary therapy failure will be more likely than a reinfection is what.
So, like in Japan, you know, it's mostly because even to age of 90 years, you know, they're having the ESGs and EMR being done on the patients because of the H. pylori, which is getting the things worse up there. So what do you say for our country patterns?
No, so I mean, in terms of therapy, there are clear-cut indications, but once you treat, you should check for eradication. Beyond that, after eradication, you keep checking again. There is no real. I don't think anywhere else there is a guideline to
Do
do
you
that.
think that the H. pylori kits available in the market are good enough, sir? I mean, to give us the result?
So these kits, most of them will tell you how they have got it validated. So you look at the kit manufacturer and see how they got validated. There has to be some validation of anything you use actually. So without a validation idea, it will be difficult. So either or you validate in your own center. So these kits, I think, so that question is right what you're saying. Kits manufacturer may not be this thing. So you look at that data.
This was an audience. Anu, you can.
I have a question for Dr. Nitish. Excellent talk as usual. Regarding multiple rapid swallows, how do you do it? Two is, how does it really helps us to take a decision for fundoplication in GERD patients?
See, basically, what we are looking at in multiple rapid swallows, we give 2 ml swallows at 2 to 3 seconds, and after the swallow, we can see whether it is helping, whether the peristalsis is there, or there is a means if it is becoming ineffective. The main indication for these is that in patients who have got GERD and dysphagia, if there is ineffective esophageal motility of more than 70%, these are the patients who are going to have a higher chance of dysphagia. So these are the, and so this is the group you have to be careful about.
Dr. Dheeraj Nagaj, question just ignited here only a very practical problem. Even in the somatic related GERDs, upper or dyspepsia or IBS, whenever we are opting for antidepressants, those kind of drugs. So later on, what going to be the step down strategy or the continuous strategy? Because the response is there. Now, how to go forward? Some thoughts on this.
So for the antidepressants, the recommendation is initial therapy about 3 to 6 months, not more than that. After that, you try to stop actually. And then if the patient has recurrent symptoms, you have to use. But initial therapy should be about 3 to 6 months. That's what I would say. Subsequently, it depends on individual patient how the symptom profile is. Okay. A low dose amitriptyline doesn't have too much of a side effect even for long. But you should always be judicious in your use of the medicine.
So just a quick thing to add, when you're going to add any of these neuropsychiatric drugs, tell the patient that these will act only after 3 to 4 weeks. Don't expect an instant response like they do for PPIs or any other drugs. So just inform them, otherwise they'll come back in 2 weeks and say no effect.
One question to Dr. Shobana ma'am. There is another very cheap drug which is here ma'am. Ma'am here.
Okay, sorry, I didn't see.
I'm Dr. Kunal. So
Yes.
there is a very one cheap old drug which is tetracyclines.
Yes.
That is also useful in H. pylori. What do you think its role? Especially seeing that many of the newer age drugs are getting resistance high, and even in UTI we have an old drug nitrofurantoin which is coming again. So what are your thoughts?
That's a very good question. Recently, there have been no studies on tetracycline in India. There have been some studies of international studies where they have replaced tetracycline with doxycycline with very good results. And I don't know, somehow tetracycline seem to have lost their popularity over the last 20 years for all diseases. It's not only for H. pylori, but yes, there is no resistance to to tetracycline so far. So it's a good drug, but somehow it has just died down as a popular drug. But maybe if it comes back, it's a good drug to look at.
In some of the guidelines, it is used as a replace or third rescue therapy.
So this
Tetracycline is still available in India.
very
Yeah.
cheap.
It's
This
very
is
cheap.
not a question. Yeah.
Yes.
This is not a question. Comment only. Whether to treat H. pylori patient or not. Let's say you have H. pylori. So whether we will eat H. pylori eradication therapy or not, we will take or not. Because a few years back from my center, we have done one study that is only published in Digestive Disease, epigenetic study. Out of 100, around 90, out of 100 gastric cancer patient, epigenetically we have seen not gastric. It is used to say that when there is ulcer or cancer, H. pylori used to evade from there. H.
Yes.
pylori we not get H. pylori in there. But out of 100, more than 90% gastric cancer patient that after resecting it, normal mucosa have H. pylori in that. So that is one comment. Whether we have to treat or not, we have to think that. That is one. Second is metronidazole resistance. In my again from our group, we have shown from Odisha that is 95% resistance. That data is showing 95% metronidazole resistance is there in this subgroup. So these two comments.
The first comment, I think I want opinion of the two speakers, because is it cost effective to treat all patients to prevent gastric cancer? That's the issue.
I think Dr. Amit has said that test and treat strategy for India is not working. But for Maastricht guidelines 2021, they say that you should test and treat every patient of dyspeptic if do endoscopy.
So
what is
remember
your
that.
thought?
Remember one thing, that Maastricht guidelines, European guidelines, USA guidelines are all based on an H. pylori frequency of less than 10% in the population. So they don't have to treat too many patients. Whereas we have to look at 50% of our population. So you cannot, I mean, first of all, we are not even sure that H. pylori is as harmful as it sounds. Gastric cancer, yes, if a family member has gastric cancer, if a patient has a duodenal ulcer with a bleed, and this patient is going to be put on any antiplatelets or on NSAIDs for a rheumatic disease, I would give the patient definitely an H. pylori eradication. But for other fuzzy indications, which is non-ulcer dyspepsia, mild pain, I'm not in favor of treating everybody who comes. I'm not even in favor of testing everybody who comes. Secondly, remember that every endoscopist goes and does an RUT without thinking that the patient is already on a PPI. If the patient is on PPI for the last 1 week, that RUT is going to be negative.
Last two questions.
Sir, is there a ground for pantoprazole 80 milligram per day, the tablet, in any of the conditions, like for H. pylori double doses to reduce the pill burden, or any other condition?
I think H. pylori, ma'am already said that high dose you can use as one of the option to improve your eradication rate. For GI bleed patient, we use double dose for 3 days. GI bleed, when somebody comes to you 3 days, you use double dose therapy. Sometimes for healing of this severe esophagitis, in some situations, a double dose has been shown to have a bit more extra healing rate compared to a standard dose. So in the setting of a severe esophagitis, GI bleed first 3 days, and sometimes as a therapy, one of the options, if somebody not respond to H. pylori, to initial 1 or 2 therapy. These situations, I would say high dose can be considered. Yeah.
The last question.
80 milligrams once a day.
No, 40 BD, you can make it 40 milligram twice daily.
Do not use single dose PPIs.
40 milligram twice daily.
There's a study showing that 20 milligram rabiprazole 4 times a day is better than 40 milligram twice a day or 80 milligram once a day. So giving the PPI more frequent is more important than giving a high dose at one time.
The last question.
Last question, please.
In view of resistance to the antibiotics for H. pylori, can we use levofloxacin-based triple-based therapy, triple drug therapy as a primary or a rescue therapy? Because the number of tablets to be taken is less in comparison to the bismuth-based medication.
So 4 drug therapy is always better than a 3 drug therapy. Whether you use levofloxacin or any other drug, 4 drugs is always better than a 3 drugs. And in patients where you suspect there is a resistance, bismuth-based is the best.
One more question: should we test for H. pylori in a patient with a normal upper GI?
That's what I just said, that testing without looking for a reason to test has no use.
Symptomatic, not responding to
yes, you can. Yes, you can. Yeah.
Thank you.
I think we are running out of time, and
Yes,
we have
one
got a
more
symposiums. coming up at from
Okay.
6 to 7.
Yeah.
2 symposiums are waiting. So
Sorry.
probably I think friend, you can ask. Any of the speakers. Just after this talk, I think it had endless number of questions. on this topic, and we can just keep on discussing it later on. Thank you all.
Thank you.
Thank you, speakers and chairpersons, for this excellent session. I have a few important announcements to make. The first one is, although we have ended the session here, Hall B and C are still running till 7 o'clock. 8 o'clock, we have a banquet dinner and musical evening. Kindly carry your badges with you. That is very important. Tomorrow morning, we start 8 o'clock here in Hall A for the CPC session. So in the booklet, it's mentioned in Hall B and C, so CPC is in Hall A. Tomorrow at 12 noon, there is an ISG award function in Hall E, that is upstairs. Kindly collect your ISG update book from the ISD counter. And today from 7 to 7:30 we have arranged alumni meet, and the institutes that have the alumni meet are CMC Vellore, AIMS, PGI and Santokh Durlabji Institute. So it would be the halls upstairs. Thank you so much.